ಯಾರಿವನೋ 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 ಇವನ್ಯಾರ ಮಗನೋ ಕಾಣೆ ನ ಕಾಣೆ ಹೆಸರೇನೋ ಹೆಸರೇನೋ ಯಾವೂರ ನಾಯಕನೋ ಜೊತೆಗಾರನಾಗುವನೋ ಕಾಣೆ ನನ್ನಾಣೆ ಎದೆ ಬಡಿತ ಯಾತಕೋ ತಾಳ ತಪ್ಪಿದೆ ಇಂದು ಇವನ ನೋಡಿ
ಇದೇನು ಸೂಚನೆ ಇದೆಂಥ ಯೋಚನೆ ಹೊಸದಾದ ದಿಗಿಲು ಯಾಕೋ ಕಾಣೆ ತುಟಿಲಿ ಕಂಪನ ಎದೆಲಿ ತಲ್ಲಣ ಬಹಳನೇ ಇಷ್ಟ ದೇವರಾಣೆ
ಯಾರಿವನು 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 ಇವನ್ಯಾರ ಮಗನೋ ಕಾಣೆ
Hello. Check, check. Hello. Good morning, everybody. Today's seminar, we are going to start immediately. Two things I want to convey. One is this is going to be live stream now. It's already on the YouTube as well as Zoom. ಝೂಮಲ್ಲಿ ಸ್ಟ್ರೀಮಿಂಗ್ ಆಗ್ತಿರೋರಿಗೆ ದಯಮಾಡಿ ನಿಮ್ಮ ಕಡೆಯವ್ರಿಗೆ ಹೇಳಿ ಎಲ್ಲರೂ ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳೋಕೆ ಹೇಳಿ ಅಲ್ಲಿ, ಸೊ ಡಿಸ್ಟರ್ಬೆನ್ಸ್ ತುಂಬ ಆಗುತ್ತಿಲ್ಲ ಅಂದರೆ. ಆಮೇಲೆ ಲೈವ್ ಇದು ಯೂಟ್ಯೂಬ್ ಲಿಂಕ್ ಕಳಿಸಿದ್ದೀನಿ, ಅವರು ಇಂಟ್ರಾಕ್ಟಿವ್ ಸೆಷನ್ ಇರಲ್ಲ, ಸೆಮಿನಾರ್ ವಾಚ್ ಮಾಡ್ಬೋದು ಅಷ್ಟೇ, ಎಲ್ಲಿದ್ರೂ ವಾಚ್ ಮಾಡ್ಬೋದು ಆ ಲಿಂಕ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು. ದೀಸ್ ಟೂ ಥಿಂಗ್ಸ್ ಆರ್. ಆ್ಯಂಡ್ ನಾವು ಐ ರಿಕ್ವೆಸ್ಟ್ ಆಲ್ ದಿ IGA members to come on to the dais. I welcome President Vinod sir, IGA President. I welcome Munagoda sir, Vice President IGA. I welcome Prakash Goda sir, Treasurer IGA. I welcome Agri Committee Chairperson Ramesh sir. And also I welcome today's speaker, Dr. Channabaswana Goda, onto the stage. I request Vinod sir to welcome Dr. Channabaswana Goda with a garland, flowers. As a formal inauguration of this program, today's program, by lighting the lamp by all the dignitaries, I request them to light the lamp for the inaugural session. Thank you for inaugurating this session, today's session. Once again, welcome to you all. Those who are not make their registration, please, there is a registration sheet. You can write your name clearly and the company name. So, now I request President sir to have opening remarks.
Welcome to all of you. Welcome to Dr. Channabaswana Goda and all the IGA members and my beloved field people. This session is basically intended for the knowledge about the biostimulants. We are all using so many biostimulants, but unfortunately the knowledge what we have about the biostimulants is very less. We do not know how it functions in the plant. We do not know when to spray, when not to spray. So these are all very important things. So IGA thought we should have a session on this, and I thank Lakshmish for recognizing Dr. Channabaswana Goda, a molecular biologist, and has good experience with Novozyme, who is a master in enzymes. So we thought we should make it a live stream also, because we cannot train everyone from IGA. So we are training basically the senior people, and I request the senior people to go to their offices and train the field staff over there in the offices, so that the knowledge can be transferred to the field. Transferring knowledge to the field is most important thing, and IGA is bent upon transferring the knowledge to the field so that the production increases and the cost decreases. So the live stream is also on, and people who are in the offices, please have good session of this. Please concentrate and note down the points also in the offices. Of course, you will be trained by the people who have attended this program. But if you have a basic knowledge, then it would be much easier for the people to train you. So that is the main reason why we arrange for this live session also. So thank you very much. Thank you one and all for attending this training session. I hope you take a lot of knowledge from this session. Thank you.
Thank you, Vinod sir. I just wanted to introduce Dr. Channabasavanagoda. I thought of you have. WhatsApp ಕಳಿಸ್ತೀನಿ. Overlay I can tell. About Dr. Channabasavanagoda. He is an agriculture postgraduate from US Bangalore in 2001. He later completed his PhD in plant molecular biology from Max Planck Institute of Chemical Ecology in Jena, Germany. After his PhD, he worked as postdoctoral scholar CNR Stansberg, France in 2008. Researcher at Copenhagen University 2012 at Copenhagen University, Denmark. During his tenure, he was awarded the prestigious FTP fellowship from Danish Science Council for his work on improving barley genotype for malting industry. As an academician, he has published 10 international peer-reviewed articles in plant biology. From 2013, he returned to India to head a R&D group at Novozymes and worked towards characterizing biostimulants for agri-farms 2018. He was commercial head of Asia Pacific head for CHR Hassan, including India, a Danish company, and was responsible for promoting biological and biostimulant sales in South Asia, including India. In 2022 onwards, he was given additional responsibility of animal nutrition along with plant health. From 2025 July onwards, he has started his own company, Lemma Biosolutions Private Limited, and is a managing director in this firm. He is currently promoting collagen-based products for animal health and serving as consultant for UK-based plant biostimulant company. Apart from work, he is a cultural artist and a great supporter of theatre and arts. He was folk dancer during his university days. He stays in Bangalore, married, and has 18 years old son who pursuing his UG in Southampton University. He also a avid trekker and like traveling and nature. This is about Dr. Channabasavanagoda. We heartily welcome Dr. Channabasavanagoda. The session is yours now.
Okay, thank you. To be honest, I'm very surprised today. Lakshmi told me that it was very small gathering, or I didn't expect that it was so big. But anyway, I am very, very happy to be here, and also very happy to share my knowledge, whatever I have gained or experienced all these years. So before I start, I'd like to thank President Vinod, Vice President Munegoda, Ramesh sir, and Lakshmi for organizing this, and also giving me an opportunity to come and talk to you guys. So this is not just, for me, this is not just one-way communication. It's also for me an opportunity to learn from you of how Gherkin cultivation is practiced, where biostimulants are fitting in this role, and do we really have a case that biostimulants are really worthful in Gherkin cultivation? Something that we need to, probably we can understand today here. So I want to keep this session very informal and I always like dialogues. I like questions and I like interactions. Many times I may not have the answer for everything, which is fine. We are all human beings, so we are all in the learning phase. But we can also find out, or we can make some good guess, or make some, we can anticipate what it could be the answers and all those things. But please, wherever you feel you have anything, please raise your hand, and I will be very happy. We'll exchange it. We are okay with the time. We'll be good with all this. We have divided this session into so the whole thing into 3 sections. The first is as discussed with Vinod sir and Lakshmi here. We are going to touch upon the fundamentals of biostimulants. So it's like, you have been talking a lot about biostimulants, but do we really know what they are, what they do, how they affect your crops? So that is something we will discover. It's a little scientific presentation, but I will try to be as much as clear and make it simple for you to understand that it is not only it's not very high level science in this thing. But as most of you are all in this field, you should understand probably how this works and what is the mechanism behind it. So that is what we are going to cover, and that's going to be the core of this thing. And then probably after that we have a coffee break, right? After the coffee break, we'll go into the little bit of application side. Whereas in the application side, we will try to address how to choose a biostimulant, what really goes into this. And this is very interactive. So we would like to understand more from you guys, whatever criteria we put towards, you can challenge us. And probably there are experts here, they can also give. And the idea is, at the end of the second session, is for you to understand, okay, what are the 4 or 5 points that are required when you have to choose a biostimulant, or you have to work with a growth promoter, or because they are not very discrete, they are all interconnected in many ways here, so that you will get an understand, okay, before I go choose a biostimulant, all these days, there should be a way to do that. That is my objectives for the second session. And finally, after lunch, probably we will have a panel discussion. We would like to all have the things which I am coordinating with Lakshmi, so where we can understand whatever we learned today, where are still the technology gaps, where are the commercial gaps, where are the knowledge gaps still in this one, and how we can address this so that you Gherkin society can really benefit from this. And one point of caution is, though I am associated with many companies, I am not promoting any product. So there will no be any name of any brand name in this session, because it is not in the interest of me to be telling those names here. So whatever product name, it will be a generic name, it will be a unique. So because I don't want to be biased with saying any company names or anything. Not audible? Okay. It's too much. Okay, so that looks like the session structure. Anything to add, Lakshmi, to the structure, or it's fine? It's fine. question, not gain any knowledge. That is my thing. There is no good question, there is no bad question. There is a question, that's all. So please don't hesitate. And sometimes in my experience, now I've been in this field for more than 25 years, I have seen the best questions coming from people who are very new to the subject. They ask one of the most brilliant questions. So, I think I'm also always keen to learn from you guys. So please ask questions, stop it. We will manage that time, don't worry. But we would like to have an interactive session, okay? Okay, so let's start. So the title I put is Fundamentals First. I know here everybody inherently is an agronomist in a way. Though you may be you are coming from some non-science background or anything, but after you worked with a Gherkin industry for invariably you become an agronomist. That is how I feel, because you are working in the field, you are seeing how crop is grown, what fertilizers are added, everything. So you are agronomist first. So the application part is fundamental, is very nice with you all guys. You know how crops are grown, everything. But I think then the background of how things work at the cellular level, molecular level is something where I think maybe we can help you with in this. So with that first, so the first session is to get into the fundamentals first. Ida. ಅಲ್ಲ ಇದಿದೆ ಆಗುತ್ತೆ ಇದು ದೊಡ್ಡ ಮಾಡಿಲ್ವಾ ಕ್ಲೋಸ್ ಮಾಡಿಬಿಡಿ ಎಲ್ಲ ಒಂದು Okay, so let's start with this. So this is my favorite slide, a little theoretical. What it says is the whole concept of agriculture. Light off madukku. Okay, it's better now? Okay, so the base, this is my, like I said, it's my favorite slide, is because if you have to summarize the whole of agriculture and you ask any good crop physiologist or anybody, they will put it, or even geneticists, they will put it into these three bubbles here, these three things. One is the yield potential of the crop. This is actually determined by your genes. It cannot be altered. So, for example, it is some effect of your molecular architecture of the plant that determines the yield. Now, if you have a variety which is like a 10 metric tons of yield per acre or acre I guess, and this is done in standard conditions in the breeder farms, and geneticists have worked on for 5-6 years, and they've said that, okay, this is the yield potential. That means that it cannot be changed. So if somebody claims in newspaper, sometimes I see some newspapers, some sugarcane, though it is like let's just say 120 metric ton is the yield potential. Somebody said, I got 160 metric tons of this. It's not a believable data. You cannot get more than your genetic potential. There is a cap to that one. So that is where, so if you put a variety, you can ask the breeder, we said, what is the maximum yield of this thing under your standard conditions? And if they say, look, on this Gherkin variety, we can get around 12 metric tons. Okay, so 12 metric tons. Believe me, that will be the end. No matter what you do in the field, you cannot get more than 12 metric ton. That is the simple definition of the yield potential per plant. Now, so what are the two things that are really defining this yield potential is how much tolerance your plant has for the stress, and how good is your fertilizer or input regime. So it's always the interplay between these two that will determine. Even if you will control the stress 100% and the fertilizers or as per the recommended dose, no change, everything is given at pinpoint, you can still yield only to the maximum yield potential to this one. So please bear in mind, one thing is when you are doing a gherkin cultivation, you have a limit for the yield. No matter how much good your cultivation practice is there, how many good biostimulants you add or good fertilizers you add, you cannot go more than the yield potential. So all we are working is, for you Gherkin people, this is called a zone of economic importance, where the plant is healthy and it starts stressing. This is where you play with fertilizers, you play with stress, you play with biostimulants, you play with hormones, everything. And there is a point, it's called point of no return. There, after that you don't, even if you do anything, nothing comes out. So it is gone. So this is the only zone where you have to work on this one. And stresses, I don't have to tell you, because you are dealing with stress every day. Every day it is your job to control abiotic stress, biotic stress, and environmental stresses, everything. And inputs, so many new innovations, so many recommendations, soil types. If you get these two, you will hit the yield potential. So the basic fundamental understanding for our next step. So everything starts with genetic potential and also ends with genetic potential in a crop cultivation. So with that, if you see what happens when you are, okay, if you see this triangle, yield potential is defined already. You don't have to do anything. There is genes, there is breeder, variety. Now you only have to worry about these two things. One is the stress. Yeah. Oh, alli adda idena. ಇಲ್ಲ ಸರ್, ನಾನು ಅಡ್ಡ ಇದ್ದೀನಿ ಅನ್ಸುತ್ತೆ, ಇವಾಗ ಸರಿ ಹೋಗುತ್ತೆ. ನಾವು yes please. No, it is fine. Yes.
Same area, like in terms of that's particularly taken same company, they're providing same number of those, same quantity of fertilizers, the climate is same, everything is same, but still fields are different.
Yes.
So what would be the reason behind?
Again.
That potential already defined with some people they get 20 tons. Some farmer. Some farmer will get 10 tons.
Okay.
Some other company will get 5 tons.
Exactly.
Same
Got it.
Same fertilizer management. Even that they get lesser, or there is a variation in the game.
Yes. Again, these two, there, see, even the guy who is getting 20 tons, he might not have hit highest potential still. Still, it could be 25 is the potential. But he has managed the stress and fertilizer very well. He's gone close to the potential. So, and the one who is 10 metric ton, there the stress and fertilizer inefficiencies are so pronounced that it has pulled down the genetic potential of the crop completely. So if you say, okay, somebody has got 20 tons, you should check with the breeder. They will say exactly, in my conditions, what was the highest yield they got. Theoretically, it is impossible to get that yield in the field. You can never get it, because it is so much of factors that is there. So that is why they are saying that if you don't, it's the same variety, two different environment, means you are not managing the stress and the fertilizer efficiency in the most optimum way. That is the only reason. Yes, Ramesh? Okay. Neeru, one bottle. Ah, it's okay. Keep it. Let's sit. Sit. ಹೌದಾ? ಹಾಂ, ಓಕೆ. ಅದು ನಮಗೆ ಫ್ಲೋ ಅಲ್ಲಿ ಹೋಗ್ತಾ ಇರುತ್ತೆ. ಮಾತಾಡೋಣ. ಹೌದಾ? ಹಾಂ. ಓಕೆ. ಸೊ ಇದು ಕನ್ನಡದಲ್ಲೂ ಮಾತಾಡಿ ಯಾವಾಗ ಅಂತ ಹೇಳಿದ್ರು ಅವರು. ಸೊ ಇದು ಬ್ರೀಫಾಗಿ ಹೇಳಬೇಕು ಅಂದರೆ ಕನ್ನಡದಲ್ಲಿ ಇದು ನಿಮ್ಮ ಇಳುವರಿ ಇದು ಏನೋ ಇದಿದೆಯಲ್ಲ ಪೊಟೆನ್ಷಿಯಲ್, ಅದು ಜೆನೆಟಿಕಲಿ ಡಿಟರ್ಮಿನ್ ಆಗಿರುತ್ತೆ. ನೀವು ಏನು ಮಾಡಿದ್ರು ಬ್ರೀಡರ್ ಎಷ್ಟು ಹೇಳ್ತಾರೆ ಅದ್ರ ಮೇಲೆ ಏನೂ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಸೊ ಇದ್ರದ್ದು ಇದೇನಂದರೆ ಸಮರಿ, ಸ್ಟ್ರೆಸ್ಸನ್ನು ಎಷ್ಟು ಟಾಲರೇಟ್, ಎಷ್ಟು ಕಂಟ್ರೋಲ್ ಮಾಡ್ತೀರಾ, ಆಮೇಲೆ ಎಷ್ಟು ಆಪ್ಟಿಮಮ್ ಆಗಿ ಫರ್ಟಿಲೈಸರ್ನ ಯೂಸ್ ಮಾಡ್ತೀರಾ, ಅದ್ರ ಮೇಲೆ ನಿಮ್ಮ ಕ್ರಾಪ್ ಜೆನೆಟಿಕ್ ಪೊಟೆನ್ಷಿಯಲ್ ಆಚೆ ಬರುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ವಾಟ್ ಈಸ್ ಆಲ್ ಅಬೌಟ್ ದಿಸ್ ಸಮರಿ. ಆ್ಯಂಡ್ ಇಫ್ ಯು ಸಿ, whenever you plant a gherkin, from day one, it is already under stress. In India or everywhere, maybe in Europe it is little stress. India, please note that there is always a background of stress. Stress ಇಲ್ಲದೆ ನಿಮಗೆ ಯಾವ ಕ್ರಾಪು, ಎಂಥ ಒಳ್ಳೆ ಮಾನ್ಸೂನ್ ಸೀಸನ್ ಏನೇ ಇಲ್ಲ, ಸ್ಟ್ರೆಸ್ ಇಲ್ಲದೆ ಸಾಧ್ಯನೇ ಇಲ್ಲ ಬೆಳೆಯೋಕ್ಕೆ ಇಂಡಿಯಾದಲ್ಲಿ. ಆ್ಯಂಡ್ ಇಫ್ ಯು ಟ್ರೈ ಟು ಲಿಸ್ಟ್ ದ ಸ್ಟ್ರೆಸ್ಸಸ್, ಪ್ರಾಬಬ್ಲಿ ಯು ಕ್ಯಾನ್ ಲಿಸ್ಟ್ 100 stress, minimum 100. Many of them we don't even know. Okay, so there is also wind stress. ಎಷ್ಟು ಜನಕ್ಕೆ ಗೊತ್ತು? There is wind stress. Bright sunlight, very good photosynthesis. But do you know how much UV is there in that? That is also a stress. So even you need sunlight, very good sunlight. The sunlight in Turkey is different from in say Tamil Nadu. So photosynthesis. So there are hundreds of stresses, and you cannot manage everything. You have to live with this. Please note it. So that means that whenever there is a stress, or you put an input like a fertilizer, the molecular response starts. That is, plants start stimulating. Beautiful sensors are there on the cells. They will understand, oh, this is the clue from fertilizer. This is a clue from hormones, soil. They are so beautifully programmed. They will sense it. That is where genes start expressing, proteins come out, calcium signaling happens. All this is fundamentally driven by the phytohormones or plant hormones. Plant hormone is the fundamental for your, either it is gherkin or it is corn or any crop. Without plant hormones, it goes. It goes human for humans also. What are we? We are made up of hormones. If I'm happy, it's because of my happy hormone. If I'm sad, it's because of this one. Microbes, everything, there is this hormones start regulating a lot. So hormones kick in, whether it's in seed, seedling or anywhere, hormones kick in and hormones regulate it. Then they will determine how much to invest in stress response, how much to invest in the fertilizer response. So you have to get these two correct with your inputs so that the response is correct, and then you will see a improvement in the yield. And your objective is to go as close to the genetic potential of the crop. How many of you, my question is, do you know the genetic potential of your varieties in Gherkin? Have you discussed with Rizwan or any other seed providers? What is the standard condition? What could be the yield that this can give? Any idea, anybody? Well, currently, what is the yield, sir, huh? Okay. Per acre. Okay. But
There's a difference between bigger size.
Highest yield is around 25 tons. Highest.
where is that? Where is that, sir? In India? Now, the same variety, if it is put in Netherlands, what would be the yield? That is also not the real genetic potential of the crop. That is also not, because even in Netherlands there is some stress probably, because you cannot define what is the real stress like that. So because in the US, the corn yield is 4 times than the Indian corn yield. Same Cortiva varieties in Indiana, Iowa, and all these places, they get 3 to 4 times more yield. How do they? And because they are planting close, and there it can tolerate even the close planting. So that genetic potential is very enigmatic. We don't know where it is. So That is where it is. So if you manage this, you are always going to the closer to the genetic potential. Okay. Now coming to the plant hormones. Though it's a biostimulant thing, but I cannot get away without talking about plant hormones. Plant hormones are the key. So if you know plant hormones, we always feel that they are responsible for only growth, development. They play very, very big role in the stress response. Now you see here, the same hormones I have listed here. They also play a role in stress response, and the same one they also play a response in fertilizer. And if you look at my triangle here, this is for the stress, this is for the fertilizers here. And the same hormones play a role here. So they can play dual role. In their stress, they play one role, and then they play another role. Example, ABA. When stress comes, they can close the stomata. They can induce, they can induce drought tolerance. But also when you have abundant nitrogen, the same ABA can also improve nitrogen assimilation in your crop. So that was, it starts with this thing. So, and auxin, for example, root response. But also auxin improves nitrogen assimilation. It is required. If you induce a lot of auxin in your plant and there is no nitrogen, the apical dominance will not happen. It requires nutrients to be drawn. So they're all interlinked in all these ways. And cytokinin, photosynthesis it affects, it also affects grain yield. Brastinosteroids, you see it can protect cells and improve metabolism, and they also indirectly help root and shoot growth. Salicylic acid, very, very neglected hormone. It is produced in all the plants. It is little difficult to detect, but especially fruits and vegetables, salicylic acid plays very, very big role. It plays a role in growth. It also plays a role in pathogen resistance, not insect resistance, but pathogen resistance. Whenever you have a downy mildew, you will see invariably salicylic acid improved in this one. So the point I'm trying to make is that stress tolerance inputs are interrelated. They are the only key things that determine your yield. You focus only on those things, and this is controlled by hormones to a great extent. The summary till now. It's cell division. It is involved in cell division. Okay, but maybe see every hormone in higher concentration has an inhibitory role. Okay, sir. So now, if you say auxin, auxin is the hormone which is involved in, any idea what is the first term we get when you use auxin? It is apical dominance. Apical dominance, we see wherever there is a shoot tip or a root tip, auxin is produced. And then when auxin is there, it starts mobilizing nutrients from the zone, from the root zone to the points. And then they start elongating towards the growing points. Now, when auxin decreases, then you get gibberellins, and cytokinins and gibberellins. They are in the lateral development. Imagine like this: anything you want to go tall is auxin. Anything you want to go sidewards is cytokinins and gibberellins. Gibberellins has a dual role. When there is flower development, shoot development, flower buds development, gibberellin kicks in. So these three, auxin, cytokinin, and gibberellins, they are growth promoting. Basically, they can see as growth promoting auxins. Abscisic acid, and there is one more called ethylene, which I have not listed here. They are stress response, that they always try to bring down the growth. Why is because they want to go into flowering. They don't want to invest too much into this thing. Ripening hormone. Ethylene is a ripening hormone, but ethylene also kicks up when the plant have to senescence very fast. Now, for example, in your gherkin, some reason for 3 weeks you stopped watering, or water stopped, they'll start kicking abscisic acid, ethylene. They'll try to senescence. Leaves start to dry, and then they start to produce whatever fruit they can make. See, for you, your objective is to get 15 metric tons per acre. But for plant, the ultimate objective is to get at least one seed from the plant, because it has to survive. So what it will do is, it will kick in the ethylene pathway, it will initiate the senescence, That means they start even making small seeds, and then they will go into senescence. Very interesting study in the US, soybean. And once there was some severe drought in the Iowa region where it is a soybean growing area. And scientists said, okay, it's not gone, I think gone, crop is gone now, because there they don't use any irrigation. There is so much, but some reason that day it was very drought. And after that, the plants were dried, completely dried. And they went to the field to see what is the response to this one. You know what interestingly they found? They found that every soybean plant had at least one pod and at least had one seed in that pod, though it was severe drought. So that tells you that the plant's objective is to make at least one offspring to the next generation. So they will play very, very cleverly with your hormones. You have to be smart not to give plant the chance to sense the drought, not to sense the salinity, not to sense any other abiotic stress. Otherwise, they will kick in into this one. So, adu sir, ಈಗ ಹಾರ್ಮೋನ್ಸ್ ಅಂದರೆ ಆಕ್ಸಿನ್, plant growth, apical dominance, root. If you see any plant which is really grown deep inside it, very likely there is auxin into this. Auxin metabolism is very good. Or the plants which are really growing tall is auxin. Side shoots ಎಲ್ಲ ಬರ್ತಾ ಇದೆ ಅಂತಂದರೆ ಸೆಲ್ ಡಿವಿಷನ್, ಸೆಲ್ ಲ್ಯಾಟ್ರಲ್ ಡಿವಿಷನ್ಸ್ ಜಾಸ್ತಿ, ಜಿಬರ್ಲಿನ್ಸ್ ಅದು ಜಾಸ್ತಿ ಇದಾಗ್ತಾ ಇದೆ. ಇದು ಗ್ರೋತ್ ಅಷ್ಟೇ. ಆಮೇಲೆ ಸ್ಟ್ರೆಸ್ಸಲ್ಲಿ ಯು ವಿಲ್ ಸಿ ಆಲ್ ಎಲ್ಲ ಆಕ್ಸಿನ್ ಕಮ್ಮಿ ಇರುತ್ತೆ, ಸೈಟೋಸಿನ್ ಕಮ್ಮಿ ಇರುತ್ತೆ, ಜಿಬರ್ಲಿನ್ ಕಮ್ಮಿ ಇರುತ್ತೆ, ಬಟ್ ನಿಮಗೆ ಏನೋ ಇದು ABA ಅಬ್ಸಿಸಿಕ್ ಆ್ಯಸಿಡ್ ಆ್ಯಂಡ್ ಎಥಿಲಿನ್ ಜಾಸ್ತಿ ಇರುತ್ತೆ ಸ್ಟ್ರೆಸ್ಸಲ್ಲಿ. ಸೊ ದಟ್ಸ್ ಅ ಫಂಡಮೆಂಟಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್. ವಿ ಕ್ಯಾನ್ ಗೋ ಡೀಪ್. ಇಟ್ಸ್ ಅ ಬಿಗ್ ಓಷನ್ ಆಫ್ ದಿಸ್ ಥಿಂಗ್. ಓಕೆ, ಎನಿ ಕ್ವಶನ್ಸ್ ಸೋ ಫಾರ್? ಗುಡ್. ಯಾವ್ದು? ಹಾಂ, ಹೋಗುತ್ತೆ. ಹ್ಞೂ 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 ಹೌದು ಹೌದು. ಹ್ಞೂ ಹ್ಞೂ ಲಾಂಜಿವಿಟಿ ಮೇಲೆ ಹೌದೌದು. ಓಕೆ, ಹಾಂ, ಓಕೆ ಸರ್, 100% ಆನ್ಸರ್ ಇಲ್ಲ ನನಗೆ ಇದರಲ್ಲಿ. ಬಟ್ ನಾನೇನು ಹೇಳ್ಬೋದು ಅಂದರೆ ಎನಿ ಗ್ರೋತ್ ರೆಸ್ಪಾನ್ಸ್ ದಟ್ ಈಸ್ ಇಂಟರಪ್ಟೆಡ್ ಬೈ ಸ್ಟ್ರೆಸ್ ವಿಲ್ ಹ್ಯಾವ್ ಎಫೆಕ್ಟ್ ಆನ್ yield. ಓಕೆ, ಲಾಂಜಿವಿಟಿ ಕಮ್ಮಿ ಆಗುತ್ತೆ because plant will always try to finish its life cycle soon as possible. They will, this one. In that case, what you will see is, you will see small fruits, even bitter fruits. Bitter fruits ಯಾಕೆ ಮಾಡುತ್ತೆ ಅಂತಂದರೆ, they will, instead of putting more sugar in the fruit, they will put more secondary metabolites. I don't know, in cucumber it is possible, in gherkins I don't know. You will feel very bitter fruit. Bitter ಯಾಕೆ ಅಂತಂದರೆ, they don't want anybody come and eat the fruit and disperse the immature seed. ಹೋಗ್ಬಿಡ್ಬೇಕು ಬರ್ಡ್ಸ್ ಎಲ್ಲ, and then they put sugars into that, so that it's all evolutionarily longevity will be affected. Stress will always bring down the longevity. It will shorten the cycle. Okay. Sir.
Hormonal imbalance to get rid of this?
Good question, sir. Yes and no. Answer is yes and no. Yes andre it's highly programmed. You cannot do much. Andre you will see stress after 4 days, but the plant will see stress within hours.
So what is the remedy?
It's too late already for you to do anything in that one.
There is no remedy for this?
There is remedy. You have to have solid markers. You have to have, nowadays with the precision agriculture, you have to invest in precision agriculture. Sir, you have to, if you are really Gherkin, you have to invest in precision agriculture, where you will understand that soil is drying after 4 days, but now there are equipments within 4-5 hours, 6 hours, they will give you real-time indicators. If Gherkin, that way you can manage. In that case, you have bio-stimulants. You have, if you want to have, now you know it is going to go into stress, you have some inhibitors that can control the abscisic acid, like malic hydrazide is there. You can spray that and then you can bring it. My only thing is, it is programmed, but you have to be very fast in sensing. And with the current conventional things, you cannot sense it faster than the plant.
So you said precision agriculture, no?
Yes.
So can we do it something with regeneration agriculture?
No, not regeneration. This is mechanical. What I am saying is, you have weather station, you have soil, this thing. For example, you don't have to have a very 1 acre, 4 or 5 soil sensors, and every day you are seeing it in your app. The moment you see suboptimal moisture, you give an alarm in your phone, and then you do some critical irrigation. Avoid the stress, because once it reaches the tipping point, like he said, fruit size will come down. Everything will be reduced already, and your yield will be affected. So the summary is, sense your stress as soon as possible. ನೀವು ಎಷ್ಟು ಬೇಗ ಇದನ್ನು ಮಾಡ್ತೀರಿ, ಇದು ಐಡೆಂಟಿಫೈ ಮಾಡ್ತೀರಿ ಸ್ಟ್ರೆಸ್. ಇಟ್ ಕುಡ್ ಬಿ ಸಾಯಿಲ್ ಸ್ಟ್ರೆಸ್, ನ್ಯೂಟ್ರಿಷನ್ ಸ್ಟ್ರೆಸ್, ಇನ್ಸೆಕ್ಟ್ ಸ್ಟ್ರೆಸ್, weather stress, 4 days cloudy forecast. That is the key. It goes same for humans also, right? Your disease is cured when you diagnose it correctly, faster. That's it.
Yeah.
That's how it is. Yes.
Thank
so,
you.
Yes and no. Yeah. So this is the little summary of the plant phytohormones. I'll tell you why I said, because it's a fundamental. Hormones are the fundamental. You take biostimulant, fertilizer, you cannot bypass hormones. You have to go through the hormones only in your plant response. Okay, now coming to our favorite subject, biostimulant. So maybe I can ask you, what are the definitions of biostimulants? What is that you have heard about biostimulants? Anybody? Just, it's okay. I mean, I want to know what is the level of understanding of biostimulant in this one. We all been using, I know you are using humic acid, seaweeds, protein hydrolysates. So I don't know what to use, sir. But any guess, what is biostimulant in your opinion? Growth promoter. Okay, good. Any other keywords at least, if you say? Okay, sir. Yield enhancer is there. Okay. Very good. Resosphere, good. Disease tolerance, excellent. I like that answer. Excellent. See, all this is there here in this. Yes, I think your understanding is fine, but it's not just one. They do multiple rules. Okay. Number one, they are not fertilizers, biostimulants. They don't add nutritive value to the plant, except few. Example, some seaweed, they have some zinc, magnesium. But most biostimulants, they don't add any nutritional value. If you say, I can reduce my fertilizer using biostimulant, wrong, boss, you are wrong. It's not. Don't do that one. Please follow your fertilizer regime as strictly as possible, no matter what people recommend you, whichever the biostimulant they want to use, you use it, but don't stop the fertilizer regime. So that, but what it does is, when you have a fertilizer, it optimizes the fertilizer efficiency. It has transporters. It can bring the fertilizer from the root to the growing tissue. It can make better utilization of it. It makes cells more responsive to use the fertilizer. So ಏನಾಗುತ್ತಂದ್ರೆ ನೀವು ಫರ್ಟಿಲೈಸರ್ ಇರೋದನ್ನ ಚೆನ್ನಾಗಿ ಇನ್ನೂ ಚೆನ್ನಾಗಿ ಏನಾಗುತ್ತೆ ಅದರದ್ದು ಯುಟಿಲೈಸೇಷನ್ ಎಫೆಕ್ಟ್ ಮಾಡುತ್ತೆ. ಓಕೆ, ಸೊ ದಟ್ ಈಸ್ ಒನ್. ಫಸ್ಟ್ ಈಸ್ plant metabolism, stimulate ಮಾಡುತ್ತೆ, because it is giving lot of enzymes, lot of proteins, lot of energy molecules, everything. So it is like, it's, for example, now we are taking some supplements, right, every day. So what do supplements do? It is like helping your digestion, helping your muscle mass build. It is like stimulating your responses. Kids go to gym. I know youth also go to gym. After that you take protein supplement. Why? Because your gym is already made a lot of lactic acid, it has made a lot of ATP, it is building your muscle. But the protein hydrolysates or the whey protein gives you the essential amino acids. It stimulates muscle building faster. Similarly like that, this metabolism of plant metabolism is improved by this stimulant. Crop yield and quality, I'll come to that. How many beautiful pathways are they biostimulant, they stimulate? Color, flavors, texture, fruit softening. But you should know what is that exact biostimulant that can do. That's why they improve the quality and yield on this one. And impact of abiotic stress. For example, some biostimulants can reduce abscisic acid. They can reduce ethylene. They'll say, no, plant, don't induce it. So they have, like, they can block it. That's why they will withstand it. So then they enhance biological activity in the soil because there's so much carbon, there is so hydrogen, there's no nitrogen in this one. Microbes, they love this thing. So they also, so like all you said, you had these things, they do multiple roles depending on one. There is not like one biostimulant can do all this. There is no such biostimulant in the world till today that can do all the roles. But you can combine many things to get a good optimum disease. So this is how it is. Just like how phytohormones regulated stress and fertilizer use efficiency, biostimulants will also do the same job, but in a different way. To put it, phytohormones are very specific. They are very, very programmed. They know, okay, this I just act on abscisic acid and I will do on ethylene. But biostimulants are more generalistic. They act on many pathways here and there. So it's very generalic response. It is not like, for example, you have body pain, you have fever, you have a headache, you can take a paracetamol. But if you have something different, like a specific blood clot or something, then you need to take a specialist. That specialist is phytohormones. Whereas biostimulant is like a paracetamol, you can say. They can do a lot of things, and they are not very highly potent like phytohormones. They can also stimulate phytohormones indirectly. So what they will do it is both phytohormones and this, optimize your fertilizers, reduce your stress. That's what they do. And yes, what you can, yeah, this is one. You can consider they are very generic compared to phytohormones. So that's our definition of biostimulant. There will be a lot of definitions, don't worry about it. You just have to understand that they do a lot of things: stimulation, metabolism, activation, defense elicitation, nutrition efficiency, soil, everything, and your understanding is correct like that. Okay. Okay, now we will deep dive into each of these things. Okay. So now first is your favorite, right? I think many of you have understood what is a humic acid. Right? Any idea what is the source of humic? So anybody in this hall, yaradru, even from the audience, where is humic acid coming from? Anybody? We all used humic acid. Huh? So where is the humic acid coming from? Huh? Terrestrial soils. Okay. A little deeper. I also gave the hint. Absolutely. Okay. So, so if you go to Dubai, or if you go to Morocco, or you go to Turkey, anywhere where a lot of fossil fuels are there, but they are not lucky with the oil, but they are lucky with other things there. So if you go deep, first, I think in the end you get coal, and after the coal you get something else, and then you get humus material, and then you get lignite, and then you get peat, and then you get your soil. So there is this fraction. As you go down, the carbon content increases, become very thick in carbon. And as you go up, carbon decreases, oxygen, methane groups, sulfur groups, all functional groups add. It comes like that. So you are mining everything and then removing only that fraction just before the coal or something. Take that one. That is your humic acid. Now, how many companies fractionate that effectively? What is the humic acid you are getting? Is it containing lignite in that one? Is it containing coal fraction in that? I am not here to judge, but there are a lot of products in the market which are not pure humic acid because the extraction procedure is not easy. Okay, sir. You are testing me now? Okay. I'll give that, sir. I think next, can you note that question, sir? Okay, so impurities in the thing, because I have to be very specific because I cannot give you any answer. It should be usually lead, heavy metals, arsenals are all there. So we will, I can give you some publications where I will provide you some lead in it. Maybe I'm guessing in that coal, what is there? Mixture of petroleum products, some plant materials will be there. Undecomposed materials are there, and if you don't take it out, there will be peat. Soil is also there. So all these things. So we have to be. I'll give you the precise details for what is the criteria for a. Yes, and I don't know how much, maybe some companies claim, some companies do not claim. That is one thing. Second thing is solubility is also very issue, So not all humic acids are soluble in all the pH. I'll come to that maybe I have more slides in the next slide. So when you say humic acid, you should please remember there are three components to this. One is the humic acid, then there is a fulvic acid, and there's this one is the humin. And you can see that humin is a very big, large molecule, followed by humic acid, and then fulvic acid is very small molecule. Second thing is the amount of oxygen in this is fulvic acid has the highest oxygen compared to all the three. Okay. And then when you say oxygen, what is the first word that comes to your mind? Any guess? Respiration is good. Close. Photosynthesis. No, it requires more carbon dioxide and water in that one. Okay. So any molecule which is having oxygen is reactive. Yes, there you got it. It's reactive. That means that it can bind to anything. They will catch it because everybody is affinity to oxygen. They will bind. So that is good for plant. You need something which is binding to heavy metals, toxins, other chemicals, proteins, carbohydrate. So fulvic acids has that. So general rule, maybe it is not 100%. Fulvic acid is for foliar. F is F. Remember that. Humic acids for soil. This is generally, but you can also add fulvic acids to the soil also, and else one. But generally, because of the reactivity, fulvic acids, pure fulvic acids, are usually sprayed on the leaves and foliage, whereas the humic acid, because of this heavy thing, they are usually added into the soil. It is how it is. But there is no harm if you do different things depending on all these things. So they are different in color. One is very, very dark brown is humic acid. If you say light yellowish to whitish color, it is fulvic acid. It is purified fraction. And it's not an easy fractionation. They have to do a lot of distilling to remove humic and then fulvic acid and all these things. And then humin is also, it's very, very dark in color. Humin is mostly used in the soil. So please, when you take a bottle next time, you see the label. You read it and see whether what is it. Is it humic acid, fulvic acid or humin? What percentage is it? If it is say 60% humic acid, only 5% fulvic acid, and if you are spraying on the foliage, you wasted money. You just wasted money because it's not going to be effective. It's not reactive. It should be better to this one. They will not recommend. So just please have that this thing. Okay. And they are having, because of this, heavier the molecule, less is the oxygen, general rule of thumb. So you see these are the brown color, and the more darker it is means more carbon is there, simple. More lighter means carbon is replaced, that is the way. So that's it. Now let's dive into how these humic acids function. And I would like to spend some time because this is very important for you to know how they do this. Okay, before that, where did I go? Okay, before that, these are all the categories of biostimulant. I think the one slide jumped here, okay, but it's not a harm. So at least some 8-9 categories are there, okay. But maybe I can ask you guys before we go here, how many categories of biostimulants we know? Can we just throw around some name? We have seaweeds, humic acids, other than that, protein hydrolysates, then? So still 6 are there, sir. Okay then. Seaweeds. Yeah, yes, okay. So, okay, let's go. So, there are botanical extracts. In that category, seaweeds come. That is the first one. Maybe I have this. Yeah, here. So, seaweed extracts andre I'll go to that seaweed where it is. It all comes under general category, botanical extract. Then biochemicals. These are synthesized. Label ಮಾಡಿರ್ತಾರೆ. Methyl group, methyl hydroxide. They have some affinity to stimulate some water things. This is most recent and very young field is biochemicals. And this is going to be the future next. In next 5 years, all the seaweeds, everything will be outdated. All these nano biochemicals will come into the market. Be ready for that. Where you spray very, very minute quantity and you can get the best benefit. From bulkiness, you are getting into nanoness. Next 5 years, be ready. A lot of good research has happened. BASF, Bayer, even UK-based companies, Indian companies are also doing. UPL is also working on all those things. Nanourea is also a biochemical, but that is a delivery agent. But here molecules are itself made like this one. Example, some esterified methyl group compounds like that, which are analogs for abscisic acid inhibitors. But you don't have to spray in so high quantity, just minute, and the delivery is very fast. They go into the plant and they affect in this one. So biochemicals are going to keep an eye on all the publication, what is coming in biochemicals in this one. Vitamins are also used, all types of vitamins, especially when you have growth. Vitamin is also a biostimulant. Protein hydrolyzed and amino acids, you said. So that means you take a crude protein, you either add acid or you add enzyme, they break down into peptides. If you hydrolyze it further, they break down into amino acids. Both these fractions are used. And they are also, I'll tell you what they do in this one in the next slide. Anti-transparent, sorry. Anti-transparents. Have you heard of this before, anybody? Do you use any anti-transparents in Gherkins? Any idea what anti-transparents do? It's transpiration. Transpiration is the removal of water from the root. It is going through the leaves here. On a very dry, sunny day, more moisture is removed. But you need transpiration. Without transpiration, nutrients cannot be solved, brought from the root to the growing tips. But it comes at a cost. That water is also gone. But you can reduce the water content. That is, anti-transparents are used. Okay, they are transpiration. Water movement of water from the root to through the leaves, openings, stomata, leaf tips, water goes away. That they have to reduce, especially in drought areas. And antioxidants. Whenever there is stress, lot of reactive oxygen species come. Then you need, that is also this thing. But another famous is the cell-free extracts. Like you said, cell-free extract is, you kill the bacteria, but bacteria, everything they produce, they throw out of their cells. Sir, cell-free extract is cell-free microbial products. Means any bacteria which is growing in media, they produce lot of beautiful chemicals or metabolites. But they don't keep that metabolites inside the cell. They throw it outside. And the reason they do it is because they don't want any other fungus to come close to them. Yeah, all Pseudomonas, Bacillus, most of them have a cell-free extract. Polysaccharides, they sell outside. We researchers, we have collected all those. And then we dried the water, made that powder and gave it to Gherkin growers. Gherkin growers, Trichoderma is good. So one is cell-free extract. The second one is microbe itself. That is microbes. Microbe itself, PG or PGPR, plant growth promoting rhizobacteria, or plant growth promoting microbes, PGPM. Lot of names are there. So live bacteria is also a biostimulant. And finally is our favorite, the fulvic acid and humic acid. So these are some of the general terms. So how many of you have used more than 2 here in your crop cultivation? Okay. Okay. Have anybody heard about calcium carbonate, calcium oxalate, all these chemical like chitosan waxes, everything that can be sprayed on the, especially broad leaves like gherkin, where maximum transpiration have. Sir, I'll share this slide and also I'll give you all the publication. So there are some good chemicals, which cyclobutrozone. No, it is not suppressing growth. For example, your yield, for example, what do you say? If you say 10 buckets of water, or even you say 7 buckets of water, the yield is not affected. So that means you can save water here. Say in Tamil Nadu or in some North Karnataka, there is severe drought. Plant response is taking a lot of water from the root, throwing it away outside. Means it is drawing a lot of nutrients, but that much of water is not required to waste from the plant. It is physiologically it is made. So you add this spray, calcium oxalates, and then chitosan waxes, everything. So they will, they will stick to the leaf. They will reduce the water evaporation. So you are drought. Silicate. Yeah, there are all different morphology, chemical combinations. Salicylic acid. Yes. Yeah, they have all good research, but I can tell most European companies have this. American companies have this anti-transparents in this one. But I think it's high time we should try that. Where is it? I don't know. I don't know the real answer whether what is effect it has on the gherkin. Maybe I don't know, but I just listed the general thing. But we can find it out. Why do you think potassium is bad for gherkin? Sulfate. Sulfate. Acha. No, I don't know about gherkins in general, but we can find it out. I think. Yeah, you are right. Yeah, exactly. No, these are all lists. That's all. You have to talk with the manufacturer. They would have tell, okay, this is good for cucurbits. This is not suitable. This is for solanaceous. They will tell you. That is why talking to manufacturers is very key here. Otherwise, you are like taking your going to the counter and taking whatever medicine you want. It is don't do that one. Just do some research a little bit on what crops they have researched, what effect they have. You can ask them, do you have any research? You are all progressive growers. You are experienced in this and you are having high value crop which is exported to Europe. You should be seeking a lot of scientific data from manufacturers is what my advice is. Yeah. So this is, sir, this is all the category. There is not any more category, anything which usually fits into this one, in this category. Okay. Humic acid I said. Now, there are 3 ways where humic acid help in this, since we are all using humic acid. First mode of action. Now, what happens during drought stressor, like drought stress or salt stress, for example. You see here, this part is like a salt stress. You see lot of salts get accumulated. And what do? The salts are blocking the roots. And water can actually is repelled. They don't really like to go through those things. So, though there is water in a saline soil, the water is not available because salts are accumulated in this. Now, you are adding these humic acids, huge. Humic acid is a charged molecule. It can bind to positive charge, negative charge, cations, anions like this. And the more you add into the soil, what they do is, they bind all the salts, sodium salts, bicarbonate salts, everything, and they free up the roots. So whatever water is available, they will take it up and they will thrive in this. So first mechanism is that they have very, very high affinity, humic acid, to bind to heavy metals and salts in the soil, and it frees the roots. And if you ask why people are adding this humic acid to roots, this is one of the first reasons, is it can bind to lot of heavy metals and salts in this one. Okay, that's one thing. Now, I have one question for you guys. Now, if humic acid and fulvic acid are so reactive, and they can they also bind nutrients and make them unavailable for plants? See, they can bind to anything, and nutrients also, like potassium is a positive charge potassium. There is phosphorus, magnesium plus calcium, everything are charged nutrients. Whatever nutrients you add, they have to become ions, then only plant absorbs. Similar to sodium, humic acid can also bind all the nutrients, right? Yes or right? What do you think? The answer is yes. They will bind. They will bind to all the nutrients also. But it is a good thing for us, because the good thing about humic acid is the binding is exchangeable. It's not a permanent binding. Okay, so instead of in the soil's condition, nutrients getting leached out, humic acid help actually sequester them. In way, this is very good, right? That means you are keeping all the nutrients close to that, and then when the moisture is right, then dilution happens, then this exchange, they can be exchanged. So, again, it depends. See, it's a tricky question, but how much of salt is released, how much of fertilizer is released is a question. And we think that based on the research, fertilizers are small ions, they are released faster, whereas the salts can be hardly bound. And it depends on the molecule. If there are 3 or 4 bind sites, it's very toughly bound, whereas nutrients are small ions, they can be released faster. That's the case. So, yes, the answer is true that they can bind both nutrients as well as this one, but it also make nutrients bioavailability. Looking at the positive side of this one is humic acid, this, and it is not like permanently binding. Plants can extract it also from somehow it is, and you have good moisture. No, it's not fixation. It's not fixation, but based on the number of reactive spots, if it is a tightly bound thing, then it will be tightly bound, otherwise it will be thing. Sir, coming back to your question, yes, it binds to sodium salt, but it is good. It is sequestering all the sodium salts towards the humic acid, and it is freeing the water for this one. After that, it may release again. Let it be. Sodium salt you cannot remove, but what you are doing is you are making the roots less accessible to salts using humic acid. It's like that. But they are also released, but temporarily they will bind and release. That will happen. It is like a filter. Yeah. So, not very clearly established mechanism, but the reactivity of humic acid is that they can do it. So, Kannada okay. So, ಕನ್ನಡದಲ್ಲಿ ಏನಾಯ್ತು ಇದರಲ್ಲಿ ಏನಾಯ್ತು ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಬಂದು ನಮ್ಮಲ್ಲಿರೋ ಈ ಉಪ್ಪಿನಾಂಶಗಳು ಆಮೇಲೆ ಈ ಬೇರೆ ಬೇರೆ ಟಾಕ್ಸಿಕ್ ಕೆಮಿಕಲ್ಸ್ ಏನೇನಿರುತ್ತೆ ಅದನ್ನೆಲ್ಲ ಹಿಡಿದಿಟ್ಕೊಂಡು ಬೇರುಗಳನ್ನೆಲ್ಲ ಫ್ರೀ ಮಾಡುತ್ತೆ. ನೀರಿಗೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ಗೆ ಅಬ್ಸರ್ವ್ ಮಾಡ್ಕೊಳ್ಳೋದಕ್ಕೆ. ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಇಲ್ಲ ಅಂದರೆ ಏನಾಗುತ್ತೆ, ನೀರು ಉಪ್ಪಿರೋದ್ರಿಂದ ನೀರು ಬೇರೆ ಹತ್ರ ಬರಲ್ಲ. ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಏನು ಮಾಡುತ್ತೆ, ಉಪ್ಪನ್ನೆಲ್ಲ ಎಳೆದಿಟ್ಕೊಳ್ಳೋದ್ರಿಂದ ನೀರು ಬೇರಿಗೆ ಹೋಗುತ್ತೆ, ಆಮೇಲೆ ಅದಕ್ಕೆ ಸಿಗಬೇಕಾದ ಇದಕ್ಕೆ ಸಿಗುತ್ತೆ. ಬಟ್ ಇದರಲ್ಲಿ ಒಂದು ಡಿಸ್ಅಡ್ವಾಂಟೇಜು ಇದೆ, ಕೆಲವೊಂದು ಸಲ ಪೋಷಕಾಂಶಗಳು ಅವು ಸಹ ಇದು ಆಗುತ್ತೆ, ಅವುಗಳು ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ಗೆ ಬೈಂಡ್ ಆಗುತ್ತೆ, ಅವು ಅಂಟ್ಕೊಳ್ಳುತ್ತೆ. ಬಟ್ ಎಲ್ಲೋ ಒಂದು ಕಡೆ ನೀರು ಮತ್ತು ತೇವಾಂಶ ಜಾಸ್ತಿ ಆದಾಗ ಇವು ಮತ್ತೆ ಆಚೆ ಬರುತ್ತೆ. ಈ ಒಂದು ಏನು ಬೈಂಡಿಂಗ್ ಅಂತಾರಲ್ಲ, ಇದು ಪರ್ಮನೆಂಟ್ ಬೈಂಡಿಂಗ್ ಅಲ್ಲ. ನಥಿಂಗ್ ಬೈಂಡ್ಸ್ ಟು ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಪರ್ಮನೆಂಟ್ಲಿ. ಅವು ಹೋಗ್ತಾ ಇರುತ್ತೆ, ಬರ್ತಾ ಇರುತ್ತೆ. ಸೊ ನೀವು ಬೇರಲ್ಲಿ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಹಾಕಿ ಒಳ್ಳೆಯದು, ಬಟ್ ಒಳ್ಳೆ ಕ್ವಾಲಿಟಿದು ಹಾಕಿ. ಅದು ಬೇರೆ ಹತ್ರ ಇದ್ದರೆ ಒಳ್ಳೆಯದು. ಬೇರೆ ಹತ್ರ ಅದನ್ನು ಡ್ರಿಪ್ಪಲ್ಲೂ ಹಾಕ್ತಾರೆ, ಈಗ ಪೌಡ್ರಲ್ಲೂ ಹಾಕ್ತಾರೆ. ಸೊ ಸರ್, ಒನ್ ಈಸ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರರ್. ಹೂ ಈಸ್ ದಿ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರರ್? ಯು ಹ್ಯಾವ್ ಟು ಸಿ, and you have to see what experience they have. If people are usually in the mining business, they produce good humic acid. First criteria, because they know where to take lignite out, where to take peat out, where to do this, and they have the necessary equipment. So go with the thing. Understand, are they in the petroleum business? Even petroleum company like Armaco. Huh? Most of them are. Sir, it's a very pertinent question, but actually none other answer illa. Only thing I can go is Israel produces best humic acid. Israel. Lebanon produces good. Morocco also produces this one. If you know if they are sourced somewhere from this, you can be of assured guarantees. China, I don't know. In fact, they say even Russia has good humic acids, but I don't know whether they're doing it one. Yes. Okay. ಇದೆ ಸರ್ ರೂಟ್ ಜಾಸ್ತಿ ಆಗುತ್ತಂತ ಹಾಂ ಓಕೆ ಸರ್ ಒಂದು ನೋಡಿ 1 out of 3 ಅಂತ ಹಾಕಿದ್ದೀನಿ. ನೀವೊಂದು 2 ಸ್ಲೈಡ್ ವೇಟ್ ಮಾಡಿ, ಈ 3 ಸ್ಲೈಡ್ ಹೇಳಿದ್ಮೇಲೆ ನಾನು ಮತ್ತೆ ರಿವಿಸಿಟ್ ಮಾಡ್ತೀನಿ. ಇದು ಒಂದು ಮೆಕ್ಯಾನಿಸಮ್. ಒಂದೇನಂತಂದರೆ ಈ ಉಪ್ಪು ಅಂಶನೆಲ್ಲ ಹಿಡಿದಿಟ್ಕೊಳ್ಳುತ್ತೆ, ಅದು ಒಂದಾಯಿತು. ನೆಕ್ಸ್ಟ್ ಹೇಳ್ತೀನಿ. ಅದಾದಮೇಲೆ ವಿ ವಿಲ್ ಸಮರೈಸ್ ದ ಹೋಲ್ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಬಿಕಾಸ್ ಇಟ್ ಇಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ಯು ಗೈಸ್ ಮೋರ್ ದನ್ ಅದರ್ ಥಿಂಗ್ಸ್. ಎಸ್ ಎಸ್ ಹಾಂ ಓಕೆ ಹಾಂ ಓಕೆ
ನನಗೇನು ಮೈಕ್ ಬೇಕು?
ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಹಾಕಿದ್ರೆ ಡ್ರೂಪ್ ಆಗುತ್ತಾ ಸ್ಪ್ರೇ ಮಾಡಿದಾಗ
ಎಲೆ?
ಸರ್ ನಮ್ಮ ರಿಸರ್ಚ್ ಪ್ರಕಾರ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಅಂದರೆ ಅದರಲ್ಲಿ 3 ಫ್ರಾಕ್ಷನ್ ಇರುತ್ತೆ ಸರ್. ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಅಂತ ಇರುತ್ತೆ, ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಇರುತ್ತೆ, ಹ್ಯೂಮೇಟ್ಸ್ ಅಂತ ಇರುತ್ತೆ. ನೀವು ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇರೋ ಫ್ರಾಕ್ಷನ್ ಸ್ಪ್ರೇ ಮಾಡಿದ್ರೆ ಅದು ಒಳ್ಳೇದಲ್ಲ ಎಲೆಗೆ. ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇರೋ ಫ್ರಾಕ್ಷನ್ ಸ್ಪ್ರೇ ಮಾಡಬೇಕು ನೀವು ಫೋಲಿಯರ್ಗೆ, ಎಲೆಗೆ ಹಾಕಬೇಕು ಅಂದರೆ. ಸೊ ಅದೇನಾಗುತ್ತೆ ಅಂದರೆ ಅದು ಸೆಲ್ ಒಳಗಡೆ ಹೋಗಿ ರಿಯಾಕ್ಟ್ ಆಗಿ ಅದು ಏನದು ಹೊಸ ಹೊಸ ಪಾತ್ವೆ ಕ್ರಿಯೇಟ್ ಮಾಡಿ ಈಗ ಫ್ರೂಟಿಗೆ ಬೇಕಾದ ಕಲರ್, ಬೇಕಾದ ಪಿಗ್ಮೆಂಟ್ಸ್ ಕೊಡುತ್ತೆ, ಫ್ಲೇವರ್ಸ್ ಕೊಡುತ್ತೆ, ಅವೆಲ್ಲ ಮಾಡಿಸುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಏನು ಗೊತ್ತಾ ಸರ್, ವಿ ಹ್ಯಾವ್ ಟು ಸಿ ವಾಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ದೇ ಆರ್ ಸ್ಪ್ರೇಯಿಂಗ್.
ಈಗ
ಆನ್ಸರ್ ದಟ್
ನೀವು ಹೇಳಿದ್ರಿ ಕೆಲವು ದೇಶದ್ದು ಒರಿಜಿನಲ್ ಚೆನ್ನಾಗಿರುತ್ತೆ ಅಂತ. ಇಲ್ಲೀಗ ನಮಗೆ ಮಾರ್ಕೆಟಲ್ಲಿ ಸಿಗ್ತಾ ಇರೋದು ಚೆನ್ನಾಗಿದೆಯೋ ಇಲ್ಲೋ ಅಂತ ಹೇಗೆ ಪತ್ತೆ ಹಚ್ಚೋದು ನಾವು?
ಲ್ಯಾಬ್ ರಿಪೋರ್ಟ್ ಕೇಳಬೇಕು ಸರ್ ನೀವು. ಇಲ್ಲ, ಯು ಸಿ ವಾಟ್ ಐ ಕ್ಯಾನ್ ಸೇ ಇಸ್, ಇಫ್ ಯು ಆರ್ ಗರ್ಕಿನ್ ನೀವೆಲ್ಲ ಯು ಶುಡ್ ಹ್ಯಾವ್ ಫ್ರಮ್ ಒನ್ ಸಿಂಗಲ್ ಸೋರ್ಸ್. Maybe, because you are a big buyer, sir. Talk to Lebanese companies. I will help you with we can talk to those guys. Nan favor madanga, but I'm not consulting any of those companies. But I can tell you which countries they make it. See, one of my previous companies, I don't want to give the name. Only thing is, they were sourcing from US, they were sourcing from Lebanon, they were sourcing from this one, and that had very positive effect in this thing. I have seen on the row full humic acid ringittu. Without ಇದು ಹೆಂಗಿತ್ತು ಸೀಡ್ಲಿಂಗ್ಸ್, ಹೌ ದೇ ವರ್ ಗ್ರೋಯಿಂಗ್, ಹೌ ದೇ ವರ್ ಶೈನಿ, ಎವ್ರಿಥಿಂಗ್. ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಎಫೆಕ್ಟ್ ವಾಸ್ ದೇರ್. ಸೊ ವಿ ಹ್ಯಾವ್ ಟು ಹೆಲ್ಪ್ ಸರ್. ಮಾಡಬೇಕು ಸರ್, ನಮಗೂ ಗೊತ್ತಿಲ್ಲ ಆನ್ಸರು ಹೆಂಗೆ ಮಾರ್ಕೆಟಲ್ಲಿ ಏನಿದೆ, ಏನೇನು ಬಂದಿದೆ ಅಂತ.
ಈಗ ಇನ್ನೊಂದಿದು ಗ್ರ್ಯಾನ್ಯೂಲ್ಸ್ ತಗೋಬೋದು ಅಥವಾ ಲಿಕ್ವಿಡ್ ತಗೋಬೋದು. ಅದರಲ್ಲಿ ಇಂಗ್ರೀಡಿಯೆಂಟ್ಸು ಯಾವ್ಯಾವ್ದು ಇರುತ್ತೆ, ಎಷ್ಟು % ಇದ್ರೆ ಒಳ್ಳೇದು ಅನ್ನೋದು ಏನಾದರೂ?
ಓಕೆ, ಅದೇ ಸರ್, ನೀವು ಲೀಫ್ಗೆ ಆಗೋದಾದರೆ ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇರಬೇಕು. ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಅಂತ ಇರುತ್ತೆ, ಅದ್ರ ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಕಂಟೆಂಟ್ ಜಾಸ್ತಿ ಇದ್ದಷ್ಟು 50-60% ಇದ್ದರೆ ಇಟ್ ಈಸ್ ಗುಡ್ ಟು ಸ್ಪ್ರೇ. ಅದಕ್ಕೆ ಫಲ್ವಿಕ್ ಆಸಿಡ್, ಈ ಪ್ಯೂರ್ ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಬರುತ್ತೆ. ಫಲ್ವಿಕ್ ಆಸಿಡ್ ಇಸ್ ವೆರಿ ವೆರಿ ಪೊಟೆಂಟ್. ಈವನ್ ಅನಿಮಲ್ಸ್ ಅಲ್ಲಿ ಪೌಲ್ಟ್ರಿ ಕೊಡ್ತಾರೆ, ಕ್ಯಾಟಲಿಗೆ ಕೊಡ್ತಾರೆ ಫಲ್ವಿಕ್ ಆಸಿಡ್. ಇಟ್ ಇಂಪ್ರೂವ್ಸ್ ಮೆಟಬಾಲಿಸಮ್, ಇಟ್ ಇಂಪ್ರೂವ್ಸ್ ಡೈಜೆಷನ್, ಬಿಕಾಸ್ ಆಕ್ಸಿಜನ್ ಇರೋದ್ರಿಂದ ಒಳಗಡೆ ಆಕ್ಸಿಜನೇಟೆಡ್ ಮಾಲಿಕ್ಯೂಲ್, ವೆರಿ ವೆರಿ ರಿಯಾಕ್ಟಿವ್ ಪ್ಲಾಂಟ್ ಮೇಲೆ. ಸೊ ನಿಮಗೆ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಬರೀ ಕಾರ್ಬನ್ ಹೈಡ್ರೋಜನ್ ಇರೋದು ಸ್ಪ್ರೇ ಮಾಡಿದ್ರೆ ಎಲೆ ಮೇಲೆ ಗೊಬ್ಬರ ಹಾಕ್ದಂಗೆ ಆಯ್ತು ನಿಮಗೆ.
ಅಲ್ಲ, ಗೊಬ್ಬರ ಹಾಕೋದು ಒಳ್ಳೇದೇ ಅಲ್ವಾ?
ಕೊಟ್ಟಿಗೆ ಗೊಬ್ಬರ ಎಲೆ ಮೇಲೆ ಹಾಕಿದ್ರೆ ಏನಾಗುತ್ತೆ?
ಅದು ಅನುಕೂಲ.
ಆ ಥರ ಅದೇ ಇದು. ಯಾಕಂದ್ರೆ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಅಲ್ಲಿ ಕಾರ್ಬನ್ ಹೈಡ್ರೋಜನ್ನೇ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಬೇರೆ ಕಮ್ಮಿ ಇರುತ್ತೆ. ಬಟ್ ನೀವು ಫಲ್ವಿಕ್ ಆಸಿಡ್ ತರ ಹೋದಾಗ ಏನಾಗುತ್ತೆ, ನಿಮಗೆ ಆಕ್ಸಿಜನೇಟೆಡ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಜಾಸ್ತಿ ಬರುತ್ತೆ.
ಈಗ ನಮ್ಮ ಗರ್ಕಿನ್ಗೆ ಗ್ರಾನ್ಯೂಲ್ಸ್ ಉಪಯೋಗಿಸೋದು ಒಳ್ಳೇದು, ಲಿಕ್ವಿಡ್
ಅಷ್ಟೇ? ದಟ್ ಈಸ್ ದ ಆನ್ಸರ್.
ಸಾಯಿಲ್
ನೀವು ಈಗೇನಾಗುತ್ತೆ, ಫಸ್ಟಲ್ಲಿ ನೀವು ಡ್ರಿಪ್ಪಲ್ಲಿ ಕೊಡ್ತೀರಾ, ಅವಾಗ ನೀಟಾಗಿ ಟ್ರಿಕ್ಕಲ್ ಆಗುತ್ತೆ, ಹೋಗ್ತಾ ಹೋಗುತ್ತೆ. ಒಂದು 2 months ಆದಮೇಲೆ ಬೇರ್ ತುಂಬ ಒಳಗಡೆ ಹೋಗಿರುತ್ತೆ. ನೀವು ಅವಾಗ ಡ್ರಿಪ್ಪಲ್ಲಿ ಕೊಟ್ಟರೆ ಪ್ರಯೋಜನ ಆಗಲ್ಲ. ಅಂದರೆ ಅವಾಗ ಏನಾಗತ್ತಂದರೆ ಈ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಆಗೋದೆಲ್ಲ ರೂಟ್ ಟಿಪ್ಪಲ್ಲಿ ಆಗುತ್ತೆ ಸರ್, ರೂಟ್ ಕೆಳಗಡೆ ಇರ್ತವೆ. ಯಾವಾಗಲೂ ನೋಡಿ ಬೇರಲ್ಲಿ ಹೆಂಗಿರುತ್ತಂದರೆ ಬೇರಿಗೆ ಬೆಳೀತಾ ಹೋಗುತ್ತಲ್ಲ, ಎಂಡಲ್ಲಿ ರೂಟ್ ಹೇರ್ಸ್ ಬರುತ್ತೆ ಫೈನ್, ದೇ ಆರ್ ದ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಪಾಯಿಂಟ್ಸ್. ನೀವು ಅದು ಗ್ರೋ ಆದಮೇಲೆ ಏನಾಗ್ಬಿಡುತ್ತೆ, ಡೀಪ್ ಹೋಗ್ಬಿಡುತ್ತೆ ನಿಮಗೆ.
ಡೀಪ್ಗಿಂತ ಸ್ಪ್ರೆಡ್ ಆಗುತ್ತೆ ಅಂತ ನಮ್ಮ
ದಟ್ಸ್ ಫೈನ್. ಹಂಗಾದ್ರೆ ಸರಿ ಸರ್. ನಾನೊಂದು ರೆಕಮೆಂಡೇಶನ್ ಇದೆ ಲಾಸ್ಟಲ್ಲಿ, ಒಂದು ಸ್ಲೈಡ್ ಇದೆ ಮಧ್ಯಾಹ್ನ ನಿಮಗೆ ತೋರಿಸ್ತೀನಿ. ಏನು ನಮ್ಮ ಪ್ರಕಾರ ಏನು ಮಾಡಬೇಕು ನೀವು. ಕೆಲವು ಹಾರ್ಮ್ಲೆಸ್, ಅದರಲ್ಲಿ ಐ ಆಮ್ ಆಲ್ರೆಡಿ ಜಂಪಿಂಗ್ ದ ಗನ್. ಇಲ್ಲಿ ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಹಾಕಿ ಸರ್. ಸಾಯಿಲ್ಗೆ ಎಷ್ಟು ಇನ್ನೂ ಬೇಕಾದ್ರೆ ಹಿಂಗೆ ಡ್ರಿಲ್ ಮಾಡಿ ಹಾಕ್ತಾರೆ ನೋಡಿ, ಫರ್ಟಿಲೈಸರ್ ಥರ ಡಿಬ್ಲಿಂಗ್ ಮಾಡಿ ಅದರಲ್ಲಿ ಹಾಕಿ, ಆ ಕಡೆ ಒಂದು ಈ ಕಡೆ ಒಂದು ಹಾಕಿ ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಹಾಕಿ.
ಅಲ್ಲ, ಅದು ಈಗ ನಾವು ಮಾಡ್ತಾ ಇರೋದು ನಮ್ಮ ಫ್ಯಾಕ್ಟ್ರಿದು ಹೇಳ್ತೀನಿ. ಬೇಸಲ್ಲಾಗಿ DAP, ಪೊಟಾಶು ಈ ಥರ ಕೊಡ್ತೀವಲ್ಲ, ಅದಕ್ಕೇನೆ
ಆರಾಮ್ಸೆ
ಗ್ರಾನ್ಯುಯಲ್ ಮಿಕ್ಸ್ ಮಾಡಿ ಕೊಡ್ತಾ ಇದ್ದೀವಿ.
ಆರಾಮ್ಸೆ ಕೊಡಿ. ನೆವರ್ ಅದು ಏನಾದರೂ ಅವೆಲ್ಲ ಅಲ್ಲೇ ಇರುತ್ತೆ, ಪೂಲಲ್ಲೇ ಇರುತ್ತೆ. ಇನ್ನೊಂದು ಏನು ಗೊತ್ತಾ, ಸಿ ಪೀಪಲ್ ಆರ್ ಟಾಕಿಂಗ್ ಅಬೌಟ್ ಸ್ಲೋ ರಿಲೀಸ್ ಫರ್ಟಿಲೈಸರ್ಸ್. ಐ ಡೋಂಟ್ ನೋ ಹೌ ಮಚ್ ವಿ ಹ್ಯಾವ್ ಟಾಕ್. ನಮ್ಮಲ್ಲಿರೋದೆಲ್ಲ ರ್ಯಾಪಿಡ್ ರಿಲೀಸ್ ಫರ್ಟಿಲೈಸರ್ಸ್, ವಿಚ್ ಇಸ್ ನಾಟ್ ಗುಡ್. We should also have slow release fertilizer. By using humic acid, you have made a slow release fertilizer. Indirectly, you have made a slow release fertilizer. It will ನಮಗೆ ಅಷ್ಟೇ ಬೇಕಾಗಿದೆ ಸರ್. ಪ್ಲಾಂಟ್ಗೆ ಒಂದೇ ಸಲ ಸರ್ ಒಂದು ಏನಂದರೆ, ನಿಮಗೆ ವಾರ ನಾವು ಪಾರ್ಟಿ ಮಾಡಿದ್ರೆ ಚೆನ್ನಾಗಿರುತ್ತೆ. 6 ತಿಂಗಳಿಗೆ ಒಂದೇ ಸಲ ಪಾರ್ಟಿ ಅಂದರೆ ಅದು ಚೆನ್ನಾಗಿರಲ್ಲ.
ಈ ಗರ್ಕಿನ್ ಗಿಡಕ್ಕೆ ದಿವಸ ಪಾರ್ಟಿ ಮಾಡಬೇಕು. ಅಲ್ವಾ ಸರ್? ಇಲ್ಲೇ ಇದಾರಲ್ಲ ನಮ್ಮ ಡಾಕ್ಟ್ರು.
ಸೊ ದಿಸ್ ಇಸ್ ದಿ ಐರನಿ, ಬಟ್ ಗುಡ್, ಯುವರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಇಸ್ ಗುಡ್. ಐ ಲೈಕ್ ದಿ ಇಂಟ್ರಾಕ್ಷನ್. ಯಾಕಂದರೆ ವಿ ಶುಡ್ ಬಿ ಓಪನ್, ವಿ ಶುಡ್ ನಾಟ್ ಜಸ್ಟ್ ಸೇ ಚೆನ್ನಾಗಿದೆ ಅಂತಲ್ಲ. ಆ್ಯಂಡ್ ಐ ಆಮ್ ನಾಟ್ ಸೇಯಿಂಗ್ ದಟ್ 100% ಸೊಲ್ಯೂಷನ್ ಅಲ್ಲ. ದೇರ್ ಆರ್ ಡ್ರಾಬ್ಯಾಕ್ಸ್ ಆಲ್ಸೋ ವಿತ್ ದ್ಯಾಟ್ ಒನ್. ಓಕೆ, ಹಾಂ, ಓಕೆ ಸರ್. ಈಗ ಕಂಪ್ನಿ ತೊಗೊಂಡಿರ್ತಾರೆ, ಅವರು ಇನ್ನೊಬ್ರ ಹತ್ರ ಡಿಸ್ಟ್ರಿಬ್ಯೂಷನ್ ತೊಗೊಂಡಿರ್ತಾರೆ, ಇಂಪೋರ್ಟ್ ಮಾಡಿರ್ತಾರೆ. ಪೇರೆಂಟ್ ಸಪ್ಲೈಯರು ಬೇರೆ ಇರ್ತಾನೆ, ಪ್ರಿನ್ಸಿಪಲ್ ಸಪ್ಲೈಯರ್ ಬೇರೆ ಇರ್ತಾನೆ. ಕೇಳಿ ಸರ್, ಇದು ಎಲ್ಲಿಂದ ಬಂದಿರೋದಪ್ಪ ಏನಂತ ಸ್ವಲ್ಪ ನೋಡ್ಕೊಳ್ಳಿ. ಏನು ಮಿಕ್ಸ್ ಮಾಡಿದಾರೋ ಏನೋ ನನಗಂತೂ ಗೊತ್ತಿಲ್ಲ ಇದರಲ್ಲಿ. ಸೊ ಮಾಡಿಸ್ಕೋಬೇಕು.
100 numbers of companies, sir.
Sir, but I am saying is go with the reputed multinational companies like BASF ಹೋಗಿ. I mean, I should not recommend it. It's not, but any good company, ಅವ್ರ ಹತ್ರ ಹೋಗಿ you can also check with them, no problem. And say ಅವರು ರಿಪೋರ್ಟ್ ಕೊಡ್ತಾರೆ, ಒಂದು SDS ರಿಪೋರ್ಟ್ ಕೊಡ್ತಾರೆ, ಮೆಟೀರಿಯಲ್ ಸೇಫ್ಟಿ ಡೇಟಾ ಕೊಡ್ತಾರೆ, ಇದು ಇದೆ ಏನಿದೆ ಅಂತ ಕೊಡ್ತಾರೆ. Then you can be assured of this. Why you take a lot of ಸರ್ ಅದರಲ್ಲೇ ಇಂಪ್ಯೂರಿಟೀಸ್ ಕೊಡ್ತಾರೆ, ಸಾಲ್ಯುಬಿಲಿಟಿ ಕೊಡ್ತಾರೆ, ಯಾವ pH ಅಲ್ಲಿ ಡಿಸಾಲ್ವ್ ಆಗುತ್ತೆ ಅಂತ. ಇನ್ನೊಂದು, ಈಗ ಅವ್ರು ಹೇಳಿದ್ರಲ್ಲ, ನಾವು ಗ್ರಾನ್ಯುಲ್ ಕೊಡ್ತೀವಿ, ಇದ್ರಲ್ಲಿ ಕೊಡ್ತೀವಿ, ಅದು ಕೊಡ್ತೀವಿ. ಫಸ್ಟ್ ಎಷ್ಟು ರೇಂಜ್ pH ಅಲ್ಲಿ ಸಾಲ್ಯುಬಲ್ ಆಗುತ್ತೆ ಅದನ್ನು ನೋಡಿ ಸರ್. ಇಫ್ ದೇರ್ ಈಸ್ ಸಮ್ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ವಿಚ್ ಡಿಸಾಲ್ವ್ಸ್ ಓನ್ಲಿ ಬಿಟ್ವೀನ್ 5.5 and 7, ಪ್ಲೀಸ್ ತಗೊಳಕ್ಕೆ ಹೋಗ್ಬೇಡಿ. ಏನೇನಾಗುತ್ತೆ ಸಾಯಿಲಲ್ಲಿ ಅಂತ ನಿಮಗೂ ಗೊತ್ತಿಲ್ಲ, ನಮಗೂ ಗೊತ್ತಿಲ್ಲ. ಇದು ಇಟ್ಸ್ ಲೈಕ್ ಎ ವಾರ್ ಝೋನ್ ಅದು ಕೆಳಗಡೆ. ಸೊ ಯು ಶುಡ್ ಟೇಕ್ ಒನ್ ವಿಚ್ ಇಸ್ ಹೈ ರೇಂಜ್ pH ಅಲ್ಲಿ ಡಿಸಾಲ್ವ್ ಆಗಬೇಕು. ಫಸ್ಟ್ ವಾಟರ್ ಸಾಲ್ಯುಬಿಲಿಟಿ ಇದಿರಬೇಕು, ಆಮೇಲೆ ಟಾಕ್ಸಿನ್ಸ್ ಕಮ್ಮಿ ಇರಬೇಕು, ಆ್ಯಂಡ್ ಇಟ್ ಈಸ್ ಮೈಂಡ್ ಫ್ರಮ್ ದ ನೋನ್ ಸೋರ್ಸು ಅದರಲ್ಲಿ. ಸೊ ಇದೆ ಸರ್ ಕ್ರೈಟೀರಿಯಾ. ನನಗೆ ತಲೆ ಅಷ್ಟು ಓಡ್ತಾ ಇಲ್ಲ, ಬಟ್ ಐ ವಿಲ್ ಗಿವ್ ಯು ಸಮ್ ರೆಫರೆನ್ಸಸ್ ಟು ಯುವರ್ ಸೊಸೈಟಿ. I have publications where you can use this as a criteria to select the good humic acids. Sir, adu inna first du aitu sir, first mode of action. Let's go to second mode of action. Enaitida? Next slide. Okay. ನೋಡಿ, ಇದು ಸ್ವಲ್ಪ ಕಾಂಪ್ಲಿಕೇಶನ್ ಇದೆ, ಬಟ್ ಯು ಹ್ಯಾವ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಿಸ್ ಒನ್. ಬಟ್ ಐ ಟ್ರೈ ಟು ಸಿಂಪ್ಲಿಫೈ ಆಗಿ ಮಾಡಿ ಹೇಳಕ್ಕೆ ಮಾಡ್ತೀನಿ ಸರ್. ಈ ಪ್ಲಾಂಟ್ ಸೆಲ್ ಇರುತ್ತಲ್ಲ, ಸೆಲ್ಲಲ್ಲಿ ಪ್ರೋಟಾನ್ ಪಂಪ್ ಅಂತ ಇರುತ್ತೆ. ಸೊ ಪ್ರೋಟಾನ್ ಪಂಪ್ ಅಂದರೆ ದೀಸ್ ಆರ್ ಪ್ರೋಟೀನ್ಸ್. ಒಂದು ಪಂಪ್ ಇರುತ್ತೆ. ಈಗ ನೀರು ಈ ಕಡೆಯಿಂದ ಆ ಕಡೆ ಬಂದು ಮಾಡ್ತೀವಲ್ಲ. ಸೊ ಯಾವಾಗಲೂ ನೀವು ಒಂದು ಸೆಲ್ ತಗೊಂಡ್ರೆ ನೋಡಿ, ಒಂದು ಸೆಲ್ಲಲ್ಲಿ ಲೀಫ್ ಸೆಲ್ ಆಗ್ಬೋದು, ರೂಟ್ ಸೆಲ್ ಆಗ್ಬೋದು, ಎಲ್ಲೇ ಆಗ್ಬೋದು, ಈ ಪ್ರೋಟಾನ್ಸ್ ಅಂತ ಇರ್ತವೆ. ದೀಸ್ ಆರ್ ನಾಟ್ ಕಾಲ್ಡ್ H ಪ್ರೋಟಾನ್. ಇವು ಸೆಲ್ ಒಳಗಡೆ ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಯಾವಾಗಲೂ ಹೊರಗಡೆ ನೀವು ಹಾಕ್ತಿರಲ್ಲ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸು, ಪೊಟ್ಯಾಷಿಯಮ್, ಮೆಗ್ನೀಷಿಯಮ್, ಕ್ಯಾಲ್ಶಿಯಮ್, ಬೋರಾನ್, ಎಲ್ಲ ಅಯಾನ್ಸ್ ಆಗಿ ಹೊರಗಡೆ ಕಾಯ್ಕೊಂಡು ಕೂತಿರ್ತಾರೆ. ಹೀಗೆ They want to go inside, but they cannot go inside. Why? Because there is a pump. Pump current illa. It is not active. Now when you add humic acids, they have reactive idu. ಏನು ಮಾಡುತ್ತೆ, ಒಂದು enzyme activate ಮಾಡುತ್ತೆ. There is an enzyme called ATPase. When that enzyme is activated, pump start ಆಗುತ್ತೆ. And humic acids, fulvic acids, they are very good in activating these proton pumps. So proton pump ಆದಾಗ ಏನಾಗುತ್ತೆ, ಇಲ್ಲಿ ನೋಡಿ ಸರ್, ಇದೆಲ್ಲ HH ಎಲ್ಲ ಇದೆಯಲ್ಲ, ಇದೆಲ್ಲ ಆಚೆ ಹೋಗುತ್ತೆ. And then all the carrier materials like calcium, magnesium, ನೀವೇನು ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಹಾಕ್ತಿರಲ್ಲ, ಅದೆಲ್ಲ ಒಳಗಡೆ ಬರುತ್ತೆ. ಎಸ್. ಸೊ ಅದು ಪ್ರೋಟೀನ್ ಗ್ರೇಡಿಂಗ್ ಕ್ರಿಯೇಟ್ ಆಗಬೇಕು. ಅಂದರೆ ಒಂದು ಈ ಥರ ಸ್ಲೋಪ್ ಕ್ರಿಯೇಟ್ ಆಗಬೇಕು. ಮಾಲಿಕ್ಯುಲರ್ ಸ್ಲೋಪ್ ಅಂತೀವಿ. ಆಲ್ ದ ಪ್ರೋಟಾನ್ಸ್ ಗೋ ಔಟ್, ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಕಮ್ ಇನ್ಸೈಡ್. ಇದು ಬೇಸಿಕ್ ಮೆಕ್ಯಾನಿಸಮ್ ಇನ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್. ನೀವು ಎಲ್ಲಾದರೂ ತಗೊಳ್ಳಿ, ನೀವೇನೋ ಫರ್ಟಿಲೈಸರ್ ಹಾಕಿ ಇದು ಗ್ರೇಡಿಯಂಟ್ ಕ್ರಿಯೇಟ್ ಆಗದೆ ಇದ್ದರೆ ನೀವು ಹಾಕಿರೋ ಫರ್ಟಿಲೈಸರ್ ಎಲ್ಲ ಆಚೆನೇ ಇರುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಏನಂದರೆ ಪ್ಲಾಂಟ್ ಶುಡ್ ಆಲ್ಸೋ ಬಿ ಹೆಲ್ದಿ, ರೂಟ್ ಶುಡ್ ಬಿ ಹೆಲ್ದಿ. ಅವಕ್ಕೆ ಎನರ್ಜಿ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾ ಇರಬೇಕು, ಫೋಟೋಸಿಂಥಸಿಸ್ ಆಗ್ತಾ ಇರಬೇಕು. 5 days, ಒಂದು 2 weeks ಮೋಡ ಮುಚ್ಕೊಂಡಿದೆ, ನೀವು ಹೆವಿ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಹಾಕ್ತೀರಾ, ಇಟ್ ಈಸ್ ಎ ವೇಸ್ಟ್. ಇಟ್ ಈಸ್ ಎ ವೇಸ್ಟ್. ಇಟ್ ಈಸ್ ಲೈಯಿಂಗ್ ದೇರ್. ಇಟ್ ಈಸ್ ನಾಟ್ ಬೀಂಗ್ ಟೇಕನ್ ಅಪ್. ಅಯ್ಯೋ ಏನು ಫರ್ಟಿಲೈಸರ್ ಹಾಕೋರು ಆಗಲ್ಲ ಅಂತ ನೀವು ಇಲ್ಕ. ಸೊ ಬಟ್ ಯು ನೀಡ್ ಟು ಹ್ಯಾವ್ ದಟ್. ಎಲ್ಲ ರೂಟ್ ಸೆಲ್ಸಲ್ಲಿ ಪ್ರೋಟಾನ್ ಪಂಪ್ ಶುಡ್ ಬಿ ಆಕ್ಟಿವ್. Humic acids can do that. They synthesize this one and all the nutrients come. ನೋಡಿ ಫೋಲಿಯರಲ್ಲಿ ಏನು ಮಾಡುತ್ತೆ, ಅದೊಂದು ಮಾಡುತ್ತೆ, ಅದು ಮಾಡಿಬಿಟ್ಟು ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಆರ್ ಅಬ್ಸಾರ್ಬ್ಡ್. ಅದ್ರ ಜೊತೆಗೆ ಒಂದು ಕ್ಯಾಲ್ಸಿಯಂ ಚಾನೆಲ್ಸ್ ಅಂತ ಕ್ರಿಯೇಟ್ ಮಾಡ್ತೇವೆ. ಅಂದರೆ ಕ್ಯಾಲ್ಸಿಯಂ ಇಸ್ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಇನ್ ದ ಪ್ಲಾಂಟ್. ಇಫ್ ಯು ಸೇ ಆಲ್ ದ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್, ಕ್ಯಾಲ್ಸಿಯಂ ಇಸ್ ಸೋ ಇಂಪಾರ್ಟೆಂಟ್. ಕ್ಯಾಲ್ಸಿಯಂ ಬಿದ್ದರೇನೆ ಫೋಟೋಸಿಂಥಸಿಸ್ಗೆ ಓಪನ್ ಆಗುತ್ತೆ, ಪಾತ್ವೆ ಆಗುತ್ತೆ, ಸ್ಟೊಮ್ಯಾಟೊ ಓಪನ್ ಆಗುತ್ತೆ, ಕಾರ್ಬನ್ ಇದು ಮಾಡೋದು. ಕ್ಯಾಲ್ಸಿಯಂ ಇಲ್ಲದೆ ಇದ್ದರೆ ಭಾರಿ ಕಷ್ಟ ಆಗುತ್ತೆ. ಕ್ಯಾಲ್ಸಿಯಂ ಚಾನೆಲ್ಸ್ ಓಪನ್ ಮಾಡೋದು ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ಸ್. ದೇ ಆಲ್ಸೋ ಓಪನ್ ದ ಕ್ಯಾಲ್ಸಿಯಂ ಚಾನಲ್ಸ್ ಇಲ್ಲಿ. ಅದರಿಂದ ನೋಡಿ ನಿಮಗೆ ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸು, ನಿಮ್ಮ ಫ್ರೂಟ್ ಕಲರ್ ಬರಬೇಕು, ಫ್ಲೇವರ್ ಬರಬೇಕು ಅಂದರೆ ಕ್ಯಾಲ್ಸಿಯಂ ಮೆಟಬಾಲಿಸಮ್ ಶುಡ್ ಬಿ ವೆರಿ ವೆರಿ ಗುಡ್. ಇಫ್ ಯು ಡೋಂಟ್ ಹ್ಯಾವ್ ಕ್ಯಾಲ್ಸಿಯಂ, ಆಲ್ ದ ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸ್, ಪ್ರೈಮರಿ ಮೆಟಬಲೈಟ್ಸ್, ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸ್ ಎರಡಿದೆ. ಪ್ರೈಮರಿ ಮೆಟಬಲೈಟ್ಸ್ ಎನಿ ಎಕ್ಸಾಂಪಲ್ ಏನು ಹೇಳಿ? ನಾವೇನು ನೋಡಿದ್ವಿ? ಪ್ರೋಟೀನ್ಸ್, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್, ಫ್ಯಾಟ್ಸ್, ದೇ ಆರ್ ಆಲ್ ಪ್ರೈಮರಿ ಮೆಟಬಲೈಟ್ಸ್. ಇವು ಎಸೆನ್ಷಿಯಲ್. ಈಗ ನೀವು ತೊಗರಿ ತಗೊಳ್ಳಿ, ಜೋಳ ತಗೊಳ್ಳಿ, ನೀವು ಗರ್ಕಿನ್ಸಲ್ಲಿ ಕನ್ವರ್ಟ್ ಆಗಿರುತ್ತೆ ಅದೆಲ್ಲ ಫ್ರೂಟ್ಸ್ಗಾಗಿ. ಸೊ ದೇರ್ ಆರ್ ಪ್ರೈಮರಿ ಮೆಟಬಲೈಟ್ಸ್, ಸ್ಟೇಪಲ್ ಫ್ರೂಟ್ಸ್. ಈ ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸ್ ಅಂತಂದರೆ ಕಮ್ಮ ಸ್ಮೆಲ್ ಬರುತ್ತೆ, ತಿಂದರೆ ಒಂಥರ ಬೇರೆ ಟೇಸ್ಟ್ ಇದೆ, ಸ್ವಲ್ಪ ಹುಳಿ ಹುಳಿ ಅರೋಮಾ, ಆಲ್ ಸೆಕೆಂಡ್ರಿ. ಆಮೇಲೆ ಇದು ಕಹಿ ಇರುತ್ತೆ, ಅವೆಲ್ಲ ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸ್. But they add value to either to your food flavor, color, defending the plant from insects. ಅದಕ್ಕೆ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತವೆ ಅವು. ಆ ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸ್ ಪಾತ್ವೆ ಆಗಬೇಕು ಅಂದರೆ ಕ್ಯಾಲ್ಸಿಯಂ ಬೇಕೇ ಬೇಕು ನಿಮಗೆ. ಸೊ ಅಂದರೆ ನಿಮಗೆ ಎಸ್ಪೆಷಲಿ ಫ್ರೂಟ್ ಗ್ರೋವರ್ಸ್ಗೆ ಕ್ಯಾಲ್ಸಿಯಂ ಇಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ದಟ್ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಡಸ್. ನೋಡಿ ಈ ಲೀವ್ಸಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಇದು ಪಂಪ್ ಓಪನ್ ಮಾಡಿಬಿಟ್ಟು ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಅಬ್ಸರ್ವ್ ಮಾಡುತ್ತೆ, ನಂಬರ್ 1. ನಂಬರ್ 2, ದೇ ವಿಲ್ ಆಕ್ಟಿವೇಟ್ ದ ಕ್ಯಾಲ್ಸಿಯಂ ಚಾನಲ್. All these beautiful aroma secondary metabolites are triggered. Roots ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಕ್ಯಾಲ್ಸಿಯಂ ಓಪನ್ ಅಪ್ ಆಗಿ ದೇ ಇನ್ಫ್ಲೂಯೆನ್ಸ್ ಆಕ್ಸಿನ್. ಆಕ್ಸಿನ್ ಬರೋದ್ರಿಂದ ನಿಮಗೆ ರೂಟ್ ಯಾವಾಗಾದರೂ ಸರ್ ನೀವು ತೆಗೆದಿದ್ದೀರಾ, ನೋಡಿದ್ದೀರಾ ರೂಟು ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಕೆಲವೊಂದು ಸಲ ತುಂಬ ದಪ್ಪ ಇರುತ್ತೆ. ಯಾಕೆ ಅಂದರೆ ಟಿಪ್ಪಲ್ಲಿ ಆಕ್ಸಿನ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಆಕ್ಸಿನ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋದು ಯಾಕೆಂದರೆ ಥ್ರೂ ಕ್ಯಾಲ್ಸಿಯಂ. ಆ ಕ್ಯಾಲ್ಸಿಯಂ ಹೆಂಗೆ ಬಂತು? ಬಿಕಾಸ್ ಆಫ್ ದಿ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಬೀಯಿಂಗ್ ಆಕ್ಟಿವೇಟಿಂಗ್ ದ ಆಕ್ಸಿನ್ ಪಾತ್ವೇ. ಸೊ ದಿಸ್ ಇಸ್ ದ ಸೆಕೆಂಡ್ ಇದು. Any doubts, sir? ಸರ್ ಅವಾಗ ಏನು ಮಾಡೋಕ್ಕಾಗಲ್ಲ, ದೇವ್ರ ಪ್ರಾರ್ಥನೆ ಮಾಡ್ಬೇಕಷ್ಟೇ. ಬಿಸಿಲು ಬೀಳಲಿ ಇದನ್ನ ಮಾಡಲಿ ಅಂತ ಅಲ್ಲ. ಅವಾಗ ಅದು ಸರ್ ಇದು ಹೆಂಗಿದೆ ಅಂದರೆ, ನಿಮಗೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಇದೆ, ಅದು ಎಳ್ಕೋಬೇಕು ಸರ್ ಪ್ಲಾಂಟಲ್ಲಿ. ನೀವು ಫೋಟೋಸಿಂಥಸಿಸ್, ಸನ್ಲೈಟ್ ಇಲ್ಲ ಅಂದರೆ ನೀರು ಹೋಗಲ್ಲ ಮೇಲೆ, ಅದ್ರ ಜೊತೆಗೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸು ಹೋಗಲ್ಲ ಮೇಲೆ. ಆಯಿತಾ? ಇದೆಲ್ಲ ಚೆನ್ನಾಗಿದ್ದಾಗ, ಸೈಕಲ್ ಇದ್ದಾಗ ಏನಾಗುತ್ತೆ, ಈ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡು ಚೆನ್ನಾಗಿ ಪಂಪ್ಸ್ನೆಲ್ಲ ಓಪನ್ ಮಾಡುತ್ತೆ, ಅವಾಗ ಎಫಿಷಿಯೆಂಟ್ ಆಗುತ್ತೆ. ವಾಟ್ ವಿ ಕಾಲ್ ಫರ್ಟಿಲೈಸರ್ ಯೂಸ್ ಎಫಿಷಿಯನ್ಸಿ ಅಂತ ಎಲ್ಲ ದೊಡ್ಡ ದೊಡ್ಡ ವರ್ಡ್ ಯೂಸ್ ಮಾಡ್ತೀವಲ್ಲ ಸರ್. ನ್ಯೂಟ್ರಿಯೆಂಟ್, ಇಟ್ ಈಸ್ ದ ಕೋರ್. ದಿಸ್ ಈಸ್ ದ ಕೋರ್. Core is activate your proton pumps. Ensure that nutrients are outside the cell in the cell ಒಳಗಡೆ ಹೋಗಂಗ್ ಮಾಡಿ, and you see the magic. ಇನ್ನು up to suboptimal fertilizer ಹಾಕಿ, but you have a good mechanism to activate your proton pumps, calcium signaling perfect ಮಾಡಿ, you will get same yield. Sir? Yes, yes. The micronutrients ಅಲ್ಲಿ ಯಾವುದೇ ಕಮ್ಮಿ ಮಾಡಿ, calcium ಕಮ್ಮಿ ಮಾಡಬೇಡಿ. Calcium is good. Yeah, see, soil ಅಲ್ಲೂ calcium ಇರುತ್ತೆ. When you have ಇದು, but micronutrients is very underrated, sir, in our country. Underrated, underutilized, and that's also ignored. So they require, but you should have, flowering ಅಲ್ಲಿ boron ಹಾಕ್ಬಿಡಿ calcium. If you ask me, you should have micronutrients from day one. From day germination you should have micronutrients available to your plant. They are small role play ಮಾಡ್ತವೆ, but significant roles play ಮಾಡ್ತವೆ. So have calcium, bore. When you add NPK, you can add a little bit of micronutrients, maybe not to that. Huh? Sir, ನೀವು ಡೋಸು ಫ್ಲವರಿಂಗ್ ಟೈಮಲ್ಲಿ ಜಾಸ್ತಿ ಹಾಕ್ತೀರಾ, ಬಟ್ ಅರ್ಲಿ ಸ್ಟೇಜಲ್ಲಿ ಅದರದ್ದು ಇವನ್ 1/5, 1/6 ಹಾಕಿ. But let there be some micronutrients in the root, also for the foliar. They are all very good. They are small players, sir. Onthara serial actorಗಳಿದ್ದಂಗೆ. They may not be, but they are entertaining us. They are keeping us alive for every day. So they are not, ಯಾರೋ ಒಬ್ಬ ಬಂದು ಒಂದೇ ದಿವಸ ಒಂದು ಹೀರೋ ಫಿಲ್ಮ್ ಮಾಡಿ ವರ್ಷಕ್ಕೊಂದು ಮೂವಿ ಮಾಡ್ತಾನೆ. But we need good TV actors. How many people, Satisha, when he expired, he was a good, we remember him because of that, like that. So, ಇಲ್ಲಿ ನೋಡಿ ಇವ್ರು ಹಾಕ್ತಾರೆ ನೋಡಿ, ಅಲ್ಲಿ ಜಿಪ್ಸಮ್ ಹಾಕ್ತಾರಲ್ಲ, ಜಿಪ್ಸಮಲ್ಲಿ ಎಕ್ಸಾಕ್ಟ್ಲಿ. ಆ್ಯಂಡ್ ಗೊತ್ತಿಲ್ಲ ಸರ್ ಅವ್ರಿಗೆ ಯಾಕೆ ಹಾಕ್ತಾರೆ ಅಂತ. ಚಿಲ್ಲಿ ರೆಡ್ ಕಲರ್ ಯಾಕೆ ಬರುತ್ತೆ ಅದು, ದೇ ಆಲ್ಸೋ ಹ್ಯಾವ್ ಅಬಂಡೆಂಟ್ ಕ್ಯಾಲ್ಸಿಯಮ್ ಇನ್ ದ್ಯಾಟ್. ಮೆನಿ ಪ್ರಾಕ್ಟೀಸಸ್ ಅವರ್ ಫಾರ್ಮರ್ಸ್ ಡೋಂಟ್ ನೋ. ಎಲ್ಲೋ ಒಂದು ಕಡೆ ಜಿಪ್ಸಮ್ ಹಾಕಪ್ಪ ಒಳ್ಳೆ ಕಲರ್ ಬರುತ್ತೆ, ಒಳ್ಳೆ ಇದು ಬರುತ್ತೆ. ಯಾಕೆ ಬರುತ್ತೆ ಅಂತಂದರೆ ಸೆಕೆಂಡ್ರಿ ಮೆಟಬಲೈಟ್ಸ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ ಅದರಲ್ಲಿ. ಹೌದೌದು, ಓಕೆ. ಹೌದು ಹೌದು. ನೋ ಕ್ಯಾಲ್ಸಿಯಂ ವೆರಿ ವೆರಿ there is calcium signaling is the key in many of these ones. So you should have some calcium always in your plants. Okay. So idu second pathway, second way how humic acids help. Now we'll come to third. Nodi ivaga if you take a clay soil, sir, clay soil alli enade nodi idu ಒಂದು clay particle iruttalla, ಈ ಎಡ್ಜ್ ಅಲ್ಲಿ ಪಾಸಿಟಿವ್ಸ್ ಇರುತ್ತೆ, ನೆಗೆಟಿವ್ ಚಾರ್ಜಸ್ ಇರುತ್ತೆ ಒಂದು ಕ್ಲೇ ಪಾರ್ಟಿಕಲ್ ಅಲ್ಲಿ. ಇವೆಲ್ಲ ಅಂಟ್ಕೊಂಡ್ಬಿಡ್ತವೆ, ಅಂಟ್ಕೊಂಡ್ಬಿಟ್ಟು ಬಿಡ್ ಬ್ಲಾಕ್ಸ್ ಮಾಡ್ಕೊಂಡ್ಬಿಡ್ತವೆ. ಸೊ ದೇ ಆರ್ ಲೈಕ್ ದೇ ಬಿಕಮ್ ವೆರಿ ಥಿಕ್ ಕ್ರಸ್ಟ್ ಥರ ಆಗಿಬಿಡುತ್ತೆ. ಏರೇಷನ್ ಇರಲ್ಲ, ಪೊರೋಸಿಟಿ ಇರಲ್ಲ, ಏನಿರಲ್ಲ. ನಾವು ನಮ್ಮ ಹೀರೋ ಎಂಟರ್ ಆಗ್ತಾರೆ ಈಗ, ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್. ಸೊ ವಾಟ್ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್, ದೇ ಆರ್ ಆಲ್ಸೋ ಚಾರ್ಜ್. ಸೊ ದೇ ಬೈಂಡ್ ಟು, ಇಲ್ಲಿ ಪಾಸಿಟಿವ್ ಇರೋದಕ್ಕೂ ಬೈಂಡ್ ಆಗ್ತವೆ, ಈ ಕಡೆ ನೆಗೆಟಿವ್ ಇರೋದಕ್ಕೂ ಬೈಂಡ್ ಆಗಿಬಿಟ್ಟು ಅವೇನು ಮಾಡ್ತಾವೆ, ಪಾರ್ಟಿಕಲ್ಸ್ನ ದೇ ವಿಲ್ ಫ್ರ್ಯಾಕ್ಮೆಂಟ್ ಮಾಡ್ತವೆ, ದೊಡ್ಡ ಇದನ್ನು ಮಾಡಿ. ಅವಾಗೇನಂತ ಏರೇಷನ್ ಇಂಪ್ರೂವ್ ಆಗುತ್ತೆ, ರೂಟ್ಸ್ ಹತ್ರ ವಾಟರ್ ಹೋಲ್ಡಿಂಗ್ ಕೆಪಾಸಿಟಿ ಇಂಪ್ರೂವ್ ಆಗುತ್ತೆ. ಬಿಕಾಸ್ ಆಫ್ humic acid has one tendency where it is the only molecule, not only one, among many molecules that can bind to both positive as well as negative charged particles. ಕೆಲವು ಸುಮಾರು ಪಾರ್ಟಿಕಲ್ಸ್ ಏನಾಗುತ್ತೆ ಬರೀ ಪಾಸಿಟಿವ್ ಬೈಂಡ್ ಆಗ್ತವೆ, ಇಲ್ಲ ನೆಗೆಟಿವ್ ಬೈಂಡ್. ಬಟ್ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ಸ್ ಕ್ಯಾನ್ ಬೈಂಡ್ ಟು ಬೋತ್ ದಿಸ್ ಥಿಂಗ್ಸ್. ಅವಾಗೇನಾಗುತ್ತೆ, ದಿಸ್ ಕ್ಲಂಪ್ ಇರೋದನ್ನು ಇಲ್ಲಿ ನೋಡಿ, ಈ ಥರ ಕ್ಲಂಪ್ ಇರುತ್ತಲ್ಲ ಸಾಯಿಲ್, ದೇ ಬಿಕಮ್ಸ್ ಇದಾಗುತ್ತೆ. ಮೋರ್ ಸಾಯಿಲ್ ಇಸ್ ಮೋರ್ ಪೋರಸ್, more especially clay ಜಾಸ್ತಿ ಇರೋ ಕಾಯಿಲಲ್ಲಿ humic acids play this role here. And second one, ಇಲ್ಲ ಈಟ್ ಜಾಸ್ತಿ ಆಗಿರ್ಬೇಕು ಸರ್ ಇಲ್ಲಿ ಡಿಸ್ಕಷನ್ ಈಟ್. ಓಕೆ, ಸೊ ಲಾಸ್ಟ್ ಇನ್ನೊಂದು ಮೆಕ್ಯಾನಿಸಮ್ ನೋಡಿ. ಇವಾಗ ನಾನು ಹೇಳಿದ್ನಲ್ಲ, ಈಗ ಸೋಡಿಯಂ ಎಲ್ಲೆಲ್ಲಿ ಇರುತ್ತೆ, ರೂಟ್ ಸುತ್ತಮುತ್ತ ಸೋಡಿಯಂ ಇದ್ದರೆ ನೀರಿದ್ರೂ ನೀರು ರೂಟ್ ಹತ್ರ ಬರಲ್ಲ. Water doesn't like to, sodium is like, it usually penetrates. The sodium chloride is okay, up is fine. Sodium chloride can absorb water, you can dissolve it. But very sodium idre, water doesn't really like to come to the core. ಈಗ ರೂಟ್ ಸುತ್ತ ಸೋಡಿಯಂ ಇರುತ್ತೆ. ನಾವು ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಹಾಕಿದ ತಕ್ಷಣ ಏನು ಮಾಡುತ್ತೆ, ಇಟ್ ಈಸ್ ಲೈಕ್ ಎ ಕೀಲೇಟಿಂಗ್ ಏಜೆಂಟ್ ಅಂತಾರೆ. ಒಂದು ಕಡೆ ಸೋಡಿಯಂ ಇಟ್ಕೊಳ್ಳುತ್ತೆ, ಇನ್ನೊಂದು ಕಡೆ ವಾಟರಲ್ಲಿ ಆಕ್ಸಿಜನ್ ಮಾಲಿಕ್ಯೂಲ್ ಹಿಂಗೆ ಇಟ್ಕೊಳ್ಳುತ್ತೆ. ಸೊ ಇಟ್ಕೊಂಡ್ಬಿಟ್ಟು ದೇ ವಿಲ್ ಜಸ್ಟ್ ಬ್ರಿಂಗ್ ದ ವಾಟರ್ ಕ್ಲೋಸ್ ಟು ದ ರೂಟ್ಸ್, ಬಿಕಾಸ್ ಇಟ್ ಈಸ್ ಆಕ್ಸಿಜನ್ ಈಸ್ ನೆಗೆಟಿವ್ಲಿ ಚಾರ್ಜ್, ಸೋಡಿಯಂ ಈಸ್ ಪಾಸಿಟಿವ್ಲಿ ಚಾರ್ಜ್. ದೇ ಕ್ಯಾನ್ ಬೈಂಡ್ ಟು ಆಫ್ ದಿಸ್. ಸೊ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಇವ್ರು ಸರ್ ಹೇಳ್ದಂಗೆ ಎಷ್ಟು ಹಾಕ್ತಿರೋ ಅಷ್ಟು ಒಳ್ಳೆಯದು, ಇದ್ದಷ್ಟು ಒಳ್ಳೆಯದು ರೂಟಲ್ಲಿ ನಿಮಗೆ. ಇದು ಸರ್ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್. So these are the 3 major roles humic acid play. Is it useful? Was it a little understanding was better, or yeah, any questions? So yeah, sir. No, black soil alla. It is, you don't know, wherever soil, sir, I'm not soil scientist, but I can say wherever soils are, charged, soil very reactive, sir. Soil now very underestimate. They are very reactive. Wherever there are charges, they bound. It breaks and makes porous. That's all. Okay, sandy soil alli humic acid hakad waste. Don't put humic acid in sandy soil. But maybe it is you activating the calcium channels. You require that one. But leach out agobidutte adralli. But clay ಜಾಸ್ತಿ ಇದ್ದಷ್ಟು soil alli you add humic acids. Good, very good question, sir. Actually, ನನಗೂ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಗೊತ್ತಿಲ್ಲ. ಬಟ್ ಥ್ರೂಔಟ್ ದ ಕ್ರಾಪ್ humic acid ಇದ್ರೆ ಒಳ್ಳೆಯದು. So at least once in 20 days, once in 30 days, some dose of humic acid early soil giving. ಏನು ಹೇಳಿ ಸರ್? ಡ್ರಿಪ್ಪಲ್ಲಿ ವಾಟರ್ ಸಾಲ್ಯುಬಲ್ ಇದ್ರೆ ಹಾಕಿ, ಇಲ್ಲ ನಿಮಗೆ ಇದಿದ್ರೆ ಡ್ರಿಲ್ ಮಾಡಿ ಗ್ರ್ಯಾನ್ಯೂಲ್ಸ್ ಬಿಡಿ ಒಳಗಡೆ. ಅದು ಡ್ರಿಪ್, ಹಾಂ, ಡ್ರಿಪ್ ಇಸ್ ಓಕೆ, ಡ್ರಿಪ್ ಇಸ್ ಓಕೆ, ಡ್ರಿಪ್ ಇಸ್ ಫೈನ್. ನಾವು ಅದರಲ್ಲೇ ಬಿಡ್ತಾ ಇದ್ವಿ. ಇನ್ನೊಂದು ಇದೆ, ಸಾಯಿಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಸಾಯಿಲ್ ಮೈಕ್ರೋರೈಸಾ ಇವೆಲ್ಲ ದೇ ಲವ್ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್. ಎಷ್ಟು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಥ್ರೈವ್ ಆಗುತ್ತೆ ಒಳಗಡೆ ಅಂದರೆ, ಬಿಕಾಸ್ ದೇ ಯೂಸ್ ಕಾರ್ಬನ್ ಆಸ್ ಎ ಸೋರ್ಸ್ ಫಾರ್ ಎನರ್ಜಿ. ದೇ ಕ್ಯಾನ್ ಯೂಸ್ ಇಟ್ ಫ್ರಮ್ ದಿಸ್ ಒನ್ ಆ್ಯಂಡ್ ದೇ ಕ್ಯಾನ್ ಅದೊಂಥರ ಏನಂದರೆ ಇಟ್ಸ್ ಎ ನೈಸ್ ಡೊಮೇನ್ ಫಾರ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಟು ಥ್ರೈವ್. ಎಲ್ಲಾದರೂ ಕಾರ್ಬನ್ನು, ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸು, ವಾಟರ್ ಬರುತ್ತಂತಂದರೆ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಫಂಗಸ್, ದೇ ಆಲ್ಸೋ ಗ್ರೋ ಇನ್ ದಟ್ ಏರಿಯಾ. ಸೊ ಐ ಹ್ಯಾವ್ ನಾಟ್ ಲಿಸ್ಟೆಡ್ ದಟ್ ಒನ್. Sorry, sir. I don't know such specifics, but humic acid is one thing that even if you add a little more, no problems. This is fine. It is in the soil, but please follow the manufacturers because they would have known the molecule very well. We don't know the molecule so well, no? So that is they will have it. So. Okay. No, they are. See, these bacteria are very specific. Rhizobium, it fixes nitrogen. That's it. No, that's all. Yeah, it is having a separate reaction to do this one. Nothing to do with the humic acids.
Sir.
Oh, good question, sir. I think it should be also other variants, but it is for both. But it is your job not to introduce a lot of pathogenic bacteria in this one. Yes, that is also there. Yeah, exactly. Yeah.
ಈ ಗರ್ಕಿನ್ನಲ್ಲಿ ಸರ್,
ಹೇಳಿ ಸರ್.
ಈಗ ಲಿಕ್ವಿಡ್ ಇದರಲ್ಲಿ ಡ್ರಿಪ್ನಲ್ಲಿ ನಾವು ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಬಿಡ್ತೀವಿ. ಎಷ್ಟು ದಿವಸಕ್ಕೊಂದ್ಸರಿ ಬಿಟ್ಟರೆ ಒಳ್ಳೇದು
ಸರ್? ಕೊಡಿ ಸರ್, 20 ಡೇಸ್ಗೆ ಒಂದ್ಸಲ ಕೊಡಿ.
20 ಡೇಸ್
ಒಂದ್ಸಲ ಕೊಡಿ. ನೀವು ಕಮ್ಮಿ ಡೋಸ್ ಕೊಡಿ.
90 ದಿವಸ ಈ ಗಿಡ ಇರ್ತವೆ.
ನೀವು 20 ಡೇಸಲ್ಲಿ ನೀವು ಡ್ರಿಪ್ಪಲ್ಲಿ ಅದೆಷ್ಟು ರೆಕಮೆಂಡೇಶನ್ ಇದೆ ಅದರದ್ದು ಫುಲ್ ಕ್ರಾಪ್ಗೆ ಎಷ್ಟು ಹೇಳಿರ್ತಾರೆ ಅದನ್ನು ಸ್ಪ್ಲಿಟ್ ಮಾಡಿಬಿಡಿ. ಒಟ್ಟಿನಲ್ಲಿ ಏನಂದರೆ ಒಂದೇ ಸಲ ಎಲ್ಲ ಡಂಪ್ ಮಾಡೋದಕ್ಕಿಂತ ರೆಗ್ಯುಲರಾಗಿ ಕೊಡ್ತಾ ಇರಿ. ರೂಟ್ಸ್ ವಿಲ್ ಬಿ ವೆರಿ ವೆರಿ ಹೆಲ್ದಿ.
ಅದು ಏನು ಕ್ವಾಂಟಿಟಿ ಬೇಕಾಗುತ್ತೆ ಅನ್ನೋದು ನಿಮಗೆ
ನನಗೆ ನಾನು ನೋಡಿಲ್ಲ, ಈ ನಡುವೆ ಲೇಬಲ್ಲೇ ನೋಡಿಲ್ಲ ನಾನು. ಅದು ಸರ್, ಅದು ನಾವು ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದಿದ್ದು 1 kg per hectare ಅಂತ ರೆಕಮೆಂಡೆಡ್ ಡೋಸ್ ಅಲ್ಲ, ಅವ್ರು ಹಂಗೆ ಮಾಡಿರ್ತಾರೆ ದ ಅಮೌಂಟ್ ಆಫ್. ಹಾಂ, ಅಷ್ಟೇ 1 kg per hectare ಇತ್ತು ನಮ್ಮದು ಡೋಸೇಜು. ಆ ರೋಡಲ್ಲಿ ಕೊಡ್ರೆ ಆಯಿತು. ಅದೇ
ಸರ್ 500 ml ಅಂದರೆ ಒಂದು ಡೋಸ್ಗೆ?
Fine. No, you see, they say one application, 1 kg per hectare. They say you can make 2 applications, you can make 3 applications. Okay, usually if it is soil, it should be at least 60, 55 and above. Humic acid should be
ಈಗ
rich in
ಹ್ಯೂಮಿಕ್
humic acid.
ಆ್ಯಸಿಡ್ ಪೊಲಿಕ್ ಆ್ಯಸಿಡ್ ಎರಡು ಒಟ್ಟಿಗೆ ಇರ್ತದೆ ಅದು?
ಸರ್, ಕೆಲವರು ಬರೀ ಯೂಮಿಕ್ ಆಸಿಡ್ ಮಾಡ್ತಾರೆ, ಕೆಲವರು ಬರೀ ಫಲ್ವಿಕ್ ಆಸಿಡೇ ಮಾಡ್ತಾರೆ. ಕೆಲವರು ಫೋಲಿಯರ್ಗೆ ಟೊಮೆಟೊ ಫ್ರೂಟ್ಸ್, ದೇ ವಾಂಟ್ ಟು ಹ್ಯಾವ್ ಎ ವೆರಿ ವೆರಿ ಗ್ಲಾಸಿ ಟೊಮೆಟೊ ಫ್ರೂಟ್ಸ್, ದೇ ಸ್ಪ್ರೇ ಓನ್ಲಿ ಫಲ್ವಿಕ್ ಆಸಿಡ್, ದೇ ಡೋಂಟ್ ಕೇರ್ ಅಬೌಟ್ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್. ಕೆಲವ್ರಿಗೆ ಈ ಲಾಡ್ಜಿಂಗ್ ಆಗುತ್ತೆ, ಗಿಡ ಇದು ಬಿದ್ದೋಗುತ್ತೆ, ಆಮೇಲೆ ರೂಟ್ಸಲ್ಲಿ ಇದು ತಿಕ್ಕಾಗಿ ಫ್ರೂಟ್ ಬರಬೇಕು. ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇರೋದೇ ಮಾಡ್ಬೋದು, ಬರೀ ಹ್ಯೂಮಿಕ್ ಆಸಿಡೇ ಐಸೋಲೇಟ್ ಮಾಡಿರ್ತಾರೆ. ಥರ್ಡ್ ಫ್ರಾಕ್ಷನಲ್ಲಿ ಹ್ಯೂಮಿಕ್ ಇರುತ್ತೆ, ಫಲ್ವಿಕ್ ಇರುತ್ತೆ. ಅದರಲ್ಲಿ ಸಾಯಿಲ್ಗೆ ಹಾಕೋದಾದರೆ ಹ್ಯೂಮಿಕ್ ಆಸಿಡ್ ಲೆಟ್ ಇಟ್ ಬಿ ಮೋರ್ ದನ್ 55%. ಬಟ್ ಫೋಲಿಯರ್ಗೆ ಅಪ್ಲೈ ಮಾಡೋದಾದರೆ ಫುಲ್ವಿಕ್ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇರಲಿ. ದಟ್ಸ್ ಎ ಜನರಲ್ ರೂಲ್ ಅಷ್ಟೇ ನಿಮಗೆ. ನೋಡಿ, ಅದು ನೋಡ್ಕೋಬೇಕು ಸರ್. ಅದು ನೋಡಿದ್ರೆ ವಿ ಕ್ಯಾನ್ ಡೂ ಏನಾದ್ರು ಫಾಲೋ ಅಪ್ ಸೆಷನ್ ಬೇಕಾದ್ರೆ ಮಾಡೋಣ. ನಾನು ಇನ್ನೂ ಅಪ್ಲಿಕೇಶನ್ ಸೈಂಟಿಸ್ಟ್ ಕರ್ಕೊಂಬರ್ತೀನಿ ಯಾರಾದರೂ. ಐ ಥಿಂಕ್ ನಾವು ವಿ ಆರ್ ಗುಡ್ ಇನ್ ಬೇಸಿಕ್ಸ್. ದೇರ್ ಆರ್ ಪೀಪಲ್ ಹೂ ಆರ್ ಫೀಲ್ಡಲ್ಲಿ ಮಾಡಿದ್ದಾರೆ, ವಿ ಕ್ಯಾನ್ ಕಾಂಪ್ಲಿಮೆಂಟ್ ವೆರಿ ವೆಲ್. ಅದು ಬೇಕಾದ್ರೆ ಒಂದು ಸೆಷನ್ ಕೊಟ್ಟರೆ ನಿಮಗೆ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಇವ್ರು ಕೇಳೋ ಕ್ವಶನ್ಗೆ ಎಷ್ಟು ಇಂಟರ್ವ್ಯೂಲಲ್ಲಿ ಯಾವುದು ಬೇಕಾದ್ರೆ ಯಾವ ಕಂಪ್ನಿದು ಎಲ್ಲ ಹೇಳಿಸ್ಬೋದು ನಿಮಗೆ. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ಏನು ಬ್ರೇಕ್ ಬೇಕಾ ಏನಾದರೂ? ಓಕೆ, ಹಂಗೆ ಮಾಡಿ ಇದನ್ನು ಸೆಷನ್ನ ಕಂಬೈನ್ ಮಾಡಿಬಿಡೋಣ. ಇಲ್ಲ, ಇದೆಲ್ಲ ಆಗಿಲ್ಲ, ಬಟ್ ವಿ ವಿಲ್ ನೆಕ್ಸ್ಟ್ ಸೆಷನಲ್ಲಿ ಬಿಫೋರ್ ಲಂಚ್ ವಿಲ್ ಕ್ಲೋಸ್ ಆಲ್ ದೀಸ್ ಥಿಂಗ್ಸ್. ಓಕೆನಾ? ಹೋಗ್ತಾ ಇದೆಯಾ? ಮರ್ತು ಬಿಡ್ತೀವಿ. Next is our favorite. I think most of people have used botanical extract. It's also a big ocean. Many people are giving lot of molecules, confusing people. Some are working, some are not working. Some work only on some crops. People blindly use spraying it on all these things. I know how much it is. It's a mafia. Anything they give you and then nobody has no interest. There is no background information on whether the plant is absorbing that botanical or not, first of all, whether it is needed or not. Okay, so row crops to takondogi fruit and vegetable kodtare. But in this, the most significant is seaweed extracts. Seaweed extracts. So how many people are familiar with seaweed extracts? Okay, seaweed, have you used seaweed extracts or? Okay. Yes. Okay. Okay. So how were you using, sir? Foliar application or? And what frequency, time you used? Pre-flowering time. Okay, good.
It was a part of a package.
Okay.
Every company, most of the companies, bio-stimulants.
And what was the objective? What were why did you use it? So for what?
To use the flower.
You have gibberellic acid, right? Why?
But still actually, they brought like that and people started using. And now people are stopped. They're using their
Okay, good. So what I'll tell you is, I can give you a little background how biostimulant, the seaweed extracts. So there are two things. There is seaweed extract. Please note, sometimes seaweed is, it's ಸಮುದ್ರದಲ್ಲಿ ಬೆಳೀತಾ ಇರೋದು ಇದು ಕಳೆ ಅಂತಲ್ಲ ಸರ್, ಕಳೆ ಅನ್ಲೇಕೆ ಹೋಗ್ಬೇಡಿ. It is a $500 billion business in the world. ನೀವು ತಿಂತಾರೋ noodles is coming from seaweed. They are using the alginate, which is a carbohydrate. When you add it to the carbohydrate, then it becomes sticky. And China is thriving on seaweeds. Seaweed is a $500 billion business. It's become $1 trillion. Medicine, food, preparations, gel ಮಾಡ್ತಾರೆ ನೋಡಿ, gels, gummies, ಎಲ್ಲದರಲ್ಲೂ seaweed, the extracts are used. Very, very important crop. ನಮ್ಮ ಇಡೀ ಇಂಡಿಯನ್ ಅಗ್ರಿಕಲ್ಚರ್ ಎಲ್ಲ ಸೇರಿಸಿದ್ರೆ ಅಷ್ಟಿದೆಯಲ್ಲ ರೆವೆನ್ಯೂ. ಸೀವಿಡ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಇನ್ ಸೌತ್ ಏಷ್ಯಾ, ಚೈನಾ, ಮಲೇಷ್ಯಾ, ಇಂಡೋನೇಷ್ಯಾ, ಫಿಲಿಪೀನ್ಸ್ ಅಲ್ಲೇ ಬರೋದು. ಸೊ ಇಟ್ಸ್ ಅ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಕ್ರಾಪ್. Though it is called a seaweed, of course, it's a weed, but it's a useful weed. Yes, sir. No, there are two things. It is actually naturally available, but they are cultivating it. So they put threads like this, they put stones in the sea, huge stone, and then they are tying this piece of seaweed all through this thing, and it grows. It is harvesting nutrients from the sea water. There are a lot of sea water, and then it is photosynthetic algae. It's called macro algae. We have micro algae. Ponds allella ಬೆಳೆಯುತ್ತಲ್ಲ, that is micro algae. This is macro algae. And there are three types: brown algae, red algae, green algae. If you ask which is ಮೂರಲ್ಲಿ ಯಾವ್ದು better, there is no answer. No answer for this thing. But generally, brown algae are most used. More, you can see number of species. Brown algae is more than compared to red algae. Second is, you cannot find brown algae everywhere. It is only in certain regions they grow. For example, in Tamil Nadu, in Karnataka, in India, the whole of coastal area, they grow Capophycus, which is so this species. Oh, sorry. Capophycus. This is very much grown in the in Tamil Nadu. Now, whereas Ascophyllum nodosum, this is grown in Norway. This Ascophyllum nodosum grows only in cold water. It has to very, very cold water, then only it grows. Now, Capophycus grows in warm water. in Tamil Nadu. Now, compared to both, Ascophyllum is here in terms of quality. Capophycus is here. And the reason is, it is a tropical seaweed. It is always stressed. It is growing in heavy sunlight. And whatever metabolite it is producing, it is for its own survival. So, ಆ seaweed ಅಲ್ಲಿ ಏನೇನು ಬರುತ್ತೆ ಅದು it is actually used ಅದು ಬದುಕಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ ಅದು ಜಾಸ್ತಿ. ಹಂಗಾಗಿ ಅಲ್ಲಿರೋ ಮೈಕ್ರೋನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಆಗ್ಬೋದು, ಫ್ಲೇವರ್ಸ್ ಆಗ್ಬೋದು, ಫ್ಲೇವನಾಯ್ಡ್ಸ್ ಆಗ್ಬೋದು, ಮಿನರಲ್ಸ್ ಆಗ್ಬೋದು, ಎಲ್ಲ ಈ ಟ್ರಾಪಿಕಲ್ ಸೀವೀಡ್ಸ್ ಅವೇ ಯೂಸ್ ಮಾಡ್ಕೊಂಡ್ಬಿಡುತ್ತೆ. ಸೊ ಇಟ್ ಈಸ್ ಬೆಟರ್ ಟು ಗೋ ಫಾರ್ ಕೋಲ್ಡ್ ವಾಟರ್ ಸೀವೀಡ್ಸ್, ಬಿಕಾಸ್ ಅಲ್ಲೇನಾದ್ರೆ ದೇ ಅಕ್ಯುಮುಲೇಟ್ ಲಾಟ್ ಆಫ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್, ದೇ ಆರ್ ನಾಟ್ ಸ್ಟ್ರೆಸ್ಡ್. So today, number one, this thing is Ascophyllum, and it is little expensive, but it is known to do it. So there are three categories: brown, red, and algae. And this one belongs to the brown algae, the Ascophyllum. There are also other species. A lot of literature is there, but you have to read and accumulate all the information. That is good. But I can recommend any company providing Ascophyllum. Then you can. And if they say, where is this Ascophyllum? If they say Ascophyllum manufactured in Malaysia, throw that bottle first. Because you cannot get Ascophyllum anywhere here. You can get only in Europe, North America, that part only. Okay, so if they say this Ascophyllum is made from Philippines, Indonesia, it is fraud. It is like that. They do all those things anyway. Okay, now, what is there? What is the composition of seaweed? Like, I don't know, you might have known. It is rich in carbohydrates, just like our thing. But only difference is, for example, if you say rice is also rich in carbohydrate, but it is starch. It is starch. So rice also has more than 70-80% probably, carbohydrates. Seaweed also has the same carbohydrates, but these are all very, very small chain, basic carbohydrates. So you get glucose, you get oligosaccharide, glucose, sucrose, starch. Ee tara hogutte. Small is glucose, then sucrose is second. Then idralli most of the fraction is the small ones, which is the glucose, their derivatives. They never make starch. So you can eat a lot of seaweed, you will not get any sugar or anything. It's like really very healthy plant to have it. And then they have a lot of proteins also. It's a protein, 15% protein. So animal feed kodtare. They use seaweed is a very good animal. For example, you can take away 50% is around soybean protein kodtare. So 15% they can remove from that one. Other seaweed kodbodu nimge poultry feed ge. Many people are using it, and it is very healthy also. That's one. So protein irutte. ಆಮೇಲೆ ಲಾಟ್ ಆಫ್ ಮಿನರಲ್ಸ್ ಬರುತ್ತೆ ನೋಡಿ, ಮಿನರಲ್ಸ್ ವೆರಿ ಗುಡ್ ಸೋರ್ಸ್ ಆಫ್ ಮಿನರಲ್ಸ್. ಮಿನರಲ್ಸ್ ಅಲ್ಲ, ಮಿನರಲ್ಸ್ ಇದೆ, ಮಿನರಲ್ಸ್ ಕಮ್ಮಿ ಇದೆ, 2% 3%. ಅದರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಎಲ್ಲ ಸೆಕೆಂಡರಿ ಮೆಟಬಲೈಟ್ಸ್ ವಿಚ್ ಯು ಹವ್ ನೆವರ್ ಹರ್ಡ್ ಆಫ್. ದಟ್ ಈಸ್ ವೈ ದೇ ಆರ್ ಯೂನಿಕ್ ಈಸ್ ದೇ ಹ್ಯಾವ್ ವೆರಿ ವೆರಿ ಯೂನಿಕ್ ಫಂಕ್ಷನ್ಸ್. ವಿ ಆರ್ ನಾಟ್ ಡೋಂಟ್ ಸಿ ಇಟ್ ಇನ್ ಆಲ್ ದೀಸ್ ಆರ್ಗನಿಸಮ್ಸ್ ಡೆವಲಪ್. ಸೊ ಪೀಪಲ್ ಡೂ 2 ಥಿಂಗ್ಸ್. ಸೀವೀಡ್ ತಗೊಳ್ತಾರೆ, ಪುಡಿ ಮಾಡ್ತಾರೆ, ಡ್ರೈ ಮಾಡ್ತಾರೆ, ಪುಡಿ ಮಾಡ್ತಾರೆ, ಪುಡಿ ಮಾಡಿ ದೇ ಯೂಸ್ ಇಟ್ ಆಸ್ ಸಾಯಿಲ್ ಅಪ್ಲಿಕೇಶನ್, ಅಮೆಂಡ್ಮೆಂಟ್. ಆರ್ನಮೆಂಟಲ್ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ದೇ ಕ್ಯಾನ್ ಆಲ್ಸೋ ಆ್ಯಡ್ ಇಟ್. ಆರ್ ಸೀವೀಡ್ ತಗೊಳ್ತಾರೆ, ತಗೊಂಡು ದೇ ಮೇಕ್ ಲಾಟ್ ಆಫ್ ಆ್ಯಸಿಡ್ ಹೈಡ್ರೋಲಿಸಿಸ್, ಎನ್ಸೈಮ್ ಹೈಡ್ರೋಲಿಸಿಸ್, ಆ್ಯಸಿಡ್ ಹಾಕಿ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ತೆಗಿತಾರೆ. ಎಕ್ಸ್ಟ್ರಾಕ್ಟಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಎಲ್ಲ ಇದು ಮೇಜರ್ ಇದರಲ್ಲಿ ಏನೇನಿದ್ವಲ್ಲ ಇವೆಲ್ಲ ಬಂದ್ಬಿಟ್ಟು ಸೆಲ್ ವಾಲ್ ಮಾತ್ರ ಅದು ಉಳ್ಕೊಳ್ಳುತ್ತೆ. ಆ ಫ್ರಾಕ್ಷನ್ ಬರುತ್ತೆ, ಅದನ್ನು ಡ್ರೈ ಮಾಡ್ತಾರೆ, ಡ್ರೈ ಮಾಡಿ ಆ ಪೌಡರ್ ಕೊಡ್ತಾರೆ ನಿಮಗೆ. ದಟ್ ಈಸ್ ವಾಟ್ ಮೆಜಾರಿಟಿ 90% ಆಫ್ ದ ಪೀಪಲ್ ಯೂಸ್ seaweed extracts, not seaweed. Okay, so far so good. Okay, ಸ್ವಲ್ಪ ಕಾಂಪ್ಲಿಕೇಟೆಡ್ ಇದೆ ಹೌ ದೇ ವರ್ಕ್. ಸೀವೀಡ್ಸ್ ಅಲ್ಲಿ ದೇ ಹ್ಯಾವ್ ಸಮಥಿಂಗ್ ಕಾಲ್ಡ್ ದಿಸ್ ಮೆಟೀರಿಯಲ್ ಇದಂತ ನೋಡಿ, ಕ್ಯಾರಗಿನಿನ್, ಲ್ಯಾಮಿನೇರಿನ್ಸ್, ಆಲ್ಜಿನೇಟ್ಸ್, ವುಲ್ವಲ್, ಅಮೈನೋ ಆಸಿಡ್, ಪೆಪ್ಟೈಡ್ಸ್. ಇವು ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಫ್ರಾಕ್ಷನ್ ಹೇಳಿದ್ನಲ್ಲ, ಸಿಂಪಲ್ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್. ಈ ನಾವು ಗ್ಲೂಕೋಸ್ ಅಂತೀವಲ್ಲ, ಸಿಮಿಲರ್ ಟು ಗ್ಲೂಕೋಸ್ is carrageenan, laminarins are there. These are all same family of carbohydrates. ಅವು ಸ್ಪೆಷಲ್ ಮಾಲಿಕ್ಯೂಲ್ಸು, ಬಟ್ ದೇ ಆರ್ ವೆರಿ ರಿಯಾಕ್ಟಿವ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಬರ್ತವೆ ಅದರಲ್ಲಿ. ಸೊ ವಾಟ್ ದೇ ಡೂ ಈಸ್ ವೆನ್ ಯು ಸ್ಪ್ರೇ ದಟ್ ಬಯೋಸ್ಟಿಮ್ಯುಲೆಂಟ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಆನ್ ಟು ಪ್ಲ್ಯಾಂಟ್, ನಿಮ್ಮ ಸೆಲ್ಲಲ್ಲಿ ಸೆಲ್ ವಾಲಲ್ಲಿ ಇಲ್ಲಿ ನೋಡಿ ಇವೆಲ್ಲ ರಿಸೆಪ್ಟರ್ಸ್ ಅಂತ ಇರುತ್ತೆ, ಕಾಯ್ಕೊಂಡು ಕೂತಿರ್ತವೆ. ಈಗ ಸಿಗ್ನಲ್, ಈಗ ಸ್ಟ್ರೆಸ್ ಸಿಗ್ನಲ್ ಕ್ಯಾಚ್ ಮಾಡ್ತವೆ, ಸೆಲ್ಗಳಿದ್ದು ಏನು ಮಾಡೋದು, ಈ ಕಡೆ ಪ್ಯಾಥೋಜನ್ ಸಿಗ್ನಲ್ ಕ್ಯಾಚ್ ಮಾಡುತ್ತೆ, ಏನು ಮಾಡಬೇಕು ರೆಸ್ಪಾನ್ಸು ಅಂತ ಕೊಡುತ್ತೆ. ಸೊ ದೇರ್ ಆರ್ ರಿಸೆಪ್ಟರ್ಸ್ ದೇರ್. ದೀಸ್ ಸೀವಿಡಲ್ಲಿರೋ ಇವೆಲ್ಲ ಇದಾವಲ್ಲ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ಸು, ದೇ ಬೈಂಡ್ ಟು ಲಾಟ್ ಆಫ್ ದೀಸ್ ರಿಸೆಪ್ಟರ್ಸ್ ಇನ್ ದ ಪ್ಲಾಂಟ್. ದ ಮೊಮೆಂಟ್ ದೇ ರಿಸೆಪ್ಟರ್, 3 ಪಾತ್ವೆ ಕ್ರಿಯೇಟ್ ಮಾಡಿದೆ, 3 ಥರ ರೆಸ್ಪಾನ್ಸ್ ಕ್ರಿಯೇಟ್ ಆಗಿಬಿಡುತ್ತೆ ಇಮಿಡಿಯೇಟ್ಲಿ ಪ್ಲಾಂಟಲ್ಲಿ. ಒಂದು ರೆಸ್ಪಾನ್ಸ್ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಡಿಫೆನ್ಸ್ ಜೀನ್ಸ್ ಆ್ಯಕ್ಟಿವೇಟ್ ಆಗುತ್ತೆ. ಸೊ ಇಫ್ ಎಕ್ಸಾಂಪಲ್ ಏನು ಪ್ಯಾಥೋಜನ್ ಬಿತ್ತು, ಇನ್ಸೆಕ್ಟ್ ಬಿತ್ತು, ಅದಕ್ಕೆ ಏನೇನು ಬೇಕು, ಆಲ್ ದೋಸ್ ಪ್ರೋಟೀನ್ಸ್, ಡಿಫೆನ್ಸ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್, ಟಾಕ್ಸಿನ್ಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಇನ್ ದ ಪ್ಲಾಂಟ್. ಸೆಕೆಂಡ್ ಈಸ್ ಆ್ಯಂಟಿಆಕ್ಸಿಡೆಂಟ್ಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್. ದಟ್ಸ್ ವೈ ಸ್ಟ್ರೆಸ್ ಇದ್ದಾಗ ಯಾಕೆ ನೀವು ಹಾಕ್ತೀರಾ ಇದು ಅಂತಂದರೆ, ದೇ ಪ್ರೊಡ್ಯೂಸ್ ಲಾಟ್ ಆಫ್ ಆಸ್ಕೋಬೇಟ್, ಶುಗರ್ಸು, ಸೋಡಿಯಂ ರೆಗ್ಯುಲೇಟರ್ಸು, ಪ್ರೋಲಿನ್ನು, ಈ ಥರದ್ದೆಲ್ಲ ಆ್ಯಂಟಿ ಆಕ್ಸಿಡೆಂಟ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಸೊ ದೇ ಪ್ರೊಟೆಕ್ಟ್ ದ ಪ್ಲಾಂಟ್ ಫ್ರಮ್ ದಿಸ್ ಒನ್. ಆ್ಯಂಡ್ ದ ಥರ್ಡ್ ಒನ್ ಈಸ್ ದೀಸ್ ಹ್ಯಾವ್ ಎ ವೆರಿ ವೆರಿ ಬಿಗ್ ರೋಲ್ ಇನ್ ಫೋಟೋಸಿಂಥಸಿಸ್. ನಿಮ್ಮ ಪ್ಲಾಂಟಲ್ಲಿ ಫೋಟೋಸಿಂಥಸಿಸ್ ಆಗುತ್ತೆ, ಆಮೇಲೆ ಈ ಫೋಟೋಸಿಂಥಸಿಸ್ಸು ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ಸ್ ಆಗೋದು, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಮೆಟಬಾಲಿಸಮು, ಎವ್ರಿಥಿಂಗ್ ಆಗಿ ಈ ಕ್ಲೋರೋಫಿಲ್ ಕಂಟೆಂಟ್ ಗ್ರೀನ್ ಆಗುತ್ತೆ. ಹೌ ಮೆನಿ ಪೀಪಲ್ ಹ್ಯಾವ್ ಸೀನ್? ಸೊ ದ ಕಲರ್ ಚೇಂಜಸ್ in a good biostimulant extract, fruit color changes, leaf glossiness improves, all because the chlorophyll, stomata, photosynthesis, phytohormone stimulation, they all are activated because they trigger the plant. They tell the plant to please produce more anthocyanins, to produce more. So they have that ability, and plant will not listen to anything, but they will listen to these small molecules in seaweed extracts. So very straightforward. Seaweed extracts do only one thing, is the molecules activate defense, nutrient metabolism, and then quality fruit color ಹೆಂಗೆ ಬೇಕೋ ಅದಕ್ಕೆ ಇಂಪ್ರೂವ್ ಆಗೋದಕ್ಕೆ ಏನೇನು ಬೇಕೋ ಅದನ್ನೆಲ್ಲ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ಸೋಕ್ಕೆ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಮಾಡುತ್ತೆ. The ideal definition of biostimulant actually fits for seaweed only. Seaweed is the only one that stimulates the plant. That's why if you see what is the best definition, seaweeds actually are the right candidates. They stimulate the plant to do what you want. Good color, good fruit, good this one. Okay, it's very simple. Any questions on this biostimulant? Yes, sir.
So you recommend seaweed extract for spray?
Spray only, because this is nobody uses seaweed for a root. But the seaweed weed itself, that can be used because it has a lot of fiber. It's not so ideal, but extract only, extract foliar, no complication. And you can use before flowering. If you use vegetative, nitrogen will be, uptake will be improved, photosynthesis will be improved, seedling growth. But if you use seaweed during the flowering and fruiting stage, flower set will be improved, fruit set will be improved, fruit colors, flavors, because it activates a lot of pathways. And you can spray as many times as possible, no harm. They will just not, it's okay, regularly, small dose, you can spray. Unlike phytohormones, instead of 100 ppm, if you spray 300 ppm, gone, they will retard. Instead of growth promotion, there will be growth retardation. Because that's why I said in the beginning, they're very specific. Phytohormones, seaweed extract, no issues. You can, little bit here and there, more sprays, no harm. Plants will regulate them so nicely. So yes, if you have the money, if you have thing, I'll tell you one example where they're using, why they're using, and that where it makes sense. You can use it, but it improves the quality more than the quantity. Seaweeds, yes, quality. Photosynthetic quality is improved. Of course, yield will be improved, but more than yield, if you want like, for example, apple growers, they use it. So for example, flower growers, they use lot of seaweed extract because the petunia, gerbera, flower quality, flower drooping, everything is improved, stem strength, all those, because they are continuously active. They spray a lot of seaweed extracts in Spain, Mexico, US, here. Floricultural seaweeds are very good. Okay. So, huh?
Biosim was manufactured by
Okat, yeah. Unlike humic acid, seaweed extracts are not so much adulterated in the market. Only thing is you check whether it is a brown seaweed, red seaweed or this one. Brown seaweed, and if it is a kelp antare. Have you heard the word kelp anywhere? K-E-L-P, kelp. Kelp is cold water. So cold water alli tegdiro seaweed na, it is called kelp. And kelp is, nobody says seaweed. The word in, if you go in Europe or everywhere, they say we applied kelp antare. Kelp is nothing but seaweed, but ascophyllum, cold water, not only that. Okay. Fine. Protein hydrolysates. How many people have used protein hydrolysates? No. Okay. Nobody is used? What? Okay, you can't see much here. So, again, what they do is, again, see, it is nitrogen. Protein alli the nitrogen irutte. So, protein, being protein assimilates nitrogen. For example, leaf alli there is nitrogen, there is abundant nitrogen in the root, but the leaves are not able to take up. Protein hydrolytes trigger that pathway. Then the plant will say, okay, now I am activating a lot of genes. So the protein, the nitrogen that is present in the sun is taken up effectively with the protein hydrolysates. And second is, even the amino acid are like building blocks. Just like what we do when we do exercise, when we take protein supplement, amino acids also help in building blocks. They help in, ಹೊಸ protein ಮಾಡೋದಕ್ಕೆ help ಮಾಡುತ್ತೆ. ಇದು ಇಲ್ಲ nitrogen ಜಾಸ್ತಿ ಮಾಡೋದಕ್ಕೂ proteins ಜಾಸ್ತಿ ಮಾಡುತ್ತೆ. And and they also have antioxidants, so flavonoids, so flavors adunu improve ಮಾಡುತ್ತೆ. Only concern here is protein hydrolysates don't work on all the crops. It has to be little bit researched, because some crops are responsive for protein hydrolysates, some are not responsive to this. It's a very specialized molecule. Protein hydrolysate can be considered as a specialized biostimulant, whereas humic acid, seaweeds are general biostimulants. They are like, you throw on any crop, they will have some effect. So plus, ಎರಡನೇದು, how is this protein molecule going into the plant? That is important. Just spraying is not important. Maybe nutrients are taken, pesticides is taken. The protein plant ಒಳಗೆ ಹಂಗೆ ಹೋಗುತ್ತಾ ಅಂತ ನೋಡಬೇಕು ಅದನ್ನ. So people use in seedling stage, flowering stage. More research is required for this one. Even our understanding protein hydrolyzer is not top notch in this area. That's well I can conclude, but I'll share some good findings in Europe or US where they have successfully used in cucumbers or something. Maybe there is something, it can change your, one application can change you also. It is? Okay, yeah. Okay. So maybe then it is, gherkins are responsive to protein hydrolysates. So it is, for example, if you use it on row crops like cereals, wheat, ragi, there it's little less. Maybe it's because the leaf area is bigger in gherkins compared to, so protein has more time to absorb and imbibe. Protein, see the results we see. ಹೌದು, ಓಕೆ, ನನಗೆ ಅಷ್ಟು ಅರ್ಥ ಇಲ್ಲ ಸರ್. protein hydrolysates, how they work in this one. Okay, okay, good. Maybe then you already have some support on that one. Okay. Transparents, very new for you guys, but ನಮ್ಗೆ ಏನನ್ಸುತ್ತಂದ್ರೆ we should explore this. So ನಾನು what ಲಕ್ಷ್ಮೀಶ್ಗೆ ಹೇಳ್ತೀನಿ, I will also find some companies who are making these anti-transparents. Basically, ಒಂದು film ಮಾಡುತ್ತೆ, they can make a film on the leaves, like they can spread it like, and water evaporate agdange, they will help you. They break the surface tension of water and they spread all over the leaf, so water is not evaporated. That is one mechanism. Second one is stomata gottu. Stomata is the, even the leaf tagondre under surface maximum stomata irutte, melenu irutte, but under surface just that is the where oxygen, water enters. Water and oxygen is exchanged in this one. If you keep this tomato open for a long time, water rapid evaporation agutte. Drought alli abscisic acid produce agid takshana tomato close. They close this one. And transparents ಏನು ಮಾಡುತ್ತೆ, they don't close them fully, they don't open it, but they keep them partial so that water loss is not happening. which is good if it is a very very bright sunny drought ಅಷ್ಟೊಂದು ಸನ್ಲೈಟ್ ಬೇಕಾಗಿಲ್ಲ. ಸೊ ಅಲ್ಲಿ ಟ್ರಾನ್ಸ್ಪರೆಂಟ್ಸ್ ಆಂಟಿ ಟ್ರಾನ್ಸ್ಪರೆಂಟ್ಸ್ ಥರ ಬರುತ್ತೆ. ದೇ ಆರ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ಕೆಮಿಕಲ್ಸ್, ವಾಟ್ರಲ್ಲಿ ಮಿಕ್ಸ್ ಮಾಡಿ ಸ್ಪ್ರೇ ಮಾಡ್ತಾರೆ ಇದನ್ನು. ಸೊ ಕೆಲವೊಂದು ಸಲ ಫೊಲ್ವಿಕ್ ಆ್ಯಸಿಡ್ ಜೊತೆನೂ ಕಂಬೈನ್ ಮಾಡ್ತಾರೆ, ಫೊಲ್ವಿಕ್ ಆ್ಯಸಿಡ್ ಜೊತೆ ಕಂಬೈನ್ ಮಾಡ್ತಾರೆ. ಏನೇನೋ ಬಂದಿದೆ, ಇಂಡಿಯಾದಲ್ಲಿ ಎಷ್ಟಿದೆ ಅಂತ ಗೊತ್ತಿಲ್ಲ ನನಗೂ, ನೋಡಬೇಕು ಇಲ್ಲಿ ಮಾರ್ಕೆಟಲ್ಲಿ ಹೂ ಆರ್ ದ ಒನ್ಸ್ ಹೂ ಆರ್ ಮೇಕಿಂಗ್ ಆಲ್ ದೀಸ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಅಂತ. ಆ್ಯಂಡ್ ದ ಥರ್ಡ್ ಒನ್ ಈಸ್ ರಿಫ್ಲೆಕ್ಟಿವ್, ಜಸ್ಟ್ ಲೈಕ್ ಸನ್ಲೈಟ್ನ ರಿಫ್ಲೆಕ್ಟ್ ಮಾಡ್ತವೆ, ದಿ ಜಸ್ಟ್ ರಿಫ್ಲೆಕ್ಟ್ ಇಟ್. Okay, so basically I don't know, they classify as biostimulant. Typically they are not biostimulants. It is, so this is all physical. Nothing happens in the cellular level except the stomata closure, but most of it is on the leaf surface. That is transparent. Now coming to the most interesting thing which you should actually include, just like humic acid, is the cell free extract. Now you see here, there must be at least 20 molecules which are produced around it, and these are medicinally important, very very economically important. Ivella cell produce ಮಾಡಿ cell ಇಂದ ಆಚೆ ಬಿಡುತ್ತೆ ಬ್ಯಾಕ್ಟೀರಿಯಾ. They can have medicinal properties. Our nutrition food ಗೂ ಯೂಸ್ ಮಾಡ್ಬೋದು, ಥೆರಪ್ಯೂಟಿಕ್ಸ್ ಯೂಸ್ ಮಾಡ್ಬೋದು, wound healing ಯೂಸ್ ಮಾಡ್ತಾರೆ, ಏನೇನಕ್ಕೂ ಅಪ್ಲಿಕೇಶನ್ಸ್ ಇರುತ್ತೆ ಇದೆಲ್ಲ. ಆ್ಯಂಡ್ bacteria produce this because to protect itself from the thing. Now agriculturists are smart, they have isolated this. Ella tagondbittu, it is like, see sir, when you take a cell-free extract from a bacteria, you are taking 10 products into one product. You can take one product alli yado ಒಂದು ascorbate irutte, ಅಥವಾ idirutte, organic acid irutte, citric acid irutte, xylanose irutte, pyrimidose irutte, ಈ ಥರ ಏನೇನೋ ಇರುತ್ತೆ. Instead of taking all the ಒಂದೇ ಒಂದು ಇದರಲ್ಲಿ you will have all the products together. Rich cocktail of metabolites. Which product, how it behaves, we don't know fully, but everything will have some impact on the plant. So, and they are all positive impact. Moreover, there are many bacteria which produce similar auxin that is produced in the plant, is produced in the cell. They produce similar gibberellic acids, they produce similar plant hormones. So you, instead of you spraying external phytohormones, just use the cell extract. So you take a bacteria, growing it, 5 days after growing it, the ಮೇಲೆ ಲಿಕ್ವಿಡ್ ಇರುತ್ತೆ, ಅಂದರೆ ನೀವು ಸೆಂಟ್ರಿಫ್ಯೂಜರ್ ಸ್ಪಿನ್ ಮಾಡಿ ಬ್ಯಾಕ್ಟೀರಿಯಾದ ಒಂದು ಲಂಪ್ ಬರುತ್ತೆ, ತೆಗೆದು ಬಿಸಾಕ್ತಾರೆ. That liquid is the rich liquid in full of metabolites. They will when you put it onto the soil or young seedlings, they trigger the enormous root growth ಮಾಡುತ್ತೆ, or they put up more shoot growth. Phytohormones ಇರೋದ್ರಿಂದ ಯಾವಾಗ ಸ್ಟ್ರೆಸ್ ಬಂದರೂ they will control the stress. So this is you should really explore. Not many companies can do this one. ಯಾಕೆ ಅಂತ ಹೇಳ್ತೀನಿ, it is not easy to formulate, because when you take the extract, ಡ್ರೈ ಮಾಡಿ ಪೌಡರ್ ಮಾಡಿದ್ಮೇಲೆ ಎಷ್ಟು ದಿವಸ ಸ್ಟೇಬಲ್ ಇರುತ್ತೆ ಅಂತ ಗೊತ್ತಿರಲ್ಲ. ಅದು ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ಕ್ಯಾನ್ ಬಿ ಸ್ಟೇಬಲ್ ಫಾರ್ 6 months, ಅದರ್ ಮಾಲಿಕ್ಯೂಲ್ ಕ್ಯಾನ್ ಡೈ ಇನ್ 2 weeks, one is stable for 3 weeks. So companies cannot give guarantee to this one. So the trick is there. Many sugarcane farmers, soybean farmers in Brazil, what they do is, ಇಷ್ಟು ಎಕರೆ ಒಂದು 400 acres ಫಾರ್ಮ್ ಇರುತ್ತೆ ಅವ್ರದ್ದು. ಐ ವಿಸಿಟೆಡ್ ದೇರ್. ಫಾರ್ಮ್ ಇರುತ್ತೆ, ಫಾರ್ಮಲ್ಲಿ ದೇ ಪುಟ್ ಒಂದು ಇನ್ಕ್ಯುಬೇಟರ್ ಹಾಕ್ತಾರೆ. ಇನ್ಕ್ಯುಬೇಟರ್ ಹಾಕ್ಬಿಟ್ಟು ಸ್ವಲ್ಪ ಅದಕ್ಕೆ ಮೀಡಿಯಾ ಹಾಕಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹಾಕ್ತಾರೆ. ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹಾಕ್ಬಿಟ್ಟು ದೇ ಗ್ರೋ. ಆಫ್ಟರ್ ದಟ್ ಹೋಲ್ ಲಿಕ್ವಿಡ್ ಈಸ್ ಪುಟ್ ಇನ್ ಟು ದ ವಾಟರ್ ಟ್ಯಾಂಕ್ ಆ್ಯಂಡ್ ಸ್ಪ್ರೆಡ್ ಟು ದ ಫೀಲ್ಡ್. ಫ್ಲಡ್ಡಿಂಗ್ ಆರ್ ಡ್ರಿಪ್, ವಾಟ್ ಎವರ್ ಇಟ್ ಈಸ್. And they do continuously, no filtration, no nothing, no quality. They are getting naturally, they are reducing fertilizers by 50%. They don't spray pesticides. This rich metabolites coming from bacteria is helping their plants thrive with all this. So idu cell-free extracts products are very rare in the market. You cannot really get this cell-free extract. It's very, very, otherwise only thing is you can make is somebody can ferment it and give drums together. Now IHR technology has this, AMC technology. So AMC technology, there are two things. There is cells, there is also metabolites in that one. They spray on pomegranate. They spray on all these things. Suddenly they see improvement in color. The improvement in root, this thing, is because of this rich metabolites that they are doing. And once the metabolites are there, mildew, leaf blot, any fungal pathogen don't like to come there. They don't grow. That is how it is. It makes plants very, what you call, resistant to diseases. Now, alternatively, if you can't take the cell-free extract, go for the cells itself. Andre the cell-free ತಗೊಳಕಲ್ಲ ಬ್ಯಾಕ್ಟೀರಿಯಾನೇ ತಗೊಂಬಿಡಿ. So this is very interesting, and I think every Gherkin grower should have a plant growth promoting microbes in your soil. Humic acid ಜೊತೆ ಇದು ಇರಲೇಬೇಕು. Now ಇಲ್ಲಿ there are two types of ಇದರಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬರುತ್ತೆ ಒಂದು, then you have fungus, phosphorus solubilizing fungus, and then third one is mycorrhiza. So these two are related to phosphorus, whereas the gram positive, gram negative bacteria, they do a lot of things. Basically nitrogen metabolism, zinc metabolism, iron metabolism, ella madutte. These are all. And if you see the role, ಏನೇನು ಮಾಡ್ತಾವೆ ಇವು ಅಂತಂದ್ರೆ, they can affect the roots, they can affect the plants and the foliar. These ಅಂದ್ರೆ you are putting ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಪ್ಲಾಂಟ್ ಮೇಲೆ ಹಾಕ್ತಾ ಇದ್ದೀರಾ ನೀವು. ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಫಂಗಸ್ನ, ಅದೇ ಬ್ಯಾಕ್ಟೀರಿಯಾ ನಮ್ಮ ಮೈ ಮೇಲೆ ಹಾಕ್ಕೊಂಡ್ರೆ we will get infected. But in the plant, you are putting it. But ಇದು ಒಳ್ಳೆ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ರಿಸರ್ಚ್ ಮಾಡಿ ಅದು ಪ್ಲಾಂಟ್ ಜೊತೆ ವರ್ಷಾನುಗಟ್ಟಲೆ ಇದೆ ಸಾಯಿಲಲ್ಲಿ, ಇದರಲ್ಲಿ ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಐಸೋಲೇಟ್ ಮಾಡಿ ವಿ ಆರ್ ಪುಟಿಂಗ್ ಬ್ಯಾಕ್ ಟು ದ ಪ್ಲಾಂಟ್. ಸೊ ಅವೇನು ಮಾಡ್ತವೆ, ಪ್ಲಾಂಟ್ ಜೊತೆ ಸಿಂಬಯಾಟಿಕ್ ಆಗಿ ಕೆಲಸ ಮಾಡುತ್ತೆ. ಸೊ ಈಗ ಇದರಲ್ಲಿ ಗ್ರಾಮ್ ಪಾಸಿಟಿವ್ ಅಂತಿದೆ, ಗ್ರಾಮ್ ನೆಗೆಟಿವ್ ಅಂತಿದೆ. ಗ್ರಾಮ್ ಪಾಸಿಟಿವ್ ಅಂದರೆ ದೇ ಗಿವ್ ಬ್ಲೂ ಕಲರ್ ವೆನ್ ದೇ ರಿಯಾಕ್ಟ್ ವಿತ್ ಎ ರಿಯೇಜೆಂಟ್, ಬಿಕಾಸ್ ದೇ ಹ್ಯಾವ್ ಎ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕನ್ ಅಂತ ಒಂದು ಪೆಪ್ಟೈಡ್ ಇರುತ್ತೆ ಸೆಲ್ ವಾಲಲ್ಲಿ, ಅದು ಗ್ರಾಮ್ ಪಾಸಿಟಿವ್. ಗ್ರಾಮ್ ನೆಗೆಟಿವ್, ನಾವು ಸಂಬಡಿ ವೇರ್ ಆಸ್ಕಿಂಗ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಯಾರು ಹೇಳಿದ್ರು, ಝಾಂಥೋಮೋನಸ್ ಏನೋ ಅಂದ್ರಲ್ಲ, ಸೂಡೋಮೋನಸ್ ಸ್ಪೀಶೀಸ್. ಸೊ ಪ್ಲೀಸ್ ಯು ಹ್ಯಾವ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಒನ್ ಥಿಂಗ್, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಂತ ತಗೊಂಡ್ರೆ PGPers, there are gram positive, gram positive. All that is listed as bacillus species. Bacillus, ಅದೆಲ್ಲ gram positive. Pseudomonas, Xanthomonas, ಆಮೇಲೆ ಇನ್ಯಾವುದಿದೆಯಪ್ಪ ಇದೆಲ್ಲ, ಸೊ ಅವೆಲ್ಲ gram negative. Now, bacteria. Agriculturally, gram negatives are more important, are more potent, have more positive effect than gram positives. But then why are we not using more gram positives? ಯಾಕೆ ಅಂದರೆ ಸೆಲ್ ವಾಲ್ ಇರಲ್ಲ, ಅದಕ್ಕೆ ಶೆಲ್ಫ್ ಲೈಫ್ ಈಸ್ ವೆರಿ ವೆರಿ ಬ್ಯಾಡ್. ಇಫ್ ಸಂಬಡಿ ಗಿವ್ಸ್ ಯು ಸೂಡೋಮೋನಸ್ ಬಾಟಲ್ ಹಿಯರ್, ಅವ್ರೇನಾದರೂ 1 ವರ್ಷ ಸ್ಟೆಬಿಲಿಟಿ ಅಂದರೆ ಇಟ್ ಈಸ್ ನೋ, ಯು ಕ್ಯಾನಾಟ್ ಗೆಟ್ 1 ವರ್ಷ ಸ್ಟೆಬಿಲಿಟಿ ಇರೋದು 2 months 3 months ಮ್ಯಾಕ್ಸಿಮಮ್, ದೇ ವಿಲ್ ಡೈ. ವೇರಸ್ ಬ್ಯಾಸಿಲಸ್ ಇದೆಯಲ್ಲ, ಸೆಲ್ ವಾಲ್, ಇದು ಹಾರ್ಡ್ ಸೆಲ್ ವಾಲ್ ಇರುತ್ತೆ, ಅದಕ್ಕೆ ಸ್ಪೋರ್ ಇರುತ್ತೆ, ದೇ ಮೇಕ್ ಎ ಸ್ಪೋರ್, ದೇ ಕ್ಯಾನ್ ಸ್ಟೇ ಫಾರ್ 2 years, ದೇ ಆರ್ ಡಾರ್ಮೆಂಟ್. But effect is gram negative is better effect than gram positives. So that we use, and you will ಮಾಡ್ತಾವೆ ಅಂದರೆ, see, they will have lot of effect on plant growth. Unique ಆಗಿ when bacteria is in the soil, iron ಎಳ್ಕೊಳ್ತವೆ, minerals ಎಳ್ಕೊಳ್ತವೆ, boron ಎಳ್ಕೊಳ್ತವೆ, and then phosphorus ನ mobilize ಮಾಡ್ತವೆ. They have very good ability to this. And second is they can thrive on the root ಇದ್ರೆ plant root ಮೇಲೆ they are growing. They take little carbon from the root, ಬೆಳೀತಾ ಹೋಗುತ್ತೆ, and they are attracting lot of nutrients and giving to the plant. ನೀನು ನನಗೆ ಕಾರ್ಬನ್ ಕೊಡು, ನಾನು ನಿಮಗೆ ಐರನ್ ಕೊಡ್ತೀನಿ. ಐರನ್ ಈಸ್ ನಾಟ್ ಈಸಿ ಟು ಅಪ್ಟೇಕ್ ಇನ್ ಪ್ಲಾಂಟ್ಸ್. ನೀವೇನಾದರೂ ಐರನ್ ಹಾಕಿದ್ರೆ ಫೆರಿಕ್ ಸಲ್ಫೇಟಲ್ಲಿ ಹಾಕ್ತೀರಾ, ಇದರಲ್ಲಿ ಹಾಕ್ತೀರಾ ಐರನ್ ಫರ್ಟಿಲೈಸರ್ಸ್. ಇಫ್ ಯು ಡೋಂಟ್ ಹ್ಯಾವ್ ಗುಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಹಾಕಿರೋ ಐರನ್ ಎಲ್ಲ ವೇಸ್ಟ್. And iron is very important for photosynthesis, fruit set, disease tolerance. Most of the micronutrients which you are applying in the field require good bacteria to transfer it to this thing. So ಅದಕ್ಕೆ if you apply good thing, complement it with good bacteria in the soil. Okay, foliar application ಅಲ್ಲಿ ಹಾಕಿದಾಗ ಡೈರೆಕ್ಟ್ ಆಗಿ leaves they can take it, but root ಗೆ ಹಾಕಿದಾಗ you need bacteria to bring it to the root, attach it to the root, and then the root will take it up. Root ಅಲ್ಲಿ ಡೈರೆಕ್ಟ್ ಆಗಿ ಮೈಕ್ರೋನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಅಬ್ಸರ್ವ್ ಮಾಡೋ ಕೆಪಾಸಿಟಿ ತುಂಬ ಕಮ್ಮಿ ಇರುತ್ತೆ. ಮೈಕ್ರೋಬ್ಸ್ ಇಸ್ ಗುಡ್. ದಟ್ ಇಸ್ ವಾಟ್ this plant growth promoting bacteria do. Okay, idu sheet ittu nanu amele heltini. So more or less this is the basics about all this biostimulant I have covered, but before I go to the next session is about identifying the right for Gherkin cucurbits, and then we can close it by 1:15 or something like that. Adagate. But I would like to, before I go to this one, some reflection about all the basics understanding of all these things, or any comments, anything. We'll give 5 minutes. We'd like to hear from you guys. Anybody like to talk on this one? Was that session useful? Did you get some good understanding of all the biostimulants, how they work, and what is the mode of action? Okay. Was it too heavy? ಜಾಸ್ತಿ ಆಯ್ತಾ? Okay.
It looks, it would have been little difficult for our field guys who are in the field and the basic knowledge. But anyhow, we understood, we have come to know. Simply we were talking, hey, ಇದು ಕೊಡಿ ಅದು biostimulants ಕೊಡಿ ಏನಂತ ಗೊತ್ತಿಲ್ಲ. So at least we are aware, awareness has come now.
Okay.
What do you say? We have got ಸ್ವಲ್ಪ ಆದರೂ ಅರ್ಥ ಆಯಿತು ಏನು ಬಯೋಸ್ಟಿಮ್ಯುಲೆಂಟು ಯಾಕೆ ಉಪಯೋಗಿಸ್ಬೇಕು ಅಂತ ಇಷ್ಟೊಳಗೆ. ಈಗ next chapter will be very clear, right?
Clear, okay.
That's my thought.
But was there anything missing from this, or what was it like? I don't know how to. Sir, I don't know. Sir, one kelsa madi. Please ask me questions, sir, because I may not have captured everything here. It is very good. Please, this is a very simplified, but please let me know what is that thing. I'm ready, because this is my field. I have worked on this for years together, and bacteria, you know. But okay. So plant growth promoter ali bacteria is the predominant. If you see all the total bacteria, 70% is actually bacterial strains. And bacteria dalli bacillus anta heladnalla adella nimage gram positive bacteria barutte. Every company makes gram positive bacteria only as their product. Yake andre stability.
Hello.
Yes. Yeah.
Sir, I am Nagaraj. I am agriculture graduate, a plant pathologist.
Yes.
Okay. My question is, you told that gram negative bacteria are good compared to gram positive bacteria. But my opinion and my experience, combination of both is good.
Yeah, I mean, I can
because gram negative cannot survive on a harsh environmental condition.
Yeah.
But in other harsh environmental condition, as you told, it forms spores, and whenever the congenial climate is available, and it starts work.
Yes.
The effect is more individual.
Yeah. No, what I meant was, if you take the capacity of both, these gram negative bacteria have more plant growth promoting ability in the plant. Bacillus species means they can activate more defense pathways. They can activate more flavonoid based pathway, phenyl propionate pathway anta madi. They can influence plant metabolism better than bacillus thing. But survivability is very, very low because of that.
Yeah.
But you are like, there are some now Bacillus subtilis species. They are also there.
Yeah.
In general, Pseudomonas, the effect Pseudomonas or Xanthomonas, Pseudomonas is there, and I can't recall. So a lot of
many species of Pseudomonas are
Pseudomonas
there.
are good species. Yeah. See, all Pseudomonas are not pathogen. They are also good bacteria. Pseudomonas, Xanthomonas, they have a very pronounced effect on the plant.
Yeah.
They are, as plant growth promoters, they have it.
And also they playing a role in suppression of disease.
Exactly. Whereas bacillus are what you call, they are soil bacteria.
Yeah.
Basically, they are soil bacteria, and people are using it to spray on the foliar, which may not be the right way to do it, but they are doing it. Okay. But if you ask any good pathologist, they say you should not spray bacillus on the foliar, because bacillus is soil bacteria, basically coming from soil. But for growth conditions, you cannot rely only on gram-negative bacteria.
Combination is
yes,
good.
combination is fine, but you should know what each bacteria is doing.
Yeah,
That's it. Yes.
main thing is
Good observation. Sir, ಈಗ ನಿಮಗೆ if you have some capability to make gram-negative bacteria, ಅದಕ್ಕಿಂತ ಒಳ್ಳೆದು ಇನ್ಯಾವುದೂ ಇಲ್ಲ. And every day you can spray on the foliar, you can add it through the drip irrigation. ಅದಾಗಲ್ಲ ಅದು. So then you go with Bacillus subtilis or Bacillus amyloliquefaciens. So Bacillus subtilis ಅಲ್ಲಿ growth promoting activity ತುಂಬಾ ಇರುತ್ತೆ. subtilis ಅಲ್ಲಿ ಓಕೆ amyloliquefaciens ಅಲ್ಲಿ pathogen control ability ತುಂಬಾ ಇರುತ್ತೆ. ಸೊ ಇಲ್ಲಿ 3 ತರ ಮೆಕ್ಯಾನಿಸಮ್ ಇರುತ್ತೆ ಸರ್. ವೆನ್ ಯು ಅಪ್ಲೈ ಎನಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಟು ದ ಸಾಯಿಲ್, ದೇ ಡೂ 3 ಥಿಂಗ್ಸ್. ಒನ್ ಈಸ್ ರೂಟ್ಸ್ಗೆಲ್ಲ ಬೈಂಡ್ ಆಗುತ್ತೆ. ಈ ಪೈಪಲ್ಲಿ ಪಾಚಿ ಕಟ್ಟಿರೋದು ನೋಡಿದ್ದೀರಾ, ಆ ಥರ ಇವು ಇದಾಗ್ಬಿಡುತ್ತೆ. ಆ್ಯಂಡ್ ದೇ ಆರ್ ಸಾಯಿಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಸೊ ದೇ ಆರ್ ಗ್ರೋಯಿಂಗ್. ಅಂದರೆ ಫಸ್ಟ್ ದಿವಸ ಒಂದು ರೂಟಲ್ಲಿ ಒಂದು ಫಿಲ್ಮ್ ಇರುತ್ತೆ, ಇನ್ನೊಂದು ಫಿಲ್ಮ್. ಸೊ ತಿಕ್ಕಾಗಿ ಹಿಂಗೊಂದು ರೂಟ್ ಇದ್ದರೆ ಅದಕ್ಕೆ ಬಯೋಫಿಲ್ಮ್ ಫಾರ್ಮ್ ಮಾಡಿಬಿಡುತ್ತೆ. ಈ ಬಯೋಫಿಲ್ಮ್ ಫಾರ್ಮ್ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ರೂಟ್ ಪ್ಯಾಥೋಜನ್ಸು ಏನು ಬರದಂಗೆ ದೇ ವಿಲ್ ಸ್ಟಾಪ್ ಇಟ್, ಬಿಕಾಸ್ ದೇ ಡೋಂಟ್ ಲೈಕ್ ಇಟ್. ಆ್ಯಂಡ್ ದೇ ಆರ್ ಆಲ್ಸೋ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಲಾಟ್ ಆಫ್ ಮೆಟಬಲೈಟ್ಸ್. ದೇ ಡೋಂಟ್ ಲೈಕ್ ಇಟ್. ಆಂಟಿಬಯೋಟಿಕ್ಸ್ ಬರ್ತಾ ಇರುತ್ತೆ. ಸೊ ಫಸ್ಟ್ ಲೈನ್ ಆಫ್ ಡಿಫೆನ್ಸ್ ಈಸ್ ಇನಿಬಿಷನ್. ದೀಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಗ್ರೋ ಟು ದ ರೂಟ್. ಯಾವ ಬೇರೆ ರೂಟ್ ಗ್ರಬ್ಸ್ ಅಥವಾ ಇದು ಇನ್ಸೆಕ್ಟ್ಸು ಅಥವಾ ನೋ ವಾಟ್ ರೂಟ್ ರಾಟ್ ಆಗೋ ಫಂಗಸ್ ಯಾವ ಹತ್ರ ಬರ್ದಂಗೆ ಆಗುತ್ತೆ. ಸೊ ದಟ್ಸ್ ವೈ ಯು ಶುಡ್ ಹ್ಯಾವ್ ದಿ PGPRs in this. First mechanism. Second mechanism is there is bad bacteria, bad fungus. Everybody wants to go to the root. ಎಲ್ಲರಿಗೂ ರೂಟ್ ಬೇಕು, ಯಾಕಂದರೆ ಅಲ್ಲಿ ಕಾರ್ಬನ್ ಇದೆ, ಇನ್ಫೆಕ್ಟ್ ಆಗ್ಬೋದು, ನೆಮೆಟೋಡ್ಸ್ ಇದೆ, ಎಲ್ಲ ಇದೆ. ಇವೇನು ಮಾಡ್ತವೆ, ದೇ ಕಾಂಪಿಟಿಷನ್, ದೇ ಎಕ್ಸ್ಕ್ಲೂಷನ್ ಅಂತ, ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸ್ಕ್ಲೂಷನ್ ಅಂತ. ಬಿಕಾಸ್ ದೇ ಹ್ಯಾವ್ ಆಲ್ರೆಡಿ ಅಕ್ವೈರ್ಡ್ ದ ರೂಟ್, ನಾಟ್ ಮೆನಿ ಅದರ್ ಥಿಂಗ್ಸ್ ಕ್ಯಾನ್ ಕಮ್ ಟು ದ ರೂಟ್. ಕಾಂಪಿಟೇಟಿವ್ಲಿ ದೇ ಎಕ್ಸ್ಕ್ಲೂಡ್. Third is, very important is, these bacteria trigger something called ISR, induced systemic response, which is and induced systemic response is triggered like bidid takshana specific bacteria they trigger the defense pathway. Defense alli enagutte, naturally phytoalexins, pathogen related proteins, even vitamin C, antioxidants, they are all produced. In that case, if downy mildew is infected for your Gherkin, they are much more protected. So, ಇವ್ರು ಸರ್ ಹೇಳಿದ್ರಲ್ಲ ಯಾವಾಗ ಹೋಗಿ ಸ್ಪ್ರೇ ಮಾಡಬೇಕು ಅಂತ. ಸರ್ ನಾನು ರೆಕಮೆಂಡ್ ಮಾಡೋದು, ಎವ್ರಿ 20 days ಆಲ್ಸೋ ಸ್ಪ್ರೇ ದಿಸ್ PGPRs ಆನ್ ಯರ್ ಫೋಲಿಯರ್. ಲೆಟ್ ದೆಮ್ ಫಾರ್ಮ್ ಎ ಫಿಲ್ಮ್. ದೇ ಆರ್ ನಾಟ್ ಎಕ್ಸ್ಪೆನ್ಸಿವ್. ಬಟ್ ಏನಾಗುತ್ತೆ ಅಂದರೆ, ನೀವು ಪೌಡ್ರಿ ಮಿಲ್ಡು ಹಾಕಿದಾಗ there is a set of plant which is sprayed with PGRs on the plant. Another set of plant which is not sprayed on PGR. ಎರಡಕ್ಕೂ ಒಂದೇ ಸಲ ಡೌನಿ ಮಿಲ್ಡ್ಯೂ ಅಟ್ಯಾಕ್ ಆಗುತ್ತೆ. ಸೊ ದಿಸ್ ಒನ್ ವಿಲ್ ಟೇಕ್ ಲಾಂಗರ್ ಟೈಮ್ ಟು ಶೋ ದ ಸಿಂಪ್ಟಮ್. ಅಲ್ಲಿ ಆಗಲೇ ಇನ್ಫೆಕ್ಟ್ ಆಗಿಬಿಡುತ್ತೆ. ಹೇಳಿ
sir. In Indian condition, the conditions like after August or November, the temperature goes high. So spraying on foliage is not so effective. Instead of spraying, feeding into the roots is better.
But for foliar disease, how will you control? There, okay, you can have a systemic resistance, something coming from this. But there are two things. One is coming from root is one response. Activating local response in the leaf is another thing.
But first thing, survival is important. When you say spray.
But you are priming the plant. Priming andre vaccination. It's just like vaccination the plant. If you spray every day, plants are ready. It's like you are ready with the guns and everything to control the disease. Whereas another set of plant where you don't have any PGPR sprayed, they will take a long time to mount the defense. ಅಷ್ಟೊತ್ತಿಗೆ ಡೌನಿ ಮಿಲ್ಡ್ಯೂ ತಿಂದಾಕ್ಬಿಡುತ್ತೆ ನಿಮ್ಮ ಪ್ಲಾಂಟ್ನ.
In cool climate, it's okay. In cool climate, this
so those exceptions are there. Okay, where efficacy. But I am saying. ಸರ್ ಇದೆ ಸರ್ ಟ್ರೇಡ್ ನೇಮ್. ಈಗ ನಮ್ಮಲ್ಲಿ ಅದು ಟಿಗ್ರೋ ಅಂತ ಒಂದಿತ್ತು ಪ್ರಾಡಕ್ಟ್. ಟೆಗ್ರೋ ಇಸ್ ಅ ಪ್ರಾಡಕ್ಟ್. ಆಮೇಲೆ ಬೇಯರ್ದೊಂದು ಪ್ರಾಡಕ್ಟ್, ವೆರಿ ಫೇಮಸ್ ಪ್ರಾಡಕ್ಟ್. ಯಾವುದದು ಏನೋ ಮರ್ತೋಯ್ತು. ಬೇಯರ್ ಹ್ಯಾಸ್ ವೆರಿ ಗುಡ್ ಬ್ಯಾಸಿಲಸ್ ಸಬ್ಟಿಲಿಸ್ ಒಂದು, ಇಂಡಿಯಾದಲ್ಲೂ ಇದೆ ಅದು ಪ್ರಾಡಕ್ಟು. ತುರಂಜೆನಿಸಿಸ್ ಈಸ್ ಬಯೋ ಕಂಟ್ರೋಲ್, ದಟ್ ಈಸ್ ನಾಟ್ ಹಿಯರ್. ಇದು ಬೇಯರ್, ಇದು ಕೊಡಿ, ಬೇಯರ್ ಬ್ಯಾಸಿಲಸ್ ಸಬ್ಟಿಲಿಸ್ ಅಂತ. ದಟ್ ಈಸ್ ಒನ್ ಆಫ್ ದ ಮೋಸ್ಟ್ ರಿಸರ್ಚ್ ಆ್ಯಂಡ್ ದೇ ಮೇಡ್ ಹ್ಯೂಜ್ ಮನಿ ಫ್ರಮ್ ದಟ್ ವರ್ಲ್ಡ್ ವೈಡ್. ಅದು ನಾನು ಹೆಸರು ನೋಡಿದ್ರೆ ಹೇಳ್ತೀನಿ ನಿಮಗೆ. ಬೇಯರ್ಸ್ ಅಲ್ಲ ಕೊಡಿ. ನಿಮಗೇನೋ ಗೊತ್ತಾ ಅದು ಕಮರ್ಷಿಯಲ್ ಟ್ರೇಡ್ ನೇಮ್ಸ್? ಓಕೆ, ಬೇಸಿಲಸ್ ಸಬ್ಟಿಲಿಸ್. ಸಬ್ಟಿಲಿಸ್ ಅದು. ಅದು ರ್ಯಾಪ್ಸೋಲ್ ಅಂತ. ಏನೇನಿದೆ ಅದರಲ್ಲಿ ಕಂಟೆಂಟ್? Serenade. Okay. Serenade is the most. Have you heard of anybody? Serenade? SERANADE. Serenade ಇದೆ. Europe ಅಲ್ಲಿ it is used ಇದು. And then other thing that they are asking. Yes, sir.
Yeah, sir. I have one basic question.
Yeah.
As we all know, the plant system has its own mechanism against any external stimulations, and also plants used to have the defense mechanism.
Yes.
By considering all the congenial condition to grow the plants, is it necessary to apply external biostimulants, maybe humic acid, maybe fulvic acid, maybe seaweed extra, any kind of the external application? Is it necessary to the plants under the congenial condition, especially where abiotic stresses are not there? If so, even after application, what may be the % increase in the yield? Like we are all here to see the output with the given condition. For example, if there is a normal, for example, 10 tons, even after application of these all growth promoters or any given stimulants, what we are going to expect the in the yield?
Did you come late to this session?
A little, I came little late.
Because the first 2 slides were all about. So there is stress and there is fertilizer use efficiency. If you have fantastic condition, all good irrigation, good sunlight, everything, no need of applying any biostimulant. Why will you apply? But
Yes,
have you ever seen that condition in India, anywhere, any year?
Sir, as a Gherkin, we are growing mainly in the 3 seasons. Especially in Karnataka, we are growing in the pre-Kharif season, like April, May, we are going to sow. June, July, we are receiving the green arrival window.
Yes,
Sowing window will be April, May. Green's arrival window will be during the June, July. Almost climate is very congenial. Like if the almost 90% I can say with respect to temperature, relative humidity, and all congenial only. With that intention, I asked you that
No.
question. Yeah.
Your definition of ideal climatic condition should be measured through yield. What
Yes.
is the okay, now I'm repeating the same thing again. Now there are Rizwan guys also, right? Rizwan companies. So what is the yield potential, sir, of your maximum best yield anywhere, same variety? Will you get that? If you don't get the 25 to 30, then it is something is wrong.
No.
Then your conditions are not ideal. I'm
Karthik,
saying.
is 25 tons
Okay,
irrespective of
forget
the grade you are
25
telling?
So 25 times 5, at least for India.
Yeah.
That is the maximum yield you can get.
It means with respect to grade, particular grade you are telling, or irrespective? Yes.
That is what I'm saying. That is why we spray all the biostimulants applies that you cannot control the stress. You don't know what stress is coming today, tomorrow what is not coming, but you are preparing the plant. We call the word priming. Priming is like before the infection, you are ready. Now, for example, if somebody is coming to attack you, Oh, he's come. Now you are going and searching the gun and coming. But here we are talking about is you are already standing here with the gun.
Okay, sir. Thank you.
That is the difference. Understand? Very basic way.
Sure, sir.
By applying all these things, you are just waiting here with the gun, and then you just shoot. The normal plant, oh, somebody has come, you don't know, you're asking, Lakshmi, where is this? Can open the door,
Yes.
go and put the bullets, everything. That you will lose it. Powdery mildew will overtake you completely. Insect will overtake you. The nutrients are gone. So prepare your plants, prime your plants with all the good biostimulants, good selfie extracts, good bacteria. That word you remember, then you will understand why people should invest in all these things. Otherwise, you are right, you don't have to waste money.
Yes.
But prepare, it's good. That's a good question, by the way. No, no. Okay, sir, coming back. So bacteria is there. So those are the two good products, but I will give you a list of other products. So Tegro is one more good product from this one. And I know 4 names from the company I am consulting for UK, but I should not tell this. It will be a conflict of interest. So I will, once we, I'll tell those names. But I'll give you that list. Lakshmi, please remind me later. We'll publish all those PGPR products. And we can also test if some people have used in Gherkins already, and also how they are, I'll do that one for you. P-solubilizing fungus, sir. Anything which is Bacillus megaterium. Have you heard? This is called Bacillus megaterium. Universities are providing a lot of free P-solubilizers. Government schemes are also giving, but I don't know quality and all those things. Yes. So that means, see, phosphorus is very complicated. Phosphorus, the moment you apply to the soil, it gets fixed. It's a very, very reactive element. And second is, that is inorganic phosphorus. And there is organic phosphorus, which is in the form of phytate. Phytate. Phytate is, it's a very complex molecule. Seeds, there is a cyclic chain, there is phosphorus, there is minerals like calcium, magnesium. ಇವೆಲ್ಲ ಬೌಂಡ್ ಆಗಿ ಒಂದು ಫೈಟೇಟ್ ಮಾಲಿಕ್ಯೂಲ್ ಆಗುತ್ತೆ. Now the phytate is there, but plants don't need phytate. They just need phosphorus. ಕಟ್ ಮಾಡಬೇಕು ಫಾಸ್ಫರಸ್ ಬೇಕಷ್ಟೇ ಅವ್ರಿಗೆ. ಇಲ್ಲ ಈ ಕಡೆ ಕಟ್ ಮಾಡಿ ಕ್ಯಾಲ್ಸಿಯಂ ಬೇಕು. This one is done by megatherium. So the megatherium has the necessary enzymes. They has the organic acid to release the phosphorus. So ಇವಾಗ ಸೀಡ್ಸ್ ಪುಡಿ ಪುಡಿ ಮಾಡಿ ಹಾಕ್ತಾರಲ್ಲ ಹೋಗುತ್ತೆ. ಇವರು ಈ ಪೋಲ್ಟ್ರಿ ಫೀಡಲ್ಲಿ ಸೀಡ್ಸ್ ತಿನ್ನುತ್ತೆ ಚಿಕನ್ಸ್. ಸೀಡಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಫೈಟೇಟ್ ಇರುತ್ತೆ. ಅದಕ್ಕೇನು ಮಾಡ್ತಾರೆ, ಅದ್ರ ಜೊತೆಗೆ ಫೈಟೇಸ್ ಅಂತ ಒಂದು ಎಂಜೈಮ್ ಕೊಡ್ತಾರೆ. ದಟ್ ಬ್ರೇಕ್ಸ್ ದಟ್. ಸೊ ಚಿಕನ್ ವಿಲ್ ಗೆಟ್ ದ ಫಾಸ್ಫರಸ್, ನಾಟ್ ದ ಫೈಟೇಟ್. ಸೇಮ್ ಥಿಂಗ್ ಇನ್ ದ ಸಾಯಿಲ್, ದಿಸ್ ರಿಲೀಸ್ ಆಫ್ ಫಾಸ್ಫರಸ್ ಈಸ್ ಡನ್ ಬೈ ಬ್ಯಾಸಿಲಸ್ ಮೆಗಟೇರಿಯಂ. ಸರ್ ಮೆಗಟೇರಿಯಂ ತುಂಬ ಪ್ರಾಡಕ್ಟ್ಸ್ ಇದೆ ಸರ್. ಟ್ರೇಡ್ ನೇಮ್ ಅಂದರೆ ಸ್ಪೆಸಿಫಿಕ್ ಕಂಪ್ನಿಗೆ ಹೋಗ್ಬಿಡ್ತೀರಾ ಅವಾಗ ನೀವು, ಈ ಕಂಪ್ನಿ ಇದೆ ಟ್ರೇಡ್ ನೇಮ್ ಅಂತ. ಮೆಗಟೇರಿಯಂ ಯಾವುದಿದೆಯಪ್ಪ ನಮ್ಮದು ಕಂಪ್ನಿದು. ಸೊ IHR ಹ್ಯಾಸ್ ಲಾಟ್ ಆಫ್ ಮೆಗಟೇರಿಯಂ ಕಂಪ್ನೀಸ್. ಅರ್ಕಾ ಬೇಸ್ಡ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಇದೆ ನೋಡಿ. ಐ ಥಿಂಕ್ ದೇ ಆರ್ ಎಸ್. ಅದಕ್ಕೆ ಬೇಸ್ಗೆ ಹಾಕಬೇಕು, ಫಾಸ್ಫರಸ್. No, it is not costly, but it is better. I have seen their megatherium is better because it is easy to make. Again, stable. Any product containing Bacillus megatherium, please use it in Gherkins. Megatherium, MEGETERIUM. So megatherium is good. The nitrogen assimilation is no problem. Plants can take nitrogen very easily. Phosphorus, potassium, all zinc, they are not easy to take. Zinc, iron, they need lot of support from good bacteria. pH change irbeku, they have receptors idave. So PGPRs can help. Now that is P-solubilizing bacteria. Third one is mycorrhiza. Have anybody heard mycorrhiza? Mycorrhiza, is it bacteria or fungus? It is fungus actually. It is fungus. if megatherium solubilizes phosphorus, ಅಂದರೆ ಫೈಟೇಟಿಂದ ರಿಲೀಸ್ ಮಾಡುತ್ತೆ, what mycorrhiza does is, it takes the phosphorus and gives it to the plant. ಪ್ಲಾಂಟ್ ಏನು ಕೊಡುತ್ತೆ? ಕಾರ್ಬನ್ ಕೊಡುತ್ತೆ, ಅದಕ್ಕೆ ಶುಗರ್ ಕೊಡುತ್ತೆ. ಆ್ಯಂಡ್ ಮೈಕ್ರೋರೈಸಾ ಆಲ್ವೇಸ್ ಫಾರ್ಮ್ಸ್ ದಿಸ್ ನಾಡ್ಯೂಲ್ಸ್ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ. ನಾಡ್ಯೂಲ್ಸ್ ಅಲ್ಲ, ಇದು ಏನಂತಾರೆ ಅದಕ್ಕೆ, ನಾಡ್ಯೂಲ್ಸ್, ಇದು ಆರ್ಬಸ್ಕ್ಯೂಲ್ಸ್. ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆರ್ಬಸ್ಕ್ಯೂಲ್ಸ್. ಅಂದರೆ ಒಳಗಡೆ ಒಂದು ಮಾಡಿ ಥೌಸಂಡ್ಸ್ ಆಫ್ ಮೈಕ್ರೋರೈಸಾ ವಿಲ್ ಬಿ ದೇರ್. ದೇ ಆರ್ ಸಿಕ್ವೆಸ್ಟ್ರಿಂಗ್ ಶುಗರ್. ಓಕೆ, ಆ್ಯಂಡ್ ದೆನ್ ಇವಕ್ಕೆ ಫಾಸ್ಫರಸ್ ಕೊಡುತ್ತೆ. ಪ್ಲಾಂಟಿಂದ ಶುಗರ್ ತಗೊಳ್ತವೆ, ಪ್ಲಾಂಟಿಗೆ ಫಾಸ್ಫರಸ್ ಕೊಡುತ್ತೆ. ಎಸ್, ಭಾಗ್ಯರಾಜ್ ಇಸ್ ದ ಪಯನಿಯರ್ ಅವರು. ಎಕ್ಸಾಕ್ಟ್ಲಿ, ಭಾಗ್ಯರಾಜ್. ಸೊ ಎಲ್ಲ ಕಾಂಬಿನೇಷನ್. ಸೊ ಯು ಕ್ಯಾನ್ ಹ್ಯಾವ್ ಎ ಪಿ ಸಾಲ್ಯುಬಲೈಸರ್ ವಿಚ್ ವಿಲ್ ವರ್ಕ್ ಔಟ್ಸೈಡ್ ದ ಪ್ಲಾಂಟ್. ಮೈಕ್ರೋರೈಸಾ ವಿಲ್ ವರ್ಕ್ ಆನ್ ದ ಪ್ಲಾಂಟ್. ಅದು ಸಿಗುತ್ತೆ ಸರ್, ಅವ್ರದ್ದು ಇದೆ. ಗ್ರಾಮ್ ಇಸ್ ವೆಸಿಕ್ಯುಲರ್ ಆರ್ಬಸ್ಕ್ಯುಲರ್ ಮೈಕ್ರೋರೈಸಾ. ದಟ್ ಈಸ್ ಬಿಕಾಸ್ ಇಟ್ ಫಾರ್ಮ್ಸ್ ವೆಸಿಕ್ಯೂಲ್ಸ್ ಆ್ಯಂಡ್ ಒಳಗಡೆ ಆರ್ಬಸ್ಕ್ಯೂಲ್ಸ್ ಅಂತಾರೆ, ಅದು ಮಾಡುತ್ತೆ. ಆ್ಯಂಡ್ ದಟ್ ಈಸ್ ವೇರ್ ದ ಸೈಟ್ ಆಫ್ ಫಾಸ್ಫರಸ್. ಫಾಸ್ಫರಸ್ ಇಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಫಾಸ್ಫರಸ್ ಇಲ್ಲಾಂದರೆ ನಿಮಗೆ DNA ಇರಲ್ಲ ನಿಮಗೆ. And you PGPRs, mycorrhizas, ಇದು ಜಾಸ್ತಿ ಆಯಿತು, ಅದು ಕಮ್ಮಿ ಆಯಿತು. ದೇ ಆರ್ ನ್ಯಾಚುರಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ದೇ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ ಸಾಯಿಲ್. ಯು ಕ್ಯಾನ್ ಆ್ಯಡ್ ಎನಿ ಟೈಮ್, ಎಷ್ಟಾದರೂ ಹಾಕಿ, ಏನಾದರೂ ಹಾಕಿ, ಅದ್ರ ಜೊತೆಗೆ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಹಾಕಿ, ಏನೂ ಆಗಲ್ಲ. ಸಾಯಿಲ್ ಈಸ್ ಎ ವೆರಿ ವೆರಿ ಏನಂತಾರೆ, ಪಾಪದ ಜೀವ ಅದು. ಏನಾದರೂ ಹಾಕಿ ಸರ್, ಅದಕ್ಕೆ ತಡ್ಕೊಳ್ಳುತ್ತೆ. ಯು ಪುಟ್ ಸೋ ಮಚ್ ಆಫ್ ಥಿಂಗ್. ರೂಟ್ ಮಾಡಬೇಕು, ನೀರು ಹಿಡಿಬೇಕು, ಮೈಕ್ರೋಬ್ಸ್ ತಗೋಬೇಕು, ಪೆಸ್ಟಿಸೈಡ್ ಹಾಕಬೇಕು, ಹೆವಿ ಮೆಟೀರಿಯಲ್ಸ್ ಇಟ್ಕೋಬೇಕು. ಬಟ್ ಇಟ್ಸ್ ಎ ವೆರಿ ಗುಡ್ ಎಕೋಸಿಸ್ಟಮ್. People, I think soil is so important. They'll do a lot of things, and it is such a dynamic thing. It will dilute things, it will concentrate. So, ನಾನು ಲಾಸ್ಟ್ ಸ್ಲೈಡಲ್ಲಿ ತೋರಿಸ್ತೀನಿ ಮೈ ರೆಕಮೆಂಡೇಶನ್. ಹೌ ಯು ಶುಡ್, ವಾಟ್ ಈಸ್ ಮಸ್ಟ್ ಟು ಹ್ಯಾವ್, ವಾಟ್ ಈಸ್ ಗುಡ್ ಟು ಹ್ಯಾವ್, ವಾಟ್ ಯು ಕ್ಯಾನ್ ಹ್ಯಾವ್. ಆ ಥರ ಒಂದು ಸ್ಲೈಡ್ ಇದೆ ನಂದು. ನೆಕ್ಸ್ಟ್ ಇದು ಮುಗಿಸ್ತೀನಿ, ಇದರಲ್ಲೇ ತೋರಿಸ್ತೀನಿ. ಸೊ ಇದು ಸರ್ ಬೇಸಿಕ್ಸ್ ಆಫ್ PGPRs ಇದರಲ್ಲಿ. ಓಕೆ, ವಾಟ್ ಐ ಡಿಡ್ ನಾಟ್ ಡೂ ಗುಡ್ ಜಾಬ್ ಇಸ್ ಐ ಡಿಡ್ ನಾಟ್ ಗಿವ್ ಯು ಟ್ರೇಡ್ ನೇಮ್ಸ್ ಆನ್ ಆಲ್ ದೀಸ್ ಥಿಂಗ್ಸ್. ಯಾಕಂದರೆ ನನಗೆ ಸ್ವಲ್ಪ ತಲೆ ಓಡ್ತಾ ಇಲ್ಲ, ಇದು ಆಯ್ತದೆ. ಬಟ್ ಐ ವಿಲ್ ಗಿವ್ ಯು ಟು. ಅದೇನಾಗುತ್ತೆ ಅಂದರೆ ಸರ್, ನಾನು ಒಂದು ಕಂಪ್ನಿ ರೆಕಮೆಂಡ್ ಆದಂಗೆ ಆಗುತ್ತೆ, ಅದಕ್ಕೆ ನಾನು ಐ ಆಮ್ ಲಿಟಲ್ ಕೇರ್ಫುಲ್ ಇನ್ ದಿಸ್. ಬಟ್ ವಿ ವಿಲ್ ಗಿವ್ ಬೇಯರ್ಸ್ ಸೆರೆನೇಡ್ ಈಸ್ ಒನ್ ಆಫ್ ದ ಕ್ಲಾಸಿಕ್ ಸ್ಟಡಿ. ಸೆರೆನೇಡ್ ಒಂದೇ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸಬ್ಟಿಲಿಸ್ ಹಾಕಿರೋದು ಅದರಲ್ಲಿ, ಆ್ಯಂಡ್ ಇಟ್ ಈಸ್ ಬೀನ್ ಇನ್ ದ ಮಾರ್ಕೆಟ್ ಫಾರ್ 20 years. ಅದು ವೆಜಿಟೇಬಲ್ಸು, ಫ್ರೂಟ್ ಆ್ಯಂಡ್ ವೆಜಿಟೇಬಲ್ಸಲ್ಲಿ ದೇ ಯೂಸ್ ಇಟ್. ರೂಟ್ಗೆ ಅಪ್ಲೈ ಮಾಡ್ತಾರೆ, ಫೋಲಿಯರ್ಗೂ ಹಾಕ್ತಾರೆ. ಇಟ್ ಈಸ್ ಸಚ್ ಎ ಗುಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಸೊ ಯು ಕ್ಯಾನ್ ಟ್ರೈ ಸೆರೆನೇಡ್. ಬಟ್ ಎಲ್ಲೆಲ್ಲಿ ಸಿಗುತ್ತೆ, ಒಬ್ಬ ಡಿಸ್ಟ್ರಿಬ್ಯೂಟರ್ ಇಟ್ಕೊಂಡಿರ್ತಾರೆ, ಕೆಲವರು ಇಟ್ಕೊಂಡಿರಲ್ಲ. ಯು ಆಸ್ ಅಸೋಸಿಯೇಷನ್ ಶುಡ್ ಕಾಂಟ್ಯಾಕ್ಟ್ ದ ಕಂಪನಿ ಸರ್, ಕಂಪನಿ. ಓಕೆ, ಆರ್ ಅಂಡ್ ಮೇ ಬಿ ಐ ಕ್ಯಾನ್ ಹೆಲ್ಪ್ ಯು ಲಿಂಕ್ ವಿತ್ ದಿಸ್ ಕಂಪ್ನಿ ಗೈಸ್. ಡೈರೆಕ್ಟ್ ಕಂಪ್ನಿಯಿಂದ ಬೈ ಮಾಡಿ ನಿಮಗೆ ಇದಿದ್ರೆ ಆ ಥರ ಮಾಡಿ, ಬೆಸ್ಟ್. ದೆನ್ ಆಸ್ಕ್ Serenade. Maybe now they have also developed a good and company should have a strong research background to develop PGRs. Not everybody can do this. Okay, sir. So that's fine. Okay. Okay. So we'll go to the next session. Though this is a little application part, maybe this is, I want to give you what are the 3 criteria. Sir, ಕೇಳ್ತಾ ಇದ್ರಲ್ಲ ಬೆಳಗ್ಗೆ ಪ್ರಶ್ನೆಗಳು. So we will answer that, sir. ನೋಡೋಣ ಎಷ್ಟಾಗುತ್ತೆ ಅಷ್ಟು ಆನ್ಸರ್ ಮಾಡೋಣ. Is how do you identify the right biostimulant for Gherkin or cucurbits? And I have done some research on the papers, publications available. And whatever I am saying is not saying my word, but there is some research. People have done that research. I'll share all those papers with you. You can, ನಿಮ್ಮ ಟೈಮ್ ಇದ್ದಾಗ ಇದನ್ನು you can go for it. So it's basically about key learnings, research institutes, what are the best practices. And we'll close for lunch here. Now, I like this saying, when they say, what is the criteria to choosing a biostimulant? You should not choose a biostimulant thinking that what is a biostimulant antalla, but what is a biostimulant, what biostimulant can do to my plant? Andre, or you can say, what can a biostimulant do to my crop, but not what they are? Hey, yara humic acid hakbudu. No, that's not the criteria. Wrong. First, humic acid hakri yen agutte nanige? You should be knowing. Just, oh, somebody used seaweed extract. I will use seaweed. Somebody used protein hydrolyzed. But know what it is. Nindu requirement is very different from the other person's requirement. Do you think that the biostimulant you are choosing will do the job? Don't waste money. Summune maadibittu effort mari labor alla. But you should be knowing and your criteria. Now, if you see, what is the biostimulant that can do? There are so many things. I have highlighted the key 6 points. And you may be interested in one or two or many or only one. So, sir, I am saying is, be sharp. Nanige uniform seed germination bakery aste. Nanige expectation jasti illa. There is a biostimulant for that. Then if you say, okay, I want good shoot and lateral branches so that I can get more fruit, there is also a biostimulant for that. There is a finding. They will say, and how much dose should you apply ಮಾಡಬೇಕು ಇದು ನಿನಗೆ ಇಲ್ಲ I want flower set flower drop ಆಗಬಾರ್ದು good flower set it is there. ಕೆಲವರು ನನಗೆ overall yield ಜಾಸ್ತಿ ಬರಬೇಕು, that is one objective. Somebody product quality, I'll show you one example how successfully they have done. Next slide ಅಲ್ಲಿ ತೋರಿಸ್ತೀನಿ. ಪ್ರಾಡಕ್ಟ್ ಕ್ವಾಲಿಟಿ ಅಷ್ಟೇ ನನಗೇನೆಲ್ಲ yield ಬರ್ತಾ ಬರುತ್ತೆ, ಬಟ್ ನನಗೆ ಸ್ವಲ್ಪ ಕಲರ್ ಚೆನ್ನಾಗಿ ಬಂದರೆ ಒಳ್ಳೆ ರೇಟ್ ಬರುತ್ತೆ. They know that. We will recommend the good biostimulant for them. Or they will say sometimes nutrient efficiency. ಎಷ್ಟು ಗೊಬ್ಬರ ಹಾಕಿದ್ರೂ ಏನು ಪ್ರಯೋಜನ ಆಗ್ತಾ ಇಲ್ಲ, ನಾನು ಡಬಲ್ ಡೋಸ್ ಮಾಡಿದ್ದೀನಿ ಆದರೂ yield ಇಷ್ಟೇ ಬರ್ತಾ ಇದೆ. Then you have a solution for that one. Maybe you have to change your root architecture, use more PGRs, more humic acid. And then crop resistant to abiotic stress. ಈ ಯಾವಾಗಲೂ ಮಳೆ ಬರುತ್ತೆ ಹೋಗುತ್ತೆ ಇದಾಗುತ್ತೆ, ಈ ಸ್ಟ್ರೆಸ್ ಜಾಸ್ತಿ ಇದು ನಮ್ಮ ಹಿಸ್ಟಾರಿಕಲಿ ಇದು ಅಂದರೆ, then you have a biostimulant for that. You can choose a for soil attack, soil property. Soil ಸರಿ ಇಲ್ಲ ರೀ, ಅದು ಮಳೆ ಬಂದರೆ ಇದಾಗುತ್ತೆ, ಅಥವಾ ಜನರಲಿ ದಿಸ್ ಇಸ್ ನಾಟ್ ಎ ರಿಚ್ ಸಾಯಿಲ್ ಅಂದಾಗ there is another thing. So the point I'm trying to make is, you should be clear to know what you want to get out of or first understand what is your problem. The solution will come. Blindly humic acid ಹಾಕ್ತಾ ಇರೋ, ಮುಂದೆ ಇದು ಹಾಕು, fulvic acid ಹಾಕು, ಇದು ಹಾಕು, ಅದು, that is not correct, wrong, without addressing the key point. Any comments on this? Okay. Oh, online first question, Papa.
Can stimulant help recover crop from pesticide or herbicide shock? Some extract have strong odor while others don't. Doesn't it affect the quality? Is it better to use under downy mildew condition? Can it be mixed with boron too?
Very long question.
You can address for, can stimulants help recover the crop from the pesticide or herbicide shock?
Herbicide shock. Possible. It is possible. And though I don't have any data to support that, any shock after that, because they will mobilize nutrients, they will mobilize water, they will they will influence phytohormones, new shoots will develop. So when you apply any biostimulant, good biostimulant, after a shock, they should trigger the crop. Yes, it is possible that they can regrow. They can regrow and then repair themselves after the shock.
And the second part of this question is, some extract have strong odor while other don't. Doesn't it affect the quality?
Odor?
Odor.
If you have some strong odor or some means biologically active anta meaning adu. So I don't know, it could be good, but strong odor will also mean that it is fermented, it is contaminated. Usually good biostimulants have some fruit, nice flavors. but very strong metabolites can also have some strong odor. So I don't have the full answer for this one. I don't know completely whether the bad odor means bad quality, good odor. It's a very tricky question. I
No,
don't
it's
know.
not. It's not on the quality of the biostimulants. It's on the product what we grow. The bad odor, if I spray on the product, will it not affect the product? My output.
No, what I am saying is some metabolites have bad odor only, and doesn't mean that it is a bad product.
Okay.
Is what I am trying to say. So we have to check case by case. For example, humic acid based products, sometimes if they are stored for a longer, it is too much concentrated, can also have a little odor, but doesn't mean that it is bad. It is still doing the job.
No. So will it not affect our product, like my Gherkins?
No, no, I think these are not like what do you call strong odor. They will get metabolized. They will go away.
Yeah.
So don't worry about it.
Okay.
So it will go away. There is no such odor. Biological odors don't stay for long. They will get metabolized. That's fine. Yeah.
And the third part of the question is it better to use under downy mildew conditions? Can it be mixed with boron also?
Yes, I think it's fine. I have some examples. They don't really, see, pesticide pathway, pesticide is direct effect. They go inside chemical effect, they not trigger any pathway. Biostimulant, they will trigger pathways. Absolutely fine, you can combine pesticides and biostimulants and you can spray because they don't interfere so much. That is chemical, these are biologicals. So they have very different mode of action. You can do it.
Okay, I think, thank you.
Good. Okay, so this is my first criteria. So please know what you want to gain from your biostimulant. Just don't blindly go for a biostimulant. After that, things will be easy. Now, I would like to give you some example from chili farmers. Chile, you know this country, right? Chile, South American country here. Beautiful country it is. They grow cherries. The cherries from Chile have huge demand in China. In China, New Year is there. Chinese New Year happens somewhere, January, February. There will be year of pig, year of donkey, all this. And they use a lot of cherries in China. 90% or maybe, I don't know, 70% of the cherries, they prefer only from Chile. Why? Europe also grows chili. Our regionally, Brazil, but why they prefer cherries from Chile is because the answer is because of one biostimulant. Because of the biostimulant. What happens is, all through the year, chili farmers, they take the chili, they market it in the local market. There is a cap, they cannot get more price. Esto ಒಂದು $5 per kg ಅಷ್ಟೇ ಸಿಗ್ತಾ ಇರುತ್ತೆ. But Chinese, they will pay up to $10, provided they get that beautiful color. This one. Now, coming back to my first criteria, is you should be knowing what the biostimulant can do for my crop. Chili farmers know exactly. If I use this biostimulant, it will change the color. It will make the shelf life of the fruit a little bit stronger. Nice flavors. 3 or 4 things. And they spray, and maybe they are spending $0.5 per hectare. They are getting $3 more for this one. So that is what I meant exactly. That they don't want to do too many things. Only improve the quality of the cherry, color, quality, this thing. Now, can you please list what is some 3 or 3 key economical criteria in Gherkins that will fetch more value when you are selling regularly? Or is there anything like that, that if you have any value addition, it will give you more value in Gherkins or texture? Is it like a rough texture or smooth textures or something? And you open it, it's a little. Okay. So you will get more money for that? then we can help you. We can help you with all the experts to choose the best biostimulant and use only for that and improve your and apply it at the right time. Yes. Why are you wasting time in doing a lot of protein hydrolysate at unnecessary time doing it? Maybe I can tell you, if you want to get a crispy or very tasty thing, maybe you have basal dose of something in the root. When the right time is right, you spray this. And this is clinically known to improve the phenyl propanide pathway, improves nice flavors, colors, and you can get a gherkin which is whatever you want. That is what. Yeah, you had a question, yeah, please. Loud, huh?
Apart from that crispiness, in gherkin, the important one is the grades.
Grades.
Grade means fruit size.
Ah, that's a good one. That's
So,
pure fruit size.
The uniformity of the fruits size in terms of size.
Okay.
So, any
sir,
biostim-
ನೀವು ಅಂದ್ರೆ ನಮಗೆ economically important gherkin traits which can trigger more economical value. If you give me that one, I will work with ಕೇಳಿಸ್ತಿಲ್ವಾ?
I'm hearing.
ಇಲ್ಲ ಅಷ್ಟೇ ಐತೆ ಅವಾಗಿಂದ.
Yeah, audible, sir. Audible. It's audible. It's audible.
ಈಗ
Audible.
now it's okay. Okay, then you're right. See, some said crispiness and and then you say fruit size. And I think the best way is to identify these 2 or 3 traits. ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಮಾಡಿ. And then I will, with my experts, whoever is, I will tell what is the best biostimulant combination. Should we add PGRs or should we combine fulvic acid with protein hydrolysate? What does the research say? Research, ಹೌದು ಇದೆಲ್ಲ ರಿಸರ್ಚ್ ಅಲ್ಲಿ ಬ್ಯಾಕಪ್ ಇದೆ. We'll do some experiment also, sir. Yes, sir. ಈಗ ಇಷ್ಟೊತ್ತು ಮಾತಾಡಿದ್ದೆಲ್ಲ ಜನರಲ್ ಇತ್ತು. ಈಗ ನಾವು we are getting into deep dive ಮಾಡ್ತಾ ಇದ್ದೀವಿ. So if you tell us that I am only looking for this good quality, ಇದು ಅಂತಂದ್ರೆ there is lot of research, lot of publication, there will be some good combination which has been tried in some, you can try it. And if you see consistently, I think it will help all the gherkin growers in this way. ಅದು ನಾವು ಮಾಡ್ಕೊಡ್ಬೋದು. So again, we have addressed the first point, just like the cherry growers in Chile. ಅವ್ರಿಗೆ ಕಲರ್ ಬೇಕು, ಕಲರ್ಗೆ ಎಷ್ಟು ಬೇಕೋ ಅಷ್ಟೇ ಮಾಡ್ತಾರೆ, ಇನ್ನೇನು ಮಾಡಲ್ಲ ಜಾಸ್ತಿ. ಆ್ಯಂಡ್ ದೇ ಆರ್ ಫೆಚಿಂಗ್ ಗುಡ್ ಮನಿ. ಈಗ ನೋಡಿ ಬ್ಲೈಂಡಾಗಿ ಹುಲ್ವಿಕ್ ಆ್ಯಸಿಡು, ಯೂಮಿಕ್ ಆ್ಯಸಿಡು, ಅದು ಎಲ್ಲ ಹಾಕಿದರೆ ಅವನ ಪ್ರೈಸ್ ಎಷ್ಟು ಜಾಸ್ತಿ ಆಗುತ್ತೆ? ಸೊ ಕಾಸ್ಟ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. ಎನಿವೇ ಫಾರ್ ಹಿಮ್ ಈಸ್ ವ್ಯಾಲ್ಯೂ ಅಡಿಷನ್ ಎಲ್ಲ ಕರೆಕ್ಟಾಗಿದೆ. ಈಲ್ಡ್ ಸ್ವಲ್ಪ ಕಲರ್ ಫ್ಲೇವರ್ ಚೇಂಜ್ ಮಾಡಿಬಿಟ್ರೆ ನಾನು ಅದಕ್ಕೆ ಕರೆಕ್ಟಾಗಿ ಹುಡುಕಿದ್ದಾರೆ. ದೇ ವೆಂಟ್ ಟು ಅಕಾಡೆಮಿಯಾ, ದೇ ಫೌಂಡ್ ದ ಬೆಸ್ಟ್ ಇದು, and they spray every year and they make money. Chinese are paying more money for that. I think for you Gherkin also, you should take some inspiration from like that and focus only on that biostimulant to do this. Okay. So this is one case study. Nodi idalli. ಈಗ second ಬಂದು my second criteria. Any questions on first criteria? That I think that was clear. The second criteria is applying a biostimulant without knowing the...
Can you take one more question?
Yeah, sure.
This question is regarding an earlier topic on humic acid. Can we mix humic acid with calcium fertilizers or sulfates?
Sulfates, I don't know, but calcium is absolutely.
Calcium can be mixed?
Yes, no problem. Because anyway, humates will only help to bind and release to the plant. They will not sequester any nutrient permanently. So you can combine nutrients and humic acid, no problem. Which one? See, element, it is what we know is any element that binds to humic acid is not a permanent bind. It is like good, But if there is any data saying that calcium and humic acid, if calcium binds to humic acid, it will never release, then I am wrong. I am wrong. But sir, this is a organic material. Humic acid is organic material. Calcium is inorganic material. Both are taken up in the plant in different ways. And I don't know, you may challenge me, I may be wrong also, but I don't see anywhere where they both are incompatible. Because the organics are different, inorganics are different. They will, how? I don't know, maybe I'm thinking from a broad perspective, but if anybody have answers, why it's not incompatible, I want to know from this one. Yeah, tell me. Sulfate ಗೊತ್ತಿಲ್ಲ sir, ನಾನು calcium ಹೇಳಿದೆ. Okay, then here is a live example. It should be, ನನ್ನ ಪ್ರಕಾರನು there should be. This guy. Sir, we can case by case ನೋಡಬೇಕು, but I am with Vinod sir here, but I don't see any compatible. But ಇಲ್ಲ, it's good to deliberate, ಯಾಕಂದ್ರೆ So the answer is
they can use.
It should be okay. It's not a big.
And one more continuous to that question: does potassium humate exhibit the same property as humic acid?
Good question. No, potassium humate are more reactive, more potent than this. This is like basic. Humic acid is basic. Here you are making it more reactive by adding a potassium group to that one. So yes, in theory, potassium humates are more specific, more what you call reactive than generic humates. That's why they made potassium humates. And second thing is stability. Everything is improved when you have potassium humates. It is like that. Yes, it is fair to assume that they are little better than the basic humic acid. So this is from the chili experiments, and I think Gherkin can also do something like that, where to identify a problem and then do only most that is necessary for them. If you are controlling abiotic stress, fertilizer, why are you worried about all the basic? Sir, hangandre ನೀವು ಈಗ sir ಒಂದು ಹೇಳಲ್ಲ sir, you cannot solve everything. I think see, for you, you are not in charity business. You are in economic business. Ultimately, ನೀವೇನೋ yield ಜಾಸ್ತಿ ಕೊಡಬೇಕು, quality ಚೆನ್ನಾಗಿ ಕೊಡಬೇಕು, you have to get money in this one. Exactly, in this one. Okay, so and you are not interested in physiology, that is fine. So that means maybe there is something to correct in nutrient efficiency. ಅದಕ್ಕೇನೋ ಮಾಡಿಬಿಟ್ಟು ಅದು ಮಾಡು. ಇಲ್ಲ ಫ್ರೂಟ್ ಸೆಟ್ ಟೈಮಲ್ಲಿ ಏನೋ ಫ್ರೂಟ್ ಡ್ರಾಪ್ ಜಾಸ್ತಿ ಆಗ್ತಾ ಇದೆ, ಅದಕ್ಕೇನು ಮಾಡಬೇಕು? Is it there is something like we can do with the biostimulant? And I am not saying that ಎಲ್ಲದಕ್ಕೂ biostimulant ರಾಮಬಾಣ ಅಲ್ವೇ ಅಲ್ಲ. Sometimes it is not even the solution. Simple phytohormones or PGR, plant growth regulators, can do the job. ಇವೆಲ್ಲ ಹೈಫೈ ಬೇಕಾಗಿಲ್ಲ. So you have to be knowing exactly ನನಗೆ ಇಷ್ಟೇ ಬೇಕು. If it is done through phytohormones, it is done, do it with phytohormones only, no worry. PGRs or illa combination of idu work agutte antandre idu. But you should know exactly this one. But what biostimulants can do for that, I can tell. I can tell you. With whatever research I know, with whatever products I do, this one. Okay. Okay, second criteria is now, like again coming back to applying a biostimulant without knowing the stress that is pulling down your yield, then that is a fundamental flaw in your agriculture practice. ಈಗ yield ಕಮ್ಮಿ ಬರ್ತಾ ಇದೆ ಅಂತಂದ್ರು, ಈಗ ಅವ್ರು ಯಾರೋ ಹೇಳಿದ್ರಲ್ವಾ, ಸೊ ಇದು ಒಂದೊಂದು ಕಡೆ ಬರ್ತಾ ಸೇಮ್ ವೆರೈಟಿ ನಾವು ಅವ್ರ ರಿಜ್ವಾನ್ ಗೈ ಸೆಡ್ ದ ಸೇಮ್ ವೆರೈಟಿ ತಮಿಳ್ನಾಡು ಅಲ್ಲಿ ಒಂದು ರೀಜನ್ 20 kilometers you go ahead, suddenly that yield is going drop. That means by this time you all should be familiar what is the exact reason that is pulling down the yield. Sir, any idea what is the major abiotic stress that can contribute to difference in Gherkin yield from your experience? Any answer? Huh? Temp. Night temperature is very important, exactly. Okay. Okay. And now guess, what can you do, sir? Now, okay, you cannot control the sun and all this. Now tell us what do you, now we heard a little bit about this thing. So what can we do? Can we use transparents, anti-transparents? Yes, anti-transparent to reduce this thing. And it is like there is a temperature outside. Even within the leaf temperature, if there are too many, the temperature is very high, most of the active proteins are degraded. They are functional proteins, they are helping metabolism. So you can use something like this, anti-transparents, to spray on the plants. So you see, this is a very simple answer. We don't need anything. Exactly. Now you both told the temperature, then we'll find the exact solution for the temperature to this one. Other than temperature, any other things? Downy mildew, is downy mildew pulls your yield down regularly? Is it soil condition? Extremely. See, this is very important. How many people know your soil type in your growing area? Is it like a, what do you call, sandy soil, silty soil? How much is the organic carbon in that one? pH, organic carbon, anything, how much we know, and how many, is it towards the salinity side or this thing? Sir, basics, sir. I mean, sometimes now we do a lot of things. We talk about biostimulant, PGR, but sometimes we don't get the basic right. Yes, sir. What is they applying?
Fertilizer, sir.
Yeah.
Water soluble fertilizer, they are applying 5 kg, 10 kg per time. And it is not getting uptaken by the plants.
Yeah, okay. Now I'll come back to, I think we covered this, very good question. Now if you just do some soil analysis and you know that there is more sodium ions in your soil, wherever in this one. I showed one slide with the humic acid. So the best humic acid can actually bind sodium and also bind water molecule. Maybe that in that case, what we have to do is, now our humic acid we'll have to change. The humic acid regime we'll have to add more in those soil. That means sodium is sequestered repeatedly. Water is more available. Atara madbodu now. Without knowing what is the problem. Now you are saying, now actually we are talking. One you are saying temperature. Now we are saying there is a lot of sodium ions in this one. Now if it is sodium, we will see, then we can design specific humic acids, good potent humic acid, repeated application. Sir, ನಿಮ್ಮ ಸಾಯಿಲಲ್ಲಿ ಸೋಡಿಯಂ ಇದೆಯಾ ಸರ್? ಜಾಸ್ತಿ ನೋಡಿದ್ದೀರಾ ನೀವು? ಅಲ್ಲ ನೀವು ಹಾಂ. ಸೊ ಹಂಗಿದ್ದಾಗ ನಾನು ಅದಕ್ಕೆ ಹೇಳಿದೆ ಸರ್, ಆವಾಗ ನೀವು ರಿಪೀಟೆಡಾಗಿ ನೀವು root is the brain, sir. Root is the brain of the plant. And if you get that root, ಅಲ್ಲಿ ಸೋಡಿಯಂನ ರಿಡ್ಯೂಸ್ ಮಾಡಿ, ಸೋಡಿಯಮ್, ಬೈಕಾರ್ಬನೇಟ್ಸು, ಆಮೇಲೆ ಬೇರೆ ಸಾಲ್ಟ್ಸ್ ಏನೇನಿದೆ ಅದೆಲ್ಲ ರೂಟಿಗೆ ಬರ್ದಂಗೆ good quality. Maybe humic acid is a good solution for that case in this way. Same variety or different variety? Okay. Okay. Absolutely, madam. Exactly. Exactly. So, two things ಒಂದು ಮಾಡ್ಬೋದು. ನನ್ನ ಕಡೆಯಿಂದ ಐ ವಿಲ್ ಲಿಸ್ಟ್ ಔಟ್ ಆಲ್ ದ ಕಂಪ್ನಿ ನೇಮ್ಸ್ ಹೇಳಿದ್ರು ಸರ್. ನಿಮ್ಮ ಕಡೆಯಿಂದ ಈ ಥರ ಈಗ ಇವರು ಟೆಂಪ್ರೇಚರ್ ಹೇಳಿದ್ರು, ಸಾಲ್ಟ್ ಹೇಳಿದ್ರು, 4 or 5 ಕೀ ಬರ್ನಿಂಗ್ ಇಶ್ಯೂಸ್ ಇನ್ ಗರ್ಕಿನ್ ಏರಿಯಾ. ಯು ಶುಡ್ ಬಿ ಲಿಸ್ಟೆಡ್ ಸರ್. ನಿಮ್ಗೆ ಹೇಳಂಗೆ ಇರಬೇಕು ಇವಾಗಲೇ. ನಮ್ಗೆ ಗೊತ್ತಿರಿ ಇದು ಅಂತ. ಈಗ ನಮ್ಗೆ 10 ಟನ್ ಬರ್ತಾ ಇದೆ, ಇದನ್ನು ಅಡ್ರೆಸ್ ಮಾಡಿದ್ರೆ 14 ಟನ್ ಬರುತ್ತೆ ಅಂತ ನಿಮ್ಗೆ ಗೊತ್ತಿರಬೇಕು ಇಷ್ಟೊತ್ತಿಗೆ. ದೆನ್ ಸೊಲ್ಯೂಷನ್ ಈಸ್ ಈಸಿ. ನಾವು ಸುಮ್ಮನೆ ಹೊಡಿತಾ ಇದ್ದೀವಿ ಗಾಳಿಯಲ್ಲಿ ಗುಂಡು ಹೊಡೆದಂಗೆ ಸರ್. ಟಾರ್ಗೆಟ್ ಬೇಕು ನಮಗೆ. ಟಾರ್ಗೆಟ್ ಇದ್ದರೆ ಹೇಳಿ. ವೆನ್ ವಿ ವಿಲ್ ಟಾಕ್ ಟು ಎಕ್ಸ್ಪರ್ಟ್ಸ್, ವಿ ವಿಲ್ ಟಾಕ್ ಟು ಗುಡ್ ಕಂಪನೀಸ್, ವಿ ವಿಲ್ ಸೇ ವಾಟ್ ಈಸ್ ದಿಸ್. ಯಾರೋ ಚಾಲೆಂಜ್ ಮಾಡ್ತಾರೆ, ಇಲ್ಲ ಇದು ಹಾಕೋದು ಇದು ಪ್ರಾಬ್ಲಮ್ ಅಂತ. ಯಾವಾಗ ಬಂದು ಸರ್ ನಿಮ್ಗೆ ಗೈಡ್ ಮಾಡ್ತಾರೆ. ಎಲ್ಲ ಒಂದು 10 acres ಒಂದು ಟ್ರಯಲ್ ತಗೊಳ್ಳಿ ಸರ್. ಇಫ್ ಯು ಸಿ ವಂಡರ್ಫುಲ್ ರಿಸಲ್ಟ್ ಸಾಲ್ವಿಂಗ್ ದಿಸ್ ಒನ್, ದೆನ್ ಯುವರ್ ವ್ಯಾಲ್ಯೂ ಈಸ್ ಆ್ಯಡೆಡ್. ಅದು ನಾವು ದಟ್ ಈಸ್ ದ ಬೆಸ್ಟ್ ವೇ ಅಂತ ನನಗೆ ಅನಿಸುತ್ತೆ ನಮಗೆ ಮಾಡೋದು. ಓಕೆ, ಸೊ ಅಟ್ಲೀಸ್ಟ್ ನೀವು ನೆಕ್ಸ್ಟ್ list me all those factors that is pulling down the yield in Gherkin. We can bring good solutions for this one. So adike ಹೇಳ್ತಾ ಇರೋದು, first you should understand the specific environment condition and nutrition challenge. Either is it stress or nutrition. No complications sir. ಇಲ್ಲಿ ಎರಡೇ ಬರೋದು. either nutrition ಇಂದ ಪ್ರಾಬ್ಲಮ್ ಬರುತ್ತೆ, ಇಲ್ಲ ಸ್ಟ್ರೆಸ್ ಇಂದ ಪ್ರಾಬ್ಲಮ್ ಬರುತ್ತೆ. ನ್ಯೂಟ್ರಿಷನ್ ಪ್ರಾಬ್ಲಮ್ ಇದೆ, ಸ್ಟ್ರೆಸ್ ಐಡೆಂಟಿಫೈಡ್ ಕ್ಲಿಯರ್ಲಿ. ಡೀಪ್ ಡೈವ್ ಮಾಡಿ. I think like I said, inherently all of you are agronomists. Course agriculture ಮಾಡಿರಲಿ ಬಿಡಲಿ, you are all agronomists. You will identify this problem. So please identify that nutrition ಅಥವಾ environment problem ಅಂತ. Then based on that select the biostimulant with known mechanism for identified stress or goal. ನಮ್ಗೆ ಇವಾಗ ಹೇಳಿದ್ನಲ್ಲ ಇಷ್ಟೊಂದು ಅವೆಲ್ಲ ಏನೇನಿದೆ, ಅದರಲ್ಲಿ ಯಾವುದಾದ್ರೂ ಫಿಟ್ ಆಗುತ್ತಾ, ಫಿಟ್ ಆಗದೇ ಇರ್ಬೋದು. There is other solution. Like I said, biostimulant is not the answer for everything. Then we'll do second step, that one. Third one is you have to follow the manufacturer recommendation and mode of application. ಇಲ್ಲೇ ತಪ್ಪು ಮಾಡೋದು. ಇಫ್ ಗೋ ಬ್ಯಾಕ್, ಅವ್ರು ಏನೋ ರೆಕಮೆಂಡ್ ಮಾಡಿ ಟೆಸ್ಟ್ ಮಾಡಿರ್ತಾರೆ ಕಂಡೀಷನಲ್ಲಿ. ನಾವು ಯಾವುದೋ ಕಂಡೀಷನಲ್ಲಿ ಅದನ್ನು ಹೊಡೆದು ಎಕ್ಸ್ಟ್ರಾ ಡೋಸಲ್ಲಿ ಹಾಕಿ ಅದ್ರದ್ದು ಎಫೆಕ್ಟ್ ಹೋಗಿ ಆಮೇಲೆ ಚೆನ್ನಾಗಿಲ್ಲ ಪ್ರಾಡಕ್ಟ್ ಅಂತ ಡಿಸ್ಕಾರ್ಡ್ ಮಾಡಿ. ಸೊ ಐ ಥಿಂಕ್ ವಿ ಶುಡ್ ಫಾಲೋ ದಟ್ ರೆಕಮೆಂಡೇಶನ್ ಅವರು ಮಾಡಿದ್ದಾರೆ. ಸೊ ಅಟ್ಲೀಸ್ಟ್ ದಿಸ್ ಈಸ್ ವಾಟ್ ಐ ಕ್ಯಾನ್ ಸೇ ಆಸ್ ಅ ಸೆಕೆಂಡ್ ಕ್ರೈಟೀರಿಯಾ ನಾವು. ಓಕೆ, good. So last criteria is based on that I have some I have two criteria. I have some recommendation. This is nothing to do with biostimulant. I have biostimulant recommendation also, but it's not the full proof. But based on some publications, what they have done here, I have listed some 6 things. One is people can use malic hydrazide 100 ppm. If the stress is more or something, I think they use 200 ppm, but range is between this and ethafone. Consistently they have seen increased in number of nodes, main stem, and number of nodes per unit length vine, fruits, fruit yield, especially flower and fruit characteristics are significantly improved in studies, and these are good publications. This is one recommendation. I don't know whether you have used it, but I am just...
Sir, which crop it is?
This is in cucumbers.
Cucumber, okay.
This is not, because there is not available much information on gherkins alone, but wherever I have picked up, but this is not exhaustive list. But ನೀವು flowering time alli, so you can try 100 ppm of malic hydrazide and this one. Malic hydrazide is a inhibitor of ABA synthesis, abscisic acid synthesis. Abscisic acid ಬಿದ್ರೆ ಏನಾಗುತ್ತೆ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ, they senescence very fast. ಏನಾಗುತ್ತೆ, in though the plant has potential to make more fruits, what will do is that abscisic acid ಇರೋದ್ರಿಂದ they will go to senescence very fast. ಅವಾಗ ಫ್ರೂಟ್ ಸೈಜು ಎಲ್ಲ ಕಮ್ಮಿ ಆಗಿಬಿಡುತ್ತೆ. So in that case people are using this malic hydrazide. So malic hydrazide inhibits this abscisic acid promoting back ಇದು ಏನೋ ಜೀನ್ಸು ಇದನ್ನು. So the fruit size is normal ಆಗಿ ಬರುತ್ತೆ ಅದು ಅದರಲ್ಲಿ. ಈ ಎತ್ತರಲ್ಲಿ ಮೇಜರ್ ಐ ಡೋಂಟ್ ನೋ ಇದು ಇವೆಲ್ಲ ಏನೋ ಪ್ರಿಕಸರ್ಸ್ ಫಾರ್ ಎಥಿಲಿನ್ ಬಯೋಸಿಂಥಸಿಸ್ ಅಥವಾ ಎಥಿಲಿನ್ ಬಯೋಸಿಂಥಸಿಸ್ ಎತ್ತರಲ್ಲಿ ಇಟ್ಸ್ ಆಲ್ಸೋ ಯೂಸ್ಡ್. ಆಮೇಲೆ people have seen gibberellic acid. I think most of you people, I want to little bit hear from gibberellic acid. How are gibberellic acid ಎಷ್ಟು ಡೋಸಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ, what is the purpose, Where are you using? Anybody, sir? Anybody from Gherkin? Okay. ಅಲ್ಲ PPM ಅಲ್ಲಿ ಹೇಳಿ ಪರ್ಸನ್ 100 PPM 100 PPM. And what is the purpose of using GA3 is to fruit set flower initiation. Okay. So you are happy with GA3? Okay. Huh? Okay. So illen heltare andre sir ನೀವು ಇದರ ಜೊತೆ combine with potassium nitrate just 2.5. Yes. Okay. So that is one recommendation based on the research findings I have found. So just ಇದು ಏನಂದ್ರೆ ನಿಮಗೆ plant growth ಜೊತೆ development production of fruits ಎಲ್ಲ ಇದಾಗುತ್ತೆ. Maybe you can try adding potassium nitrate. Yes sir. ಏನು ಹೇಳಿ? ಹಾಂ, ಏನಾಗಲ್ಲ. ಐ ಥಿಂಕ್ ಜಿಬರ್ಲಿಕ್ ಆ್ಯಸಿಡ್ ಶುಡ್ ಬಿ ಇನ್ನೂ ಸ್ವಲ್ಪ ಫ್ಲವರ್ ಸೆಟ್ ಆಗೋಕ್ಕಿಂತ ಮುಂಚೆ ಅದು. ಸೊ ಇಟ್ ಶುಡ್ ಬಿ ಯೂಸ್ಡ್. ಟೂ ಲೇಟಲ್ಲಿ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಇಟ್ಸ್ ನಾಟ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಯೂಸ್ಫುಲ್. ಎಸ್ ಪ್ರಿಕಸ್ ಎಸ್ ಎಸ್ ಓಕೆ. ಯು ಶುಡ್ ಬಿ ತುಂಬ ಲೇಟಾಗಿ ಮಾಡಿದ್ರೆ
ಏನಾಗ್ಬಿಡುತ್ತೆ, 80 ಡೇಸಲ್ಲಿ ನಮ್ಮ ಫ್ರೂಟು ಶಾರ್ಟ್ ಆಗ್ತಾ ಇರುತ್ತೆ.
ಏನಾಗ್ತಾ ಇರುತ್ತೆ?
ಫ್ರೂಟು ಶಾರ್ಟ್ ಇದು ಬರ್ತಾ ಇರುತ್ತೆ. ಸೊ ಅವಾಗ ಏನಾದ್ರು ಜಿಬ್ಲಾಕ್ ಆಸಿಡ್ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಅದು ಏನಾದರೂ ಹೆಲ್ಪ್ ಆಗುತ್ತಾ?
ಸಿ, ನೀವು ಏನೇ ಮಾಡಿದ್ರು ಒಂದು ಕ್ರಾಪ್ದು ಫ್ರೇಮ್ ಇರುತ್ತೆ, ಆ ಫ್ರೇಮ್ ಒಳಗೆ ಮಾಡಬೇಕು ನೀವು ಅಂದರೆ ಇವಾಗ 90 ಡೇಸಲ್ಲಿ ಅದರದ್ದು ಕ್ರಾಪ್ ಸೈಕಲ್ ಮುಗಿಯುತ್ತೆ ಅಂತಂದರೆ ನೀವು 80th ಡೇಯಲ್ಲಿ ಜಿಬರ್ಲಿಕ್ ಆಸಿಡ್ ಹಾಕ್ಬಿಟ್ಟು 10 ಡೇಸಲ್ಲಿ ಫ್ರೂಟ್ ಸೈಜು ಇಟ್ ನೆವರ್ ಹ್ಯಾಪನ್. ಪ್ರೋಗ್ರಾಮ್ ಆಗಿದೆ. ಇಟ್ ಈಸ್ ಲೈಕ್ ಕಂಪ್ಯೂಟರ್ ಪ್ರೋಗ್ರಾಮ್ ಪ್ಲಾಂಟ್ಸ್ ಇದು. ಸೊ ಇಟ್ ಈಸ್ ಲೈಕ್ ಮೇ ಬಿ ದೇರ್ ಇಸ್ ಅದರ್ ಡಿಫಿಷಿಯನ್ಸಿ. ಇಟ್ ಈಸ್ ನಥಿಂಗ್ ಟು ಡೂ ವಿತ್ ಇದು. ನ್ಯೂಟ್ರಿಷನಲ್ ಡಿಫಿಷಿಯನ್ಸಿ ಇತ್ತು ನಿಮ್ಮನ್ನ 80 ಡೇಸ್ಗಾಗಲೇ ಯು ಶುಡ್ ಹ್ಯಾವ್ ಎ ಡೀಸೆಂಟ್ ಫ್ರೂಟ್ ಸೆಟ್. ನೀವು ಅಂದರೆ 80 ಡೇಸಲ್ಲಿ ನಿಮ್ಗಾಗಲೇ ಫ್ರೂಟ್ ಶೇಪ್ ಸೈಜ್ ಅಂದರೆ ಶೇಪು ಅದು ಡೆವಲಪ್ಮೆಂಟಲ್ ಡಿಫಾರ್ಮಿಟಿ. ಸೊ ಮೇ ಬಿ ಓಕೆ ಓಕೆ. ಬಟ್ ನಾನು 10 ಡೇಸ್ ನಾನು ವಾಟ್ ಐ ಆಮ್ ಸೇಯಿಂಗ್ ಇಸ್ 90 ಡೇಸ್ಗೆ ಹಾರ್ವೆಸ್ಟ್ ಬಂದಿದೆ ಅಂತಂದರೆ
80 ಹಾರ್ವೆಸ್ಟ್ ಮಾಡ್ತಾ ಇದೀವಿ ಸರ್, 90 ಡೇಸಿಗೆ ಕ್ಲೋಸ್ ಆಗುತ್ತೆ.
ಓಕೆ.
ಈಗ ನಾವು 80ನೇ ದಿವಸದಲ್ಲಿ ನಮ್ದು ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಶಾರ್ಟ್ ಆಗಿ ಕಾಣ್ತಾ ಇದೆ ಫ್ರೂಟ್ಗಳು.
ಓಕೆ.
ಸೊ ಅವಾಗ ಜಿಬ್ರಾಲಿಕ್ ಆಸಿಡ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿದ್ರೆ ನಾವೇನಾದ್ರು
ನಾನು ಅದೇ ಹೇಳ್ತಾ ಇರೋದು. ಇನ್ನೊಂದು 5 ಡೇಸ್ 6 ಡೇಸ್ ಏನಿದೆಯಲ್ಲ ಕ್ರಾಪ್ ಮುಗಿಯೋಕೆ ಬರುತ್ತಂತ, ದಟ್ ಈಸ್ ಮೇ ಬಿ ಟೂ ಲೆಸ್ ಟು ಅಂತ ನನಗನ್ಸುತ್ತೆ. ಓಕೆ ಓಕೆ ಓಕೆ. ಗೊತ್ತಿಲ್ಲ ಸರ್, ಜಿಬರ್ಲಿಕ್ ಆಸಿಡ್ ಆನ್ ಫ್ರೂಟ್ ಶೇಪು ಐ ಡೋಂಟ್ ನೋ ಮಚ್ ಇನ್ಫಾರ್ಮೇಷನ್, ಬಟ್ ವಿಲ್ ಹ್ಯಾವ್ ಟು ಫೈಂಡ್ ಔಟ್. ಯಾಕಂದರೆ ಜಿಬರ್ಲಿಕ್ ಆಸಿಡ್ ಆನ್ ನಂಬರ್ ಆಫ್ ಫ್ಲವರ್ಸು ಫ್ರೂಟ್ ಸೆಟ್ಟು ಇದ್ರ ಬಗ್ಗೆ ಕೇಳಿದ್ದೀವಿ. ಶೇಪ್ ಆಫ್ ದ ಫ್ರೂಟು ವಿತ್ ಜಿಬರ್ಲಿಕ್ ಆಸಿಡ್ ಸಾರಿ ಐ ಡೋಂಟ್ ಹ್ಯಾವ್ ಮಚ್ ಇನ್ಫಾರ್ಮೇಷನ್, ಬಟ್ ಐ ವಿಲ್ ಟ್ರೈ ಟು ಸಿ ಇಫ್ ಐ ಕ್ಯಾನ್ ಗೆಟ್ ಸಮ್ ಇನ್ಫಾರ್ಮೇಷನ್. ಓಕೆ.
ಈ ಜಿಬ್ರಾಲಿಕ್ ಆಸಿಡ್ ನಮ್ಮ ಗರ್ಕಿನ್ ಗಿಡಕ್ಕೆ ಎಷ್ಟನೇ ದಿವಸದಲ್ಲಿ ಹೊಡಿಸ್ಬೋದು ನಿಮ್ಮ ಒಂದು ಸಜೆಷನ್?
ಏನು ಹೇಳಿ ಸರ್?
ಎಷ್ಟನೇ ದಿವಸದಲ್ಲಿ ಹೊಡಿಸ್ಬೋದು
ಸರ್? ಫ್ಲವರ್ ಸೆಟ್ಟಿಂಗ್ ಆನ್ ಸೆಟ್ ಆಗುತ್ತಲ್ಲ, ಆವಾಗ ಸೆಟ್ ಆಗಿರೋ ಮುಂಚೆನೇ ಹೊಡಿತಾರೆ. ಸ್ವಲ್ಪ ಅಬ್ಜೆಕ್ಟಿವ್ ಏನಂದರೆ ನಿಮಗೆ ನಂಬರ್ ಆಫ್ ಫ್ಲವರ್ಸ್ ಜಾಸ್ತಿ ಬರಬೇಕು, ಫ್ಲವರ್ಸ್ ಟು ಫ್ರೂಟ್ ಕನ್ವರ್ಷನ್ ಚೆನ್ನಾಗಿರಬೇಕು, ಆಮೇಲೆ ಈ ನಂಬರ್ ಆಫ್ ಲ್ಯಾಟ್ರಲ್ ಬ್ರಾಂಚಸ್ ಜಾಸ್ತಿ ಆದಷ್ಟು ಜಾಸ್ತಿ ಫ್ಲವರ್ಸ್ ಬರುತ್ತೆ, ಈಲ್ಡ್ ಜಾಸ್ತಿ ಬರುತ್ತೆ ನಿಮಗೆ.
ನಾವು ಅಬ್ಸರ್ವ್ ಮಾಡಿರೋಂಗೆ ಡೌನಿ ಮಿಲ್ಡಿನೂ ಜಾಸ್ತಿ ಆಗುತ್ತೆ.
ಆಗುತ್ತೆ, ಯಾಕೆಂದರೆ ನಿಮಗೆ ಶೂಟ್ ಗ್ರೋತ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ಅದರಿಂದ ಸೆಲ್ ಡಿವಿಷನ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ಅವಕ್ಕೆ ಜಾಸ್ತಿ ಫೋಲೇಜ್ ಸಿಗುತ್ತೆ, ಫ್ರೆಶ್ ಫೋಲೇಜ್ ಸಿಗುತ್ತೆ. ಆಬ್ವಿಯಸ್ಲಿ ಇದು ಟ್ರೇಡ್ ಆಫ್ ಸರ್. ಒಂದು ಬೇಕು ಅಂದರೆ ಇನ್ನೊಂದು ಕಳ್ಕೊಬೇಕು ನೀವು, ಹಂಗೆ ನಿಮ್ಗೆ ಇಳ್ಬೇಕು ಅಂತಂದರೆ ಇದಾಗುತ್ತೆ. ಇಟ್ ಈಸ್ ಪಾಸಿಬಲ್. ನೀವು ಶೂಟ್ ಮಾಸು ಇದೇನೋ ಇನ್ನೇನು ಸೆನೆಸೆನ್ಸ್ ಟೈಮಲ್ಲಿ ಜಾಸ್ತಿ ಬಯೋಮಾಸ್ ಬಂದಷ್ಟು ಥಿಕ್ ಫೋಲೇಜ್ ಬಂದಾಗ ಏನಾಗುತ್ತೆ, ಫಂಗಸ್ಗೆಲ್ಲ ಈಸಿ, ದೇರ್ ಇಸ್ ನೋ ಎರೇಷನ್, ಹ್ಯೂಮಿಡಿಟಿ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ದೇ ಬಿಲ್ಡಪ್ ಹಾಗೆ ಆಗುತ್ತೆ. ಸೊ ಫೋಲೇಜ್ ಜಾಸ್ತಿ ಆದರೆ ಫಂಗಸ್ ಬಂದೇ ಬರುತ್ತೆ. ಅದೇ ಥರ ನೀವು ಇನ್ನೊಂದು ಐ ಡೋಂಟ್ ನೋ ಹೌ ವಾಟ್ ಇಸ್ ದ ಡೋಸ್ ಆಫ್ GA3 ಯು ಯೂಸ್? PPM ಅಲ್ಲಿ ಹೇಳ್ಬೇಕು ನನಗೆ. 1 ಗ್ರಾಮ್ ಇನ್? ಓಕೆ. Okay, 1 gram in 200 liters is, okay, ಎಷ್ಟಾಯ್ತು ಒಂದು ಲೀಟರ್ ಇದಕ್ಕೆ ಎಷ್ಟಾಯ್ತು? 200 ppm is high. At least what I understand is, all the phytohormones, 100 ppm. At least GA3 we should be 100 or less. ಹೌದು, don't go for more than 200. ಹೌದೌದು. 1 gram 200 liters ಅಂತಂದರೆ ಇಫ್ ಡಿವೈಡ್ ಬೈ 5 ಅಂದರೆ 0.2 ಗ್ರಾಮ್ ಪರ್ ಲೀಟರ್ ಆಯಿತು. 0.2 ಗ್ರಾಮ್ 200 ppm ಆಯಿತು ಸರ್. ಅದು 250 ಜಾಸ್ತಿ ಆಯಿತು, 200 ppm ಜಾಸ್ತಿ ಆಯಿತು. 100 ಸಾಕು. ಫೈಟೋಹಾರ್ಮೋನ್ಸಲ್ಲಿ ಯಾವತ್ತು ಡೋಂಟ್ ಗೋ ಮೋರ್ ದೆನ್ ಅಟ್ಲೀಸ್ಟ್ ನಮ್ಮ ರೆಕಮೆಂಡೇಶನ್ ಕುಕ್ಕರ್ ಬಿಟ್ಸ್ ವೆರಿ ಸೆನ್ಸಿಟಿವ್ ಕ್ರಾಪ್ಸ್. ಸೊ ಎರಡನೇದು ಏನಾಗುತ್ತೆ ಅಂತಂದರೆ ಕೆಲವೊಂದು ಸಲ ನೀವು 200 ppm ಅಷ್ಟು ಹೋಗ್ಬಿಟ್ರೆ ಇಟ್ ವಿಲ್ ಹ್ಯಾವ್ ಎ ರಿವರ್ಸ್ ರೋಲ್. ಇನ್ಸ್ಟೆಡ್ ಆಫ್ ಗ್ರೋತ್ ಪ್ರಮೋಷನ್ ಗ್ರೋತ್ ರಿಟಾರ್ಡೇಷನ್ಗೆ ಹೋಗ್ಬಿಡ್ತಾರೆ. ಅದು ಕಾಂಪ್ಲೆಕ್ಸ್, ನಮಗೂ ಅರ್ಥವಾಗಲ್ಲ ಹೌ ಇಟ್ ಈಸ್ ಡನ್ ಅಂತ. ಸರ್ 1 ಗ್ರಾಮ್ ಇನ್ 1 ಲೀಟರ್ ಈಸ್ 1 ppm. ಸಾರಿ 1 milligram ಅಲ್ಲ, 1 milligram in 1 liter is 1 ppm. ppm ಅಲ್ಲ, ನಾವು ಒಂದು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡ್ತೀವಿ. ನೀವು ನನ್ನ ಪ್ರಕಾರ 100 ppmಗಿಂತ ಕಮ್ಮಿ ಮಾಡ್ತಾ ಇದ್ದೀರಾ. ಓಕೆ, 100 ppmಗಿಂತ ಲೆಸ್ ಹ್ಯಾಸ್ ಪಾಸಿಟಿವ್ ಎಫೆಕ್ಟ್ ಆನ್ ಕುಕ್ಕರ್ ಬಿಟ್ಸ್ ಅಂತ ಆಲ್ ಮೈ ರಿಸರ್ಚ್ ಫೈಂಡಿಂಗ್ಸ್ ವೇರ್ ಎವರ್ ಐ ಸಾ ಪಬ್ಲಿಕೇಶನ್ಸ್ ಇಸ್ ಇಫ್ ಯು ಗೋ ಮೋರ್ ಇಟ್ ಹ್ಯಾಸ್ ಸಮ್ ಎಫೆಕ್ಟ್ ಆನ್ ದಿಸ್ ಒನ್. ಆಮೇಲೆ ಈ ಡೇ ಲೆಂತ್, I think day length, of course, gibberellic acid will improve. There is no doubt about it. ಅಲ್ಲೂ ಅಷ್ಟೇ, you cannot go very, very high ಅಂತಾರೆ. ಇದು ಸಬ್ಜೆಕ್ಟಿವ್ ಸರ್, ವೆರೈಟೀಸ್ ಯಾವುದಿದೆ ಅದ್ರ ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗುತ್ತೆ. ಸೊ ವಿ ಕಾಂಟ್ ನೋ. ಎನಿವೆ ವಿಲ್ ಮೂವ್ ಆನ್. ಸೊ ವಾಟ್ ಐ ಆಮ್ ಸೇಯಿಂಗ್ ಈಸ್, just instead of using just gibberellic acid, try using with potassium nitrate. Research findings are supporting it very, very well ಅಂತ ಹೇಳ್ತಾರೆ. ಓಕೆ, ಇನ್ನು ಕೆಲವರು ಇದು NaA ಗೊತ್ತಾ ನಿಮಗೆ? ನಾಫ್ಥಲಿನ್ ಅಸಿಟಿಕ್ ಆಸಿಡ್. ಇಟ್ ಈಸ್ ಅನಲಾಗ್ ಆಫ್ ಆಕ್ಸಿನ್, ಇಂಡಿಯನ್ ಅಸಿಟಿಕ್ ಆಸಿಡ್ ಸಿಂಥಸಿಸ್. ಸೊ ಇಟ್ಸ್ ಆಲ್ಸೋ ಪೀಪಲ್ ಯೂಸ್ ವೆನ್ ಯು ಯೂಸ್ ವೆರಿ ವೆರಿ ಲೋ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಫ್ GA3, ಇಟ್ ಈಸ್ ಬೆಟರ್ ಟು ಯೂಸ್ ದಿಸ್ NaA. ಇದೇನಾಗುತ್ತೆ ಅಂತಂದರೆ, ಇದು ಜಿಬರ್ಲಿಕ್ ಆ್ಯಸಿಡ್ ಲ್ಯಾಟ್ರಲ್ಲು ಸೆಲ್ ಡಿವಿಷನ್ಸು ಇದು ಜಾಸ್ತಿ ಆಗುತ್ತೆ ನಿಮಗೆ, ಬಟ್ ಮೇಲ್ಗೆ ಹೋಗ್ತಾ ಇರುತ್ತಲ್ಲ ಅದು ಕಮ್ಮಿ ಆಗುತ್ತೆ. ದಟ್ ಈಸ್ ವೈ ಯು ಯೂಸ್ NaA. In that case, you are balancing the apical growth as well as lateral growth by adding a little bit of NaA. ಜಾಸ್ತಿನೂ ಯೂಸ್ ಮಾಡಬಾರ್ದು. See, when you use 100 ppm of this and very low concentration, you will get ideal branches. Number of branches ಎಲ್ಲ ಆಪ್ಟಿಮಮ್ ಆಗಿ ಬರುತ್ತೆ ಅಂತ. Don't use just GA3. You can combine, play around little bit with acetic acids and all those things. ಇನ್ನೇನು ಅದೇ ಮೇಜರ್ ಇಸ್ GA3 ಟ್ರೀಟ್ಮೆಂಟ್. GA3 ಇದು ಸೀಡ್ ಟ್ರೀಟ್ಮೆಂಟು ಮಾಡ್ತಾರೆ ಕೆಲವರು. ಇಫ್ ಯು ಐ ಡೋಂಟ್ ನೋ, ಇಫ್ ಯು ರೂಮ್ ಇದ್ದರೆ ಯು ಸೀಡ್ಲಿಂಗ್ಸ್ ತಾನೆ, ನೀವು ಸೀಡ್ಸ್ ಹಾಕ್ತೀರಾ? ಇಲ್ಲ, ಸೀಡ್ಸ್ ಕ್ಯಾನ್ ಬಿ ಸೋಕ್ಡ್ ಪ್ರೀ ಟ್ರೀಟೆಡ್ ವಿತ್ GA3 75 ppm. ಇದೇನಂತಂದರೆ ಎನಿ ಡಾರ್ಮೆನ್ಸಿ ಎನಿಥಿಂಗ್ ಈಸ್ ಬ್ರೋಕನ್ ಅವೇ ಆ್ಯಂಡ್ ಇದು ಇಮ್ಮಿಡಿಯೇಟ್ ಜರ್ಮಿನೇಷನ್ ಯೂನಿಫಾರ್ಮ್ ಬರುತ್ತೆ ನಿಮಗೆ. ಸಿ and 100% germination, then not a, then that's fine. Okay, that's fine. Okay. 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 I'll ask you in this a different way. If you put 100 seeds, how many seeds germinate? 95. And
Okay.
Then it's fine. Okay, then you don't have any issue. Then don't worry about to buy. So you have identified the stress correctly in this case. This is what I was telling you, identified correctly, then no need of any solution. Don't waste your time.
Sir, in seeds, when the company, whichever the company, I am telling, if 1000 seed packets coming means at least minimum some 40, 50 seeds inside kernel is not there. It is actually 950 seeds only, it is eligible for the sowing. 50 seeds will go.
Oh, I see.
Then, but if you sow the 950 seed, then it will be 900 plants.
Okay.
It will be like that. No, it is there. You can, I'm not saying any company, it is there.
Okay, good. Okay, sir. So this was a little bit about phytohormone, though it is not my topic, but I thought, I read some publication. You guys are so busy, you are mostly in the field, but I thought, okay, some good recommendations that works in cucurbits, it should work. Okay. Now, I also have some recommendations for you for the ಇದನ್ನು ಯೂಸ್ ಮಾಡ್ಬೋದು ನೀವು. ಅದರ ಫೈಟೋಹಾರ್ಮೋನ್ ಹೇಳಿದೆ, ಇದ್ರ ಜೊತೆಗೆ ನೀವು ಸೀವಿಡ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಹೇಳಿದ್ನಲ್ಲ, ಅದರಲ್ಲಿ ಅಸ್ಕೋಫೈಲಮ್ ನಡುಸಮ್ ಇರೋದು, ಕೆಲ್ಪ್ ಇರೋದು ಸೀವಿಡ್ ತಗೊಳ್ಳಿ. ಆ್ಯಂಡ್ ದೆನ್ ಯು ಟೇಕ್ ದ ಪ್ರೋಟೀನ್ ಹೈಡ್ರೋಲೈಸೇಟ್ ವಿತ್ ಫ್ರೀ ಅಮೈನೋ ಆಸಿಡ್, ಮೋರ್ ಫ್ರೀ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಇರೋದು. ಯು ಟೇಕ್ ದಟ್ ಪೋರ್ಷನ್ and then you can do 2 to 3 foliar sprayings in active growing stage and flowering stage. ಇದು ಓವರಾಲ್ ಸ್ಟ್ರೆಸ್ ಟಾಲರೆನ್ಸ್ ಆ್ಯಂಡ್ ಈಲ್ಡ್ ಗೇನ್ ಇನ್ ಗರ್ಕಿನ್ನಲ್ಲಿ ಬರುತ್ತೆ. ಸೊ ದಿಸ್ ಇಸ್ ಪ್ರೂವನ್. ದೇರ್ ಆರ್ ಸಮ್ ಪಬ್ಲಿಕೇಶನ್ಸ್ ಆನ್ ದಿಸ್ ಒನ್. ಸೊ ಎನಿವೇ ಸೀವೀಡ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ವಿತ್ ಅಮೈನೋ ಆಸಿಡ್ ಕಂಬೈನ್ಡ್, ಸ್ಪ್ರೇಯಿಂಗ್ ರೆಗ್ಯುಲರ್ಲಿ ಆನ್ ದಿಸ್ ಒನ್. ಎಲ್ಲಿ ಸ್ಪ್ರೇ ಓನ್ಲಿ ಇಫ್ ಯು ಹ್ಯಾವ್ ಐಡೆಂಟಿಫೈಡ್ ದ ಸ್ಟ್ರೆಸ್. If you have a region where plant height is variable, some plants are growing well, some are not. Sometimes ಈ ಕಡೆ ಬ್ಯಾಚ್ ಎಲ್ಲ ಚೆನ್ನಾಗಿ ಫ್ಲವರಿಂಗ್ ಆಗಿದೆ, ಈ ಕಡೆ ಚೆನ್ನಾಗಿಲ್ಲ, ಆ ಕಡೆ ಚೆನ್ನಾಗಿದೆ, ಈ ಥರ ವೇರಿಯಬಲ್ ಇದ್ದರೆ ಪ್ಲೀಸ್ ಟೇಕ್ ಅಪ್ ದಿಸ್ ಅಸ್ಕೋಫೈಲಮ್ ಆ್ಯಂಡ್ ಫ್ರೀ ಅಮೈನೋ ಆಸಿಡ್ ಪ್ರೋಟೀನ್ ಹೈಡ್ರೋಲೈಸೇಟ್ ಕಂಟಿನ್ಯೂಸ್ಲಿ. ಇದರಲ್ಲಿ ಇಟ್ ವಿಲ್ ನೋ ಹಾರ್ಮ್. ಇಫ್ ನೋ ಬೆನಿಫಿಟ್ ಇಲ್ಲ ಅಂದರೂ ಪರವಾಗಿಲ್ಲ, ನೋ ಹಾರ್ಮ್ ಅಂತೂ ಏನೂ ಇಲ್ಲ ಇದರಲ್ಲಿ. ಆಯಿತಾ? ದಟ್ ಇಸ್ ಸ್ಟ್ರೆಸ್ ಕಡೆ ಅಂದರೆ ಓವರಾಲ್ ಪ್ಲ್ಯಾಂಟ್ ಗ್ರೋತು, ಫ್ಲವರಿಂಗು, ಯೂನಿಫಾರ್ಮಿಟಿ, ಪ್ಲ್ಯಾಂಟ್ ಗ್ರೋತ್ಗೆ ಯೂಸ್ ಆಗುತ್ತೆ ಸರ್ ಇದು. ಆಯಿತಾ? ಆ್ಯಂಡ್ ಸೆಕೆಂಡ್ ಒನ್ ಈಸ್ seed incubations. Again, you said no problem is germination, so I will not put anything. So seed germination includes fresh young, andre establishmental problems idre, then you can have amino acids combined with molybdenum. Kelu products irbodu. So molybdenum element jate use ಮಾಡ್ತಾರೆ. Molybdenum gives resistance to this one, but anyway you are not having any problems with germination, we can skip that. Now again the same as this one, here also amino acid and seaweed extracts. Consistently research has shown that, so blindly start following it. If you are using seaweed extract, use kelp cold water extract and use amino acid fractions and spray on the foliar. Your probability of improving stress tolerance in gherkin is very high based on the research. Okay, so that is one thing. And then there is some gelatin hydrolyzed. So apparently sir, ನೀವು ಹೇಳಿದಂಗೆ more protein hydrolysates are used in gherkin. So after I finished this presentation, I came to know that protein hydrolysates are very effective in gherkins. So I will do one thing, I will also dig more research in this one, and you should include protein hydrolysates more in gherkin crops, is what I am also getting a feeling in this one. It's a new one, change is good always, who knows, and they are doing. Okay, so ಇದು ಏನು ಮಾಡ್ತಾರೆ, ಓಕೆ ಅಗೇನ್ ನೈಟ್ರೋಜನ್ ಅಸಿಮಿಲೇಷನ್ ಎಲ್ಲ ಇಂಪ್ರೂವ್ ಆಗುತ್ತಂತೆ ಪ್ರೋಟೀನ್ ಹೈಡ್ರೋಲೈಸೇಟ್ ಇದು. ಆ್ಯಂಡ್ ಸಮ್ ಪೀಪಲ್ ಆರ್ ಯೂಸ್ಡ್ ಸೀವಿಡ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ಸ್ ಲೈಕ್ ಪ್ರೋಟೀನ್ಸ್ ಲೈಕ್ ಕೈನೆಟಿನ್, ಜಿಬರ್ಲಿಕ್ ಆಸಿಡ್ಸ್, ಆ್ಯಂಡ್ ದೆನ್ ಇದು ನೋಡಿ ಸೂಪರ್ ಕಾಂಬಿನೇಷನ್ ಇದು. ದೇರ್ ಈಸ್ ಗ್ರೋತ್ ರೆಗ್ಯುಲೇಟರ್, ದೇರ್ ಈಸ್ ಸಮ್ ಪ್ರೋಟೀನ್ಸ್, ದೇರ್ ಈಸ್ ಸೀವಿಡ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ಸ್, ಆ್ಯಂಡ್ ದೆನ್ ದಿ ಫೈಟೋಹಾರ್ಮೋನ್. ಇಟ್ಸ್ ಲೈಕ್ ಆ್ಯಂಡ್ ಆಲ್ ಲೋ ಕಾನ್ಸಂಟ್ರೇಷನ್ಸ್, ಎಕ್ಸ್ಟ್ರೀಮ್. Yes. Yes, and this is a very good combination of combining seaweed extract and phytohormones, and probably foliar application of this has one of the most promising effect on overall growth, development, fruit set in gherkin. So if you have to take away something from me sir, I will recommend this today for you. Sir, ನಾನೇ ಮಾಡ್ಬೇಕು ಸರ್ ನನ್ನ ಕಂಪ್ನಿಯಲ್ಲಿ. ಸರ್ ಈಗ ಏನತ್ತಾ, ಇನ್ ಇಂಡಿಯಾ ಟುಡೇ ದ ಮೋಸ್ಟ್ ಡಿಮ್ಯಾಂಡಲ್ಲಿ ಇರೋ ಪೀಪಲ್ ಅಂತಂದರೆ ಫಾರ್ಮುಲೇಟರ್ಸ್. ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಮಾಡಿ. ಫಾರ್ಮುಲೇಟರ್ಸ್ ಮೀನ್ಸ್ ದೇ ನೋ ಕಂಬೈನಿಂಗ್ ದಿಸ್. ಇಟ್ ಈಸ್ ನಾಟ್ ತಗೊಂಡು ಸುಮ್ಮನೆ ಮಿಕ್ಸ್ ಮಾಡಿ ಕೊಟ್ಬಿಡೋದಲ್ಲ. ಏನು ಮಾಡ್ತೀರಾ, ನಿಮಗೆ ಒಂದು ಬಾಟ್ಲಲ್ಲಿ ಹಾಕಿ ಕೊಡ್ತಾರೆ ಇವೆಲ್ಲ. ನೀವೇನು ಮಾಡ್ತೀರಾ, ಒಂದು ಕ್ಯಾಪಲ್ಲೇ ಇಷ್ಟೇ ಹಾಕ್ಬಿಟ್ಟು ಮಿಕ್ಸ್ ಮಾಡಿ ಹೊಡಿತೀರಾ. ಬಟ್ ಆ್ಯಕ್ಚುಲಿ ಅವಾಗ ಕ್ಯಾಪಲ್ಲಿ ಹಾಕಿದಾಗ ಏನಾಗುತ್ತೆ ಅಂದರೆ, ಮೇ ಬಿ ಯು ಜಸ್ಟ್ ಯೂಸ್ಡ್ ಓನ್ಲಿ CBD ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್. ಈಗ ಉಳಿದಿದ್ದೆಲ್ಲ ಕೆಳಗಡೆ ಕೂತಿರುತ್ತೆ, ಕೈನೆಟಿನ್ ಕೆಳಗಡೆ ಕೂತಿರುತ್ತೆ, ಜಿಬರ್ಲಿಕ್ ಆ್ಯಸಿಡ್ ಮಧ್ಯದಲ್ಲಿರುತ್ತೆ. ಆಮೇಲೆ ಒಂದು ಕ್ಯಾಪ್ ಹಾಕಿದ್ನಪ್ಪ ಬಟ್ ಏನು ಆಗಲೇ ಇಲ್ಲ ಅಂತಾರೆ. ಮೇ ಬಿ ಈವನ್ ಮಿಕ್ಸಿಂಗ್ ಮಾಡಿದ್ರೂ ಅದು ಇದಾಗಲ್ಲ. ವಾಟ್ ಫಾರ್ಮುಲೇಟರ್ಸ್ ಡೂ ಈಸ್ ದೇ ವಿಲ್ ಕಂಬೈನ್ ಜಿಬರ್ಲಿಕ್ ಆ್ಯಸಿಡ್ ಸೇಮ್ ಮಾಲಿಕ್ಯುಲರ್ ಮಾಸು, ಇದನ್ನು ಸೇಮ್ ಮಾಲಿಕ್ಯುಲರ್ ಎಲ್ಲ ತೆಗೆದು ಹಾಕ್ತಾರೆ. ಹೆಂಗಿರುತ್ತಂದರೆ ಸೊಲ್ಯೂಷನಲ್ಲಿ ಈಕ್ವಲ್ ಆಗಿ ಡಿಸ್ಟ್ರಿಬ್ಯೂಟ್ ಆಗಿರುತ್ತೆ. ನೀವು ಶೇಕ್ ಮಾಡಲಿ ಬಿಡಲಿ, ದಟ್ ಈಸ್ ವೈ ಯು ನೀಡ್ ಗುಡ್ ಫಾರ್ಮುಲೇಟರ್ಸ್. ಅದನ್ನು ಇಲ್ಲ ನೀವು ಸರಿಯಾಗಿ ಶೇಕ್ ಮಾಡಿ ಮಾಡ್ಬೋದು. ಬಟ್ ಟ್ರೈ ದಿಸ್, ನೋಡೋಣ ಸರ್ ಯಾವುದಾದ್ರು ಪ್ರಾಡಕ್ಟ್ ಇದೆಯೇನೋ ಇದೆ ಅಂತ. ಅದು ಎಲ್ಲ ರಿಸರ್ಚ್ ಎಲ್ಲ ಟ್ರಾನ್ಸ್ಲೇಟ್ ಆಗಲ್ಲ ಪ್ರಾಡಕ್ಟ್ಗೆ. ಬಟ್ ರಿಸರ್ಚ್ ಹ್ಯಾಸ್ ಪ್ರೂವನ್. ನೀವೇ ತೊಗೊಂಬಂದು ಸರ್ ಅಮ್ಯುನೋ ಆ್ಯಸಿಡ್ಸು ಅಲ್ಲ, ಅದು ಬೇರೆ. ಇದರಲ್ಲಿ CBD ಎಕ್ಸ್ಟ್ರಾಕ್ಟು ನಿಮಗೆ. ಆಮೇಲೆ ಕೈನೆಟಿನ್ ಅಂತ ಪ್ರೋಟೀನ್ ಇದೆ, ಇದೇನಿದೆ ಇಲ್ಲಾಂದ್ರೂ ನಡೆಯುತ್ತೆ. ಬಟ್ ಅದಿಲ್ಲಾಂದ್ರೂ ನಡೆಯುತ್ತೆ. CBD ಎಕ್ಸ್ಟ್ರಾಕ್ಟು, ಜಿಬರ್ಲಿಕ್ ಆ್ಯಸಿಡ್, IAA ಅಥವಾ NAA ಆಗ್ಬೋದು. ನೀವು NAAನೂ ಆಗ್ಬೋದು. 3 ಹಾಕಿ ಸ್ಲೋ ಡೋಸಲ್ಲಿ ಕೊಡಿ. ಐ ಥಿಂಕ್ ದಿಸ್ ವಿಲ್ ಟೇಕ್ ಕೇರ್ ಆಫ್ ಅ ಲಾಟ್ ಆಫ್ ಸ್ಟ್ರೆಸ್ ರೆಸ್ಪಾನ್ಸಸ್ ಇನ್ ದಿಸ್ ಒನ್. ಆಮೇಲೆ ಇವರಿಗೆ ವೀಕ್ಲಿ ಡ್ರೆಂಚಿಂಗ್ ಆಫ್ ಕುಕುರ್ಬಿಟ್ಸ್. ಸರ್ ನಾನು ನಿಮಗೆ 20 ದಿವಸ ಅಂತಂದೆ, ಇಲ್ಲಿ ನೋಡಿ ನಾನು ರಿಸರ್ಚ್ ಎಲ್ಲ ವೀಕ್ಲಿ ಅಂತ ಹೇಳ್ತಾರೆ ಇವರು. ಎಷ್ಟು ಸರ್? 1.5 grams per liter ಅಷ್ಟೇ, ನೀವು ಜಾಸ್ತಿನೂ ಬೇಡ. ಸೊ ಈಗ ಕಮ್ಮಿ ಡೋಸು ರೆಗ್ಯುಲರ್ ಆಗಿ ಕೊಡಿ. ಸೊ ಇಫ್ ಯು ಯೂಸ್ ಈವನ್ 1.5 grams per liter ಆಫ್ ಅಮ್ಯುನಿಕ್ ಆ್ಯಸಿಡ್, ಈ ಫ್ರೂಟ್ ಲೆಂತು, ವೈನ್ ಲೆಂತು ಎಲ್ಲ ಇದಾಗುತ್ತೆ. ಅದಕ್ಕೆ ಕಮ್ಮಿ ಆದರೂ ಪರವಾಗಿಲ್ಲ, ಡ್ರಿಪ್ಪಲ್ಲಿ ಹಾಕ್ಬಿಡಿ ಸುಮ್ಮನೆ ಅಷ್ಟು. ಒಂದು ಒಳ್ಳೆ ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ತೊಗೊಂಡು ಮಾಡಿ. ಓಕೆ. ಸೊ ಸಮ್ ರೆಕಮೆಂಡೇಶನ್ಸ್ ಸರ್ ಫ್ರಮ್ ಮೈ ಸೈಡ್ ಅಬೌಟ್ ದಿಸ್ ಆ್ಯಂಡ್ ದಿಸ್. ಆ್ಯಂಡ್ ಫೈನಲಿ ಐ ಆಮ್ ಕನ್ಕ್ಲೂಡಿಂಗ್ ದಿಸ್ ಸೆಷನ್ ಬೈ some take home message. ಇಷ್ಟೆಲ್ಲ ಓದ್ ಮಾಡಿದ್ದೀವಿ. ಐ ಥಿಂಕ್ ಇಟ್ ವಾಸ್ ಅ ವೆರಿ hectic session. From 10 o'clock we have been talking. And so what I have is, sir, ಇದೊಂದು ಪಿರಮಿಡ್. when you use it ಇದು, ಸೊ ಬೇಸಲ್ಲಿ ನೀವು ಎವ್ರಿ ಟೈಮ್ ಪ್ಲಾಂಟ್ ಗ್ರೋತ್ ಪ್ರೊಮೋಟಿಂಗ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಮೈಕೋರೈಸಾ, ಫಂಗಸ್, ಹ್ಯಾವ್ ಇಟ್ ಆಲ್ ದ ಟೈಮ್. ನೀವು ಸೋಯಿಂಗ್ ಮಾಡ್ತೀರಲ್ಲ, ಸೋಯಿಂಗ್ ಮಾಡಿ ಸೀಡ್ಲಿಂಗ್ ಎಸ್ಟಾಬ್ಲಿಷ್ ಆದಮೇಲೆ ಡ್ರಿಪ್ ಪೈಪ್ ಹಾಕ್ತೀರಾ, ಡ್ರಿಪ್ ಪೈಪಲ್ಲಿ ಸೊಲ್ಯೂಷನ್ ಹಾಕ್ಬಿಡಿ, ಹೋಗ್ಬಿಟ್ಟು ರೂಟಲ್ಲಿ ಕುತ್ಕೊಳ್ಳಿ. ಮಲ್ಟಿಪ್ಲೈ ಆಗ್ತವೆ ಅವು. ದೇ ಮಲ್ಟಿಪ್ಲೈ, ಆಲ್ ಮೈಕ್ರೋರೈಸಾ ಮಲ್ಟಿಪ್ಲೈ, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮಲ್ಟಿಪ್ಲೈ, ಎವ್ರಿಥಿಂಗ್. ಸೊ ಡೋಂಟ್ ಸ್ಟಾಪ್. ಇಫ್ ಯು ವಾಂಟ್ ಟು ಹ್ಯಾವ್ ಎ ಗುಡ್ ಹೆಲ್ದಿ ರೂಟ್, ಪ್ಲೀಸ್, ದಟ್ ಈಸ್ ವೈ ಆಲ್ವೇಸ್ ಹ್ಯಾವ್ ಇಟ್ ಅಂತ ಹೇಳ್ತೀನಿ. ಬೇಸಲ್ಲಿ ದಟ್ ಈಸ್ ಹ್ಯಾವ್ ದಿಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಸೆಲೆಕ್ಟ್ ಫ್ರಮ್ ಗುಡ್ ಸಪ್ಲೈಯರ್ಸ್, take it, and I can help you to provide some companies. Second is regular intervals alli drench your soil with good quality humic acid and fulvic acid. Fulvic acid foliar ga haki idu haki. It is a good practice. Madbekantenalla, it is a good practice to have it. Second thing. Third one is, ನೀವು ಗ್ರೋ ಮಾಡಿದ ತಕ್ಷಣ 1 ತಿಂಗಳು ಆಯಿತು ಸರ್, ಪ್ಲೀಸ್, ಯು ಗೈಸ್ ಆರ್ ಆಲ್ ಅಗ್ರಾನಮಿಸ್ಟ್, ಮಾನಿಟರ್ ಯುವರ್ ಸ್ಟ್ರೆಸ್, ನೋ ವಾಟ್ ಈಸ್ ಕಾಸಿಂಗ್. ನೀವು ಬಂದು ಸುಮ್ಮನೆ ಬ್ಲೈಂಡಾಗಿ ಆಗ್ಬಿಟ್ಟು ಏನೋ ಆಯಿತು ಅಂದಾಗ ಬೇಡ. ಬಟ್ ನೋ ವೆದರ್ ಇಟ್ ಈಸ್ ಟೆಂಪ್ರೇಚರ್ ಜಾಸ್ತಿ ಆಗ್ತಾ ಇದೆಯಾ, ಇಲ್ಲ ವಾಟರ್ ಜಾಸ್ತಿ ಇದಾಗ್ತದೆ, ಸೋಡಿಯಂ ಜಾಸ್ತಿ ಸಾಲ್ಟ್ ಇದೆಯಾ. ಯು ಪ್ಲೀಸ್ ಪುಟ್ ಯುವರ್, ವೆನ್ ನೋ ಯುವರ್ ಸ್ಟ್ರೆಸ್ ಆ್ಯಂಡ್ ದೆನ್ ಅಪ್ಲೈ ದಿ ಇದು ಏನೋ ಬಯೋಸ್ಟಿಮ್ಯುಲೆಂಟ್. ಕರೆಕ್ಟ್ಲಿ ಮ್ಯಾಚ್ ಆಗಬೇಕು, ಅದು ಮ್ಯಾಚ್ ಆಗೋಂಥ ಬಯೋಸ್ಟಿಮ್ಯುಲೆಂಟ್ ಆಗಬೇಕು. ಸೊ ಅದಕ್ಕೇನು ಮಾಡಬೇಕು ಅಂತಂದರೆ, ನೀವು ಕ್ರಿಟಿಕಲ್ ಸ್ಟೇಜ್ನ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮಾಡ್ಕೋಬೇಕು, ಹಿಸ್ಟಾರಿಕಲ್ ಡೇಟಾ ನೋಡಬೇಕು. ಎವ್ರಿ ಇಯರ್ ಈ ಟೈಮಲ್ಲಿ ಹಿಂಗೆ ಆಗೇ ಆಗುತ್ತಪ್ಪ, ಸ್ವಲ್ಪ ಫ್ರೂಟ್ ಡ್ರಾಪ್ ಆಗುತ್ತೆ. ಸೊ ದೆನ್ ಯು ಹ್ಯಾವ್ ಸಮಥಿಂಗ್ ಲೈಕ್ ದ್ಯಾಟ್. ಡಿಸ್ಟಿಂಗ್ವಿಷ್ ಬಿಟ್ವೀನ್ ನ್ಯೂಟ್ರಿಯನ್ ಡೆಫಿಷಿಯನ್ಸಿ ಆ್ಯಂಡ್ ಸ್ಟ್ರೆಸ್. ದಿಸ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಮೆನಿ ಪೀಪಲ್ ಡೋಂಟ್ ನೋ ಇದು ಸ್ಟ್ರೆಸ್ಸಿಂದಾಗಿರೋದಾ ಇಲ್ಲ ಇದು ಪೊಟ್ಯಾಷಿಯಮ್ ಡೆಫಿಷಿಯನ್ಸಿನ ಗೊತ್ತಿಲ್ಲ. ಸೊ ಆ್ಯಂಡ್ ದೆನ್ ಯುವರ್ ಪೊಟ್ಯಾಷಿಯಮ್ ಡೆಫಿಷಿಯನ್ಸಿ ಇರುತ್ತೆ. ನಾವಿಲ್ಲಿ ಏನೋ ಬೇರೆ ಎಲ್ಲ ಸ್ಟ್ರೆಸ್ ಇದು ಕೊಟ್ಟು ವಿ ಆರ್ ವೇಸ್ಟಿಂಗ್ ದ ಟೈಮ್. ಐ ಥಿಂಕ್ ವಿ ಶುಡ್ ನೋ ಎಕ್ಸಾಕ್ಟ್ಲಿ ದಿಸ್ ಇಸ್ ಪೊಟ್ಯಾಷಿಯಮ್ ಓನ್ಲಿ. then you can tailor something like that. ಆಯಿತಾ? ಅದಾದಮೇಲೆ ಫ್ಲವರಿಂಗ್ ಸ್ಟೇಜಲ್ಲಿ ರೈಟ್ ಪ್ಲಾಂಟ್ ಹಾರ್ಮೋನ್ಸ್ ಟು ಇಂಪ್ರೂವ್ ಯುವರ್ ಫ್ರೈಡ್ ಆ್ಯಂಡ್ ಫ್ರೂಟ್. ನೌ ಐ ಆಮ್ ಸಜೆಸ್ಟಿಂಗ್ ಒನ್ ಮೋರ್ ಥಿಂಗ್, ನಾಟ್ ಜಸ್ಟ್ ಫೈಟೋ ಹಾರ್ಮೋನ್. ರೈಟ್ ಫ್ಲವರಿಂಗ್ ಸ್ಟೇಜ್ ಅಂದರೆ ಇನಿಷಿಯೇಷನ್ ಫ್ಲವರಿಂಗ್ ಸ್ಟೇಜಲ್ಲಿ ಕಂಬೈನ್ ಎ ಗುಡ್ ಸೀವೀಡ್, ಗುಡ್ ಅಮೈನೋ ಆ್ಯಸಿಡ್, ಕಾಂಬಿನೇಷನ್ ಆಫ್ 2 ಫೈಟೋ ಹಾರ್ಮೋನ್ಸ್ ಲೈಕ್ ಲೋ ಡೋಸ್ ಆಫ್ ಇಂಡೋಲ್ ಅಸಿಟಿಕ್ ಆ್ಯಸಿಡ್ ಆ್ಯಂಡ್ ಜಿಬರ್ಲಿಕ್ ಆ್ಯಸಿಡ್ ಹಾಕಿ much more broader support sigutte nimage. So much more broader than having a very, very that is one recommendation I can give you. And flowering stage treatment is a must. Though it is, I did not put here, don't miss out treatment on flower. Flower is your yield. So don't miss out on the flowering stage recommendation. And finally, I think you should all, on the istotike ಒಂದು ಜರ್ನಲ್ ಇರಬೇಕು ನಿಮ್ಮ ಹತ್ರ. ಎಷ್ಟು ವರ್ಷ ಇದ್ದೀರಿ ಸರ್ ಗರ್ಕಿನ್ ಬಿಸ್ನೆಸ್ ಅಲ್ಲಿ? 20 years 20 Okay, 30 years. 33 years ಅಲ್ಲಿ ವೆಲ್ತ್ ಆಫ್ ಇನ್ಫಾರ್ಮೇಷನ್ ಇದೆ ನಿಮ್ಮ ಹತ್ರ. ಯಾವ ಇಯರಲ್ಲಿ ಇದಾಯಿತು, ಸ್ಟ್ರೆಸ್ ಆಯಿತು, ಇದಾಯಿತು. ಮೇ ಬಿ ಯು ಹ್ಯಾವ್ ಲೈಕ್ ಎ, ಇಫ್ ಯು ಹ್ಯಾವ್ ಜರ್ನಲ್ಡ್ ಇಟ್ ಟುಡೇ, ಯು ವಿಲ್ ನೋ, ಓಕೆ ಇಂಥ ಕಂಡೀಷನ್ನಲ್ಲಿ ಇದಾಗುತ್ತೆ, ತಮಿಳ್ನಾಡಲ್ಲಿ ಪಾಸಿಟಿವ್ಸ್ ಏನಿದೆ, ನೆಗೆಟಿವ್ಸ್ ಇಲ್ಲೇನಿದೆ. ಸೊ ಮೇ ಬಿ ಐ ಆಮ್ ಜಸ್ಟ್ ರೆಕಮೆಂಡಿಂಗ್ ಇಟ್. ಇಟ್ ವಿಲ್ ಆ್ಯಡ್ ಟು ದ ನ್ಯೂ ರೆಕಮೆಂಡೇಷನ್ಸ್, ನ್ಯೂ ಪ್ರಾಕ್ಟೀಸಸ್. ದಟ್ ಇಸ್ ವಾಟ್ ವಿ ಕಾಲ್ ಅಪ್ಲಿಕೇಶನ್ ಬುಕ್ to have it, because it's a serious business. It's a crores of a business here, and all your careers are here, futures are there. India can become a top Gherkin thing. So this is my little contribution to this one. So I hope I have a finally what we can quiz. Nodi, we started with this stage planned yield influenced by stress and phytohormones, bio-stimulant. They both will work in tandem. It is not like adon madadre sakidu alla. And identifying the right combination, manage these two, you are in a good shape to manage this one. So that's all I had, and I hope you are educated a little bit. We'll have more discussion after lunch. We'll have some panel discussion. Lakshmi, you can tell me if you have more questions. We can address it later on, but that concludes my main presentation. This one.
Okay, thank you. I think we can take a lot of questions. Lunch break now, so we are going to reassemble by 2:30. And ನಿಮ್ಮ ಡ್ರೈವರ್ಸ್ಗಳಿಗೂ we can ask them to come for the lunch ಕೆಳಗಡೆ. You please inform them for the lunch. Lunch organized outside. Lunch is organized outside. Buffet.
Hello? 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 Yes. Hello? Hello?
ಇದು ಕ್ವಶನ್ಸ್ ಯಾರ ಹತ್ರ ಇದ್ದರೆ ಯು ಪ್ಲೀಸ್ ಗಿವ್ ಮೀ. ಸರ್ ಕಲೆಕ್ಟ್ ಮಾಡ್ಬಿಡಿ ಹೆಂಗೆ ಯಾರೋ somebody should collect this. ಪೇಪರ್ ಇತ್ತಲ್ಲ
ಪೇಪರ್ ಇಲ್ಲ ಇತ್ತು ಸರ್, ನೀವು ಎತ್ಕೊಂಡು
ಎತ್ಕೊಂಡಲ್ಲ. After this lunch, session 3, third session ಅಲ್ಲಿ group discussion on remaining unaddressed topics, strategies to enhance the use of biostimulants, possible collaboration to support Gherkin growers, and finalizing key focus areas for next season. This is the topic for now. To request, yes, I request a pathologist, entomologist, Dr. Nagaraj, Pradeep Sajjan, Narsimurthy from the NAMS, Rakshak Gouda from the NAMS, Regs 1 representative, and Ramesh sir, Vinod sir. To mentor ಇದು ಈಗ ಪ್ಯಾನಲ್ ಡಿಸ್ಕಷನ್ ಅಂದರೆ ಪ್ಲೀಸ್ ಕಮ್ ಫಾರ್ವರ್ಡ್. Any agronomist, entomologist, please. Technical, Nagaraj, neophytes, entomologist, pathologist, experts. You can come front. Okay. ಕೆಲಸ ನೀವು ಸ್ವಲ್ಪ ಶಿಫ್ಟ್ ಆಗ್ಬಿಡಿ ಈ ಕಡೆಗೆ. ಈ ಟೇಬಲ್ ಬೇಕು, ನೀವು ಸ್ವಲ್ಪ ಸಪೋರ್ಟ್ ಮಾಡೋದು ನಾವು ಎತ್ಕೊಂಡು ಹಾಂ ಎತ್ಕೊಳ್ಳಿ. Please come forward. There is a. One table is enough, and you can chairs ಬೇಕು. Chairs ತನ್ನಿ please. ಬನ್ನಿ. Ramesh sir. ಬನ್ನಿ please. You also come sir. Being a horticulturist, you should be here. ಅಲ್ಲ ಅವರು ಬೇಕು ಸರ್, ನೀವು ಬೇಕು. ಸರ್. And this is an interactive session. It's overall topic. ನಿಮ್ಗೇನು ಅನ್ಅಡ್ರೆಸ್ಡ್ ನಿಮ್ಗೇನು ಅರ್ಥ ಆಗಿಲ್ಲ ಆ ಟಾಪಿಕ್ನ, ಎನಿಥಿಂಗ್ ರಿಲೇಟೆಡ್ ಟು ಎಂಟಮಾಲಜಿ, ಪೆಥಾಲಜಿ, ಸಾಯಿಲ್ ಸೈನ್ಸ್, ನ್ಯೂಟ್ರಿಷನ್ ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಫಾರ್ ಬಯೋಸ್ಟಿಮ್ಯುಲೆಂಟ್ಸ್, ಇದ್ರ ಬಗ್ಗೆ ಎನಿ ಕ್ವೆಶ್ಚನ್ ಕ್ಯಾನ್ ಬಿ ಥ್ರೋನ್. ಫಸ್ಟ್ ಪ್ರಿಫರೆನ್ಸ್ ಈಸ್ ಟು ವೆದರ್ ಟು ಟುಡೇಸ್ ಟಾಪಿಕ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಆಗಿದೆಯಾ ಅಲ್ವಾ, ಅದರಲ್ಲಿ ನಿಮ್ಗೆ ಏನಾದ್ರು ಡೌಟ್ಸ್ ಇದ್ದರೆ ದಯಮಾಡಿ ವಿ ಕ್ಯಾನ್ ಪ್ರಶ್ನೆ ಕೇಳ್ಬೋದು. Nitrobenzene. Something about nitrobenzene? No. These questions, anybody have question written?
ಹೌದಾ? ಓಕೆ. ಇಲ್ಲ ಸರ್, ಕೇಳಿದ್ದೀನಿ, ಬಟ್ ಐ ಡೋಂಟ್ ನೋ ದ ರಿಯಲ್ ಮೋಡ್ ಆಫ್ ಆಕ್ಷನ್. ಬಟ್ ಎನಿಬಡಿ ನೋಸ್?
ಅಲ್ಲ, ಈಗ ಪ್ರಶ್ನೆಗಳು ಇದ್ದರೆ ಬರೆದಿದ್ರೆ ಕೊಡಿ. ತಿಳ್ಕಾರ್ ಈಗ any written question is there, please hand over. We can address these topics. First we will take up these topics and then we will continue further. I will take up this first question. This is to CBG, Dr. CBG. Can we produce humic acid by doing decomposed? If yes, then how many days we need to maintain the decomposed?
No, not possible. You cannot make humic acid from decomposition of any organic matter or something. It is naturally found. This one.
Sir, adding to this point, before joining to Gherkin industry, I was worked in the company called PJ Margo. They are producing the humic acid under the brand name of EcoHume. They are producing out of this vermicompost only by hydrating with this potassium hydroxide, and initially they are producing with NaOH based humic acid. Later, upon to improve the humic acid efficacy, they are producing now with the potash based humic acid, but exclusively with the vermicompost only. With the 4% and 8% labeling.
No, you can modify humic acid. Sir? You can modify by adding potassium and all, but humic acid is always mined from the soil, is what I'm saying. Not synthesized. Okay, it is just like petrol, you have to mine it from that.
But I don't know, sir, how they are
selling.
No. And
once you take the humic acid, you can make potassium humate. You can make monopotassium humate. You can add functional group and make a new product. But basic humic acid you cannot make at home, is what I'm saying. You cannot make. Once you take it, you can modify it. You can do a lot of chemical modification.
Hope this will answer your question. Okay. Okay. I will take up next question again for you. Which cell-free extract we can use for reduce the fungal infection in gherkins? If yes, which time we recommend?
For fungal infection, you are saying? Okay. Actually, it's a good question, in my opinion, because you are really thinking very good. Whose question is this?
Tilakaran.
Very good. I think it's a very cheap way of controlling plant diseases by doing this one. If you ask me which cell-free extract is good to reduce fungal infection, there is no simple answer for that. But any bacterial isolate which is having more metabolites, you should have like as many metabolites as possible, it is good. So in this case, what we can do is, you cannot get cell-free extract from fungus. You can get only from bacteria. So in this case, it is good to identify 3 or 4 bacteria and combine it rather than single bacteria. So it means like Bacillus subtilis, Bacillus amyloliquefaciens, and one more thing. These are the 2 things. And in that, you can have 2 or 3 species from Bacillus subtilis, 2 or 3 species from amyloliquefaciens, because when you grow all of them in different tubes like this, everybody is producing a different blend of metabolite. So you don't know which is potent, which is not potent. So more is good. So you should combine 2 or 3 bacterial cell-free extract in order to get any good fungal infection. Please note that there are 2 ways that fungal infection can be reduced here. One is these what we call phytoalexin, phytoremediation products. They will directly inhibit the fungus not to grow, because they are very like antibiotics, extracted like antibiotics. The repellent, they go. Second is, some of these cell-free extract will trigger the defense in the leaves. They make the leaves to produce what you call defense proteins, and they are primed. When the downy mildew attack happens, already there is defense in this way. So 2 ways. One is direct inhibition, second one is helping the plant to produce some defense molecules here. And your answer is just we should combine 2 or 3 bacterial isolates together and do it. Most of the products are not readily available in the market, but we should do it we should combine, not than one.
Anything you want to add, pathologist, to this? Any points you want to add?
Whether such products are available in India?
In India, it is very difficult. I don't know any company who is making hydrolysates here, but it is very easy to make cell-free hydrolysates. It is very easy. Only thing is that you don't need even fermenters. You need like 50 liters conical flask and one shaker. You add LB media, and then the cells are growing there. After that, you need one centrifuge. All the cell is done, and then the liquid remaining. Liquid, you don't do any alteration. Just mix it in the tanker, add some surfactant, spray on the leaves. That's enough.
Sir, but okay, what you told.
I don't know why companies are not doing it. I don't know.
Some of the the organisms may be harmful.
No. We are not using. We will use only organisms from IHR, for example.
Okay, already known.
Yes.
That we have to use.
Exactly.
Yeah.
Okay. Now I triggered. So you can go to IHR, Indian Institute of Horticulture Research. They have some collection of I don't know 50 or 60 known classified harmless bacteria. And you can also make contract with IHR. They will provide you these cans. They will provide you cans that you can use this one. GKVK also does this. In the microbiology department, they are regularly providing these cell-free extracts. So you should convert. The agriculture universities are the only one who are doing these cell-free extracts now, and they are doing it in a very crude manner. But it's okay. It is not going to damage your plants. They are not pathogen. They are all good bacteria that they are doing. So I would strongly recommend go to IHR or Agriculture University in GKVK. I know in Tamil Nadu also they might be having, and they will give you cans of 20 liter, 50 liters cans with this one.
With all dosage and other things they will give, or just direct?
They will give everything.
They will give
exactly. Yeah. You can also try the AMC, which is called Arca Microbial Consortia, which is having gram-positive and gram-negative. It is having Bacillus species, 2 Bacillus species, 2 Pseudomonas species. To come to your answer, that cell-free extract, people are using in pomegranates, fruit crops, cucumbers, everywhere. That is very, very effective, that cell-free extract. So you can, I can give you contact. Please contact me later after that, and I will give. And there is also one company, one of my junior is running. It's called Aracle Organics. He's doing only IHR techniques he is also fermenting it, and he is giving cell-free extract, cell and cell-free extract.
Head of extension, a very senior scientist called Balkrishna. Very, very nice guy. He is my classmate and a very friendly person. ಯಾರೇ ಹೋದ್ರು ಊಟ ಮಾಡಿ ಕಳಿಸ್ತಾರೆ. ಯಾವ ಗೌರ್ಮೆಂಟ್ ಆರ್ಗನೈಸೇಷನ್ ಆ ಥರ ಮಾಡಲ್ಲ. So he'll be overwhelmed if anything required for farmers. Please contact him. Balkrishna at IIHR, Director or Head of Extension. He controls everything.
Yes, I think IHR and Gherkin, because the horticulture crops, so they are the best ones to contact.
What is the interaction of cell-free extracts with fungicides or insecticides?
100% compatible. There is no way that, see, first you should understand how fungicides work. Fungicides work either by blocking some membrane transporters in fungus, mode of action, or they work with, what else pathology you can tell me. So they attack only those components that is required to control the reproduction of fungus. Okay. Yeah.
They will work on the mycelial growth restrictions.
Exactly. So when you use bacterial cell extracts, they are growth promoters basically. So there is no reason to believe that fungicides will act on this. Fungicides are very specific, na? They are made only to act on what these guys are telling. So it
Because
should be
why I am asking in Gherkins, we cannot go for individual spray. Generally, it will be combined with any like nutrients or fungicides.
Yes.
So that's why I am asking. Efficacy will be not reduced, or it will be same as.
Okay. See, that's what I said. Compatibility issue, I'm not having doubt, but I have to clarify one thing here. These cell-free extracts should not be used to control something, to prevent. These are all preventive, not prophylactic. That means you cannot say cell-free extract after the fungal is infected. No, it will not work. Then yes. So because if you are anticipating, okay, now there is some cloudy weather, the forecast says some, what is this, cyclone is coming, we will have some gloomy weather. You prepare the plants. Like I said, my gun example I gave now. So you are standing there with the gun when you add cell-free extract, rather than going and finding the gun. So that is, you prepare it. And that when the real fungus attacks, maybe you are spraying only 1/10 of the fungicide when you normally spray. Or the disease is completely controlled without spraying also. But no microbial extracts can control the disease. They are only preventive.
Maybe in a simple way, need to add to this. I think presently you are all using like this pasparic acid, PK40. So what exactly it will do? It will improve the leaf thickness and hairiness of the leaves. So that indirectly we are postponing the downy mildew. So like that way, what sir is also telling, this cell-free extract will also act like that.
Okay, that you answered your question, I think. So next question is online. What time and temperature is good to spray biostimulants?
I think like anything, it should be suboptimal temperature. If it is, it's just, I think we haven't really talked of that one, but it should be suboptimal or lower temperatures where the adhesion or absorption is maximum. I don't know, I can leave it to people who are experts, they know individual crops and everything. The maximum thing is it should absorb maximum, or if it is sprayed to the soil, it should, the soil should not be too warm to degrade the metabolites. That's
correct. Generally, physiological activity of the plants always better in the morning hours and the evening hours.
Hours, huh?
Yes.
Yeah, then it is
that is the time where we can go
yes,
for
yes.
this application. So
yeah,
for
okay. Then I understood. Yes, morning and evening is always better.
Always better.
Yes.
Sir, my one doubt about the calcium nutrient. Okay. Many says that calcium is highly immobile in plant system. So how you can make improve the efficiency of this calcium element? One of the session you told about we can increase by combining with acids like humic acid or audience asked question it is, I don't know. And also some of the companies coming with calcium nutrient along with gluconic acid. Whether it's really working or not, I don't know. Whether you have some information on that.
Your question has the answer. Your question has answer.
No,
What did you say?
I
Calcium and what is that?
Gluconic acid.
What is gluconic acid? It's an organic acid.
Okay.
So that whenever you have calcium in an organic acid, the absorption, dissolution, uptake is much easy.
That I don't know.
Okay, that is why they're combining with organic acids.
Okay, okay.
All
The efficiency of this
all
calcium will be more.
So you see in the root zone, in the plant, there are roots here. And if there are organic acids, you can name organic acids like so many: citric acid, citrullic acid, gluconic acid, and so many organic acids which are produced organically, not inorganically. The elements, nutrient elements, dissolve very well. Absorption is very, very well. In this
So
way.
better we use calcium nutrient with some organic acids,
Yeah,
like
exactly.
gluconic acid or
Especially-
humic acid like that.
Which one? Humic acid is also an organic acid. Humic acid, fulvic acid, uriate, citric. Krebs cycle anta barutta sir. Krebs cycle alda era barodu.
Citric acid.
Anything from organic source alli, if it is having acidic nature, is organic thing.
Because many field staff will tell that I am applying so much of CN, the fruit size or the shape is not improving. The calcium use efficiency should be more, or it has to be
Yes. Not only calcium, phosphorus, potassium, I think magnesium. Many elements are very, they become ions and they get fixed in the soil.
Okay.
So it is like what you say. You have money in your locker, but you don't know the key. You don't know the password to open it. So that is where organic acids produce. And when you treat lot of PGRs, which colonize the root, fungus, bacteria, what they are doing essentially, every day they are producing organic acids. So wherever there is root, there is like a full organic acid is there. In that pool, nutrients are much easily dissolved, and root hairs will absorb everything inside. But without the organic acid pool around the root, even if you have 10 times more nutrients, absorption is not happening. So you have to create an organic acidic environment for nutrients to absorb here. So I think this question has an answer here. Combine it with organic acids.
You mean to say at least the acidic soils are much better in nutrient absorption? Is it similar to that?
I think acidic soil is always better in many nutrient absorption. Yeah. See, the 4 to 5.5 is the best range where maximum nutrients are absorbed.
Correct.
Micronutrients, yes. That's what I said. 4 to close to 6 is where you see the maximum nutrient absorption. And you can make this pH by using organic acids.
Organic acids. Yeah. And the one more question. Biostimulants for below things. Gherkin color, fungal disease resistance, skin thickness, biostimulants can be used.
Gherkin color, fungal disease. Okay. For Gherkin colors, I think ascophyllum seaweed extract, protein hydrolysate combined. You can use before the start of flowering and regular spray. I think 20 days before flower initiation. You can try that one, ascophyllum and fulvic acid probably little bit, and protein hydrolysates, because seaweed extract will mobilize all the flower color, flavor color pathways, and Protein hydrolyze will mobilize nitrogen and other nutrient into this one. And what was the other one I said? Even fulvic acid. Fulvic acid is generally, it will trigger the calcium pathway, which is again required for colors and flavors. So I think for Gherkin colors, better to combine dose of seaweed extracts, amino acids and fulvic acid. For fungal disease resistance in foliar, I am assuming. Foliar, it is? It is, okay, they didn't say anything. Resistance is, you can spray, they usually spray some hormones like salicylic acid. Salicylic acid can be sprayed, but I don't know, I have to see if there is any literature support on that. Cell-free extracts are absolutely fantastic. So if you get this IHR cell-free extracts, and if you think that this is the right stage or stage that is susceptible for fungal disease, keep on spraying that cell-free extract onto the plant. And please note that you have to spray on the top, you have to spray also on the bottom of the leaves, so that maximum coverage is happening. And these metabolites are covered with some surfactant or something. So that you can do, or you can also spray Pseudomonas or IHR also has a lot of foliar bio-stimulant, bacteria. You can spray that also. That is already given. The shelf life will be very short, what they give, 3 months, 4 months, but you can spray that into that thing. Skin thickness and shelf life. Skin thickness, this is more nutrient management. I don't know if I can maybe biostimulants can help, but I don't have any data on this one. Shelf life, I don't know, biostimulants can help it, but it is more nutrient management and water management, I would say. Harvest time, physiological harvest time.
Understood? Any doubt in this? Yaar question kedidri idu? You understood? Okay, clear. Next question. Is it possible to grow seaweed in artificial ponds like Azolla?
Possible, but you won't get good quality. It is adapted to sea. It is coming from the sea. So you have to mimic the sea exactly. So my answer is no, that's not possible. Otherwise people would have gone. So, yes, there is freshwater seaweed also. Freshwater seaweed also. But for agricultural this thing, seaweed is the most potent. Freshwater seaweeds are also there. But this is very good. My simple answer is no, you cannot grow the natural seaweed that you find in the oceans in this one. It's not possible. Many factors. Quality, sunlight, absorb nutrients, even wastes. I don't know. So much wastage also goes in there, also absorbing it, and no, it is not possible to grow this seaweeds.
Whatever you are expecting from the seaweed, exactly the seed, that is, it is not there, so artificially grown.
No, it's not possible. It is very, very adapted species to seawater. It will not, it will not grow here.
Hope understood this? Clear? Okay. ಅಮಿನೋ ಆಸಿಡ್ ಹೆಚ್ಚು ಸಿಂಪರಣೆ ಆದರೆ ಗಿಡಗಳಿಗೆ ಹೆಚ್ಚು ತೊಂದರೆ ಇದೆಯಾ? ನೈಟ್ರೋಬೆಂಜೇಟ್ ಇದರ ಬಗ್ಗೆ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿ.
ನೈಟ್ರೋಬೆಂಜೇಟ್ ಅಷ್ಟೊಂದು ಮಾಹಿತಿ ಇಲ್ಲ ನನಗೆ, ಬಟ್ ಮೇಲಿನ ಅಮಿನೋ ಆಸಿಡ್ ಹೆಚ್ಚು ಇಲ್ಲ, ಆ ಥರ ಎಲ್ಲೂ ಇಲ್ಲ, ಆ ಥರ ಏನೂ ಇಲ್ಲ. ಸುಮ್ಮನೆ ಅದು ಹಣ ವೇಸ್ಟ್ ಮಾಡ್ದಂಗೆ ಅಷ್ಟೇ. ಅದು ನೀವು ಎಲ್ಲದಕ್ಕೂ, ಯಾಕಂದರೆ ಪ್ಲ್ಯಾಂಟು ಎರಡು ಥರ ಇರುತ್ತೆ. ಒಂದು ವೆನ್ ಯು ಸ್ಪ್ರೇ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಸಪೋರ್ಟಿವ್, ದೇರ್ ಆರ್ ಟೂ ಥಿಂಗ್ಸ್ ಹ್ಯಾಪನಿಂಗ್. ಒನ್ ಈಸ್ ಇಟ್ ಈಸ್ ಸೆನ್ಸಿಂಗ್. ಇದು ಪ್ರೋಟೀನು ಸೆನ್ಸ್ ಮಾಡಿಬಿಟ್ಟು ನೈಟ್ರೋಜನ್ ಅಸಿಮ್ಯುಲೇಷನ್ ಏನೇನು ಮಾಡಬೇಕು ಅನ್ನೋ ಅದು ಆ ಪಾತ್ವೇ ಟ್ರಿಗರ್ ಮಾಡಿ ಇಟ್ ವಿಲ್ ಯುಟಿಲೈಸ್ ನೈಟ್ರೋಜನ್ ಬೆಟರ್. ಅದೊಂದು ಪಾತ್ವೇ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ ಒಂದು. ಎರಡನೇದು ಆಕ್ಟಿವ್ ಅಬ್ಸಾರ್ಪ್ಷನ್. ಅಮೈನೋ ಆ್ಯಸಿಡ್ಸ್ are absorbed into the tissue. They are used to build proteins. in the leaf. ಜಸ್ಟ್ ಈಗ ನಾವು ಏನು ವೇ ಪ್ರೋಟೀನ್ ಕುಡಿತೀವಿ ಇದರಲ್ಲಿ, ಕುಡಿದಾಗ ಏನಾಗುತ್ತೆ, ನಾವು ಪ್ರೋಟೀನ್ ಅಂತೀವಿ, ಬಟ್ ವಿ ಆರ್ ನಾಟ್ ಡ್ರಿಂಕಿಂಗ್ ಪ್ರೋಟೀನ್, ವಿ ಆರ್ ಡ್ರಿಂಕಿಂಗ್ ಅಮೈನೋ ಆ್ಯಸಿಡ್ಸ್. And that amino acid is used in our body to make protein, muscle protein, skin protein, hair protein, ಆಮೇಲೆ ಎಲ್ಲಿ ಏನಾರು ಬೇಕು ಆ ಪ್ರೋಟೀನ್ ಮಾಡೋದಕ್ಕೆ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಯೂಸ್ ಮಾಡುತ್ತೆ. ಸೇಮ್ ಪ್ಲ್ಯಾಂಟಲ್ಲೂ ಅಷ್ಟೇ. ಸೊ ಈ ಪ್ರೋಟೀನ್ ಡಿಫಿಷಿಯೆಂಟ್ ಇತ್ತು, ಏನೋ ಒಂದು ಸ್ಟ್ರೆಸ್ ಬಂತು, ಯು ವಿ ಸ್ಟ್ರೆಸ್, ಲಾಟ್ ಆಫ್ ಪ್ರೋಟೀನ್ ಇಸ್ ಬಿನ್ ಡ್ಯಾಮೇಜ್ಡ್. ಇನ್ ದಟ್ ಕೇಸ್ ಯು ಸ್ಪ್ರೇ ಅಮೈನೋ ಆ್ಯಸಿಡ್ಸ್, ದೇ ಆರ್ ಆ್ಯಕ್ಟಿವ್ಲಿ ಅಬ್ಸಾರ್ಬ್ಡ್, ಪ್ರೋಟೀನ್ ರಿಪೇರ್ ಆಗುತ್ತೆ. ಸ್ಕಿನ್ ಇದು ಲೀಫೆಲ್ಲ ಲೀಫ್ ಟೇರ್ ಆಗಿರೋದು ಅಥವಾ ಇದು ಅದೆಲ್ಲ ಸ್ವಲ್ಪ ದೇ ವಿಲ್ ರಿಕವರ್. ಸೊ ದೇರ್ ಆರ್ 2 pathways. ಆ್ಯಂಡ್ ತುಂಬ ಪ್ರೋಟೀನ್ ಡ್ಯಾಮೇಜ್ ಆದಾಗ ಮೇ ಬಿ ಯು ಕ್ಯಾನ್ ಇನ್ಕ್ರೀಸ್ ಅ ಲಿಟಲ್ ಬಿಟ್ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಕಾನ್ಸಂಟ್ರೇಷನ್, that might help in repair. But when it comes to sensing amino acid and mobilizing nitrogen cycle in the plant, you don't need so much. ಸ್ವಲ್ಪ ಆದರೆ ಸಾಕು, ಸಿಗ್ನಲ್ಗೆ ಎಷ್ಟು ಬೇಕೋ ಅಷ್ಟು ಸಿಕ್ಕರೆ ಸಾಕು. ಅದಕ್ಕೆ
ಉತ್ತರ ಸಿಕ್ತು ಅಂತಿದ್ದೀನಿ. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶನ್. ವಾಟ್ ಈಸ್ ದಿ ರೋಲ್ ಆಫ್ ಪಂಚಕವ್ಯ ಅಂಡ್ ಟೆಂಡರ್ ಕೋಕೋನಟ್ ವಾಟರ್ ಇನ್ ಗ್ರೋತ್ ರೆಗ್ಯುಲೇಟರ್ಸ್? It is very difficult to answer. No research findings, I don't think so. All the questions have been answered from the...
See, this you were mentioning in criteria 1, apart from this helping with the plants, so you also mentioned that soil properties. So this biostimulant, how it helps in improving the soil properties? Now, as it is, what we are doing is only we are only chemical properties. So we are adding inorganic fertilizers into the soil. So will it improve on physical and biological properties?
Only, yes. Not all biostimulants will help in soil properties.
Okay, it was mentioned in your this thing.
No, only humic acids. Means that is one of the, that means you should choose which biostimulant will help you. See, you have 10 criteria. One is nutrient efficiency, fruit quality.
Yeah, it's all with the plants.
Yes.
So I'm only with the soil. So you mentioned that soil properties in the last.
Yeah, know your stress. That's what I'm saying.
Yeah, so maybe whatever it is.
That physical. Is humic acid mode of action, sir. What I showed with that clay was humic acid mode of action.
It will be like a soil conditioner.
Yes, repeat your question, sir. Maybe I didn't understand it.
See, you mentioned that soil properties, will it impact on the soil properties?
Yes.
See, like, now it is, anyway, it's stress of this, stress and nutritional balance will be controlled by this biostimulant. So will this also help in getting the soil properties?
Yes, but not all, what do you call, biostimulant. Microbial, what is it, PGR, plant growth promoting microbes will help. Humic acids will help. That's all. These are the only 2. And if you see how they help, 2 different mechanisms. The humic acid is physical. Completely, there is no physical and little bit of chemical. That is fragmenting the clay particles by embedding in between. If there is one clay particle like this, and another clay particle, both are bound like this completely, humic acid will go in between this.
Okay.
And it is like it is creating a gap. Now it is, and then this thick clump will become fragmented because interspersed. Humic acid will intersperse here and there. Now there is aeration. Macropores created. Water will go. Roots will grow very well in between these pores. This is what is doing.
It will increase.
No, apart from this foliar or through fertigation, suppose if you do it like soil application kind of
soil
thing.
application. That's why I said humic acid, general rule. Humic acid soil, fulvic acid for foliar.
Okay. So that is more
yes,
effective.
because fulvic acid is more reactive. It is having smaller molecular weight, and it has more signaling responses. When you put it in the leaf, plant sense the fulvic acid very well, and they create lot of all the calcium cycles and everything. But humic acid is not good at doing that one. Humic acid is good in improving the soil structure. So you put it into the soil structure.
Okay. One more is you were mentioning that humic acid, fulvic acid, and humin.
Humin, yeah.
What is the role of that humin?
Humin is like a more heavier than the humic acid, and where you have very problematic soils.
Yeah. So wherever problematic soil is there,
Very
we have to
yeah,
use
humin,
this.
yeah, but humin is not very
Yeah,
commonly available. Commonly available.
I also heard newly this word.
No, they are very not commonly available. It's a very heavy molecule. It is not economically so important, but it is humic acid and fractions.
Okay.
When you take the humic acid fraction, it's like you put it in a sieve, and the first you get is heavy humin, then you get humic acid, then you get fulvic acid, like that.
Okay. One more last question. Potassium skinate is used as a biostimulant?
Which one?
Skinate. Potassium skinate. Cyanate. So we have to pronounce like cyanate. Skinate or cyanate? So you were mentioning potassium sulfate.
But see, anything is a biostimulant. Anything, as long as either it is stimulating the plant, helping absorb better nutrients, giving stress tolerance, adding value to your fruits and this thing, or improving the yield. If they are doing any one of this, you can call a biostimulant.
Yeah, my question is to will it act as a biostimulant? Suppose if we use it.
Yeah, I have to check what exactly is it doing.
No, it's also supplying the nutrients. So it's a dual.
Nutrient.
Yeah, it's a nutrient basically.
So
then
whenever
it's
it
not a
applies in the soil, I think
It supplies the nutrients.
whenever it applies in the soil, I think better we'll get the better results. Maybe it might be acting as a biostimulant also. Like
Yes, it could.
when you
See,
mentioned
yeah.
that potassium sulfate, I had that.
Could be. There are dual role. But if anything is contributing to nourishment of the plant, we generally don't call it a biostimulant. It comes under fertilizer, fertigation, or what you call. But only it is helping the plant overcome stress or better utilization of nutrients. That category is only biostimulants. In this way.
But I think potassium cyanate is more of like it's containing the nutrients. It's more classified as a nutrients, and still when compared to the traditional potassium, it's in a more reactive form. So that's why you're seeing the more results than the other potassium forms.
You are seeing good result with potassium.
Yes, definitely.
The
foliar-
uptake rate will be very high.
Foliar application, that is?
No, soil application.
And how frequent is that?
It may be like, see, when we are supplying the source of potash, so we can use that. Maybe once in a week or once in 10 days, depends on the plant requirement. Whenever we feel like potassium deficiency is there, so that time we can use, instead of using potassium nitrate or potassium sulfate, instead we can use this.
It's one of the nutrients, complex nutrient, containing 3 nutrients. So by using this supplement, it will supplement the many nutrient at a time. Means, so overall growth, especially in reproductive stage, it's very helpful for the plant.
Good. I learned something.
Nutrients. Sir, one more. So since we are and you are making us to understand since from the morning, we have gone through very well all your information. One thing is finally I want to know, we have to improve the below ground and one biostimulant, and for the improvement of above ground, means foliar. In combination or individual, please suggest us what are all the combination, because you told many for germination soil improvement or cell-free extract.
Yeah.
So we cannot use all because of cost and all those things. Right now the cost is high. So what all we can combine and use, and if any products available, please tell us. Otherwise it's a waste. Tomorrow again we are going and so which I have to use.
I'll do that.
Like that, again it will end up in question only, sir.
Yeah, okay. So the exact product, please give me 2 days. I will give you the whatever product names I know, which are having some good quality. This one, I can give that names.
With this, it will
I will
continue.
give you by Wednesday only. And like I said, if I recommend some names, it would be like I am promoting those companies.
No,
That is why I like to
it's
be
not
little
like that, sir. You should
generic.
not promote
That is my problem. And also I am.
That we will use as an experiment. And
Yes. Yes. Okay.
Yes.
Yes. So that is why I wanted to keep it this session very generic.
I heard whatever has been taught from the morning. So many biostimulants have been mentioned, and the company which is supplying me is doing almost all the biostimulants. That's why I had brought I had asked my people to bring the ingredients, so it is there. I will not tell the name of the company because I would be promoting, but I am buying from them.
Any questions from the?
See, one thing I can add is go with the companies who have the history of working with this from a long time. I don't want to take name. Coppert is one good company. Coppert. Coppert. See, Coppert has been in this industry for a long time. And I know a lot of people in that company who are working. I have never been associated with Coppert nor any business dealing. But what I know is the company people who are there, they know the science. Yeah, he's there. And then our Bhatt was there, no? Uday Bhatt. In the R&D. He was there. Yes. So Coppert is one company. It's a Netherlands company. Actually, the Netherlands is the heart of all the biostimulant research. There's a university called Wageningen University. And if you see the number of publications, Wageningen is leading there. That tells a lot about that country, where they have put, and it's a agriculture country, right? So so companies like Coppert, there is now even multinational companies, I don't recommend so much because, see, they are basically chemical companies. Since they have a big name, big customer base, they want to add biostimulant to their portfolio. But inherently, they are not biostimulant makers. Okay, so it is like that. On the other hand, companies like Coppert, they started with biostimulant, they became this thing, and then they come. Even UPL is also a big name. They also know about biostimulants. But if you ask me, do they have the knowledge of biostimulant as much as Coppert? No, they don't have. Coppert knows what biocontrol is, how to use baits, how plant hormones work, which biostimulants work, where are they sourcing from. They know, there is like that. So again, I don't want to contradict myself. One name I can tell you is like this. So coming back to your question, can you recommend one or two? Like I said, if you go by my recommendation today, please invariably include plant growth promoting microbes and humic acid regular application in your roots. Okay, that's it. And then for the foliar, you combine ascophyllum and then completely seaweed ascophyllum and then protein hydrolysates. Sir, ಏನೋ ನೀವು ಬರ್ಕೊಂಡ್ರೆ ಅನ್ಸುತ್ತೆ ಸರ್, ನನಗೆ ಮರ್ತೋಯ್ತು ಅದೇನೋ ಇದರಲ್ಲಿ ಅದೊಂದು ರೆಕಮೆಂಡೇಶನ್ ಇದೆ ನಂದು, ಇಟ್ಸ್ ಇನ್ ಮೈ ಸ್ಲೈಡ್. ಓಕೆ, ಸೊ ದಟ್ ಒನ್ ಯು ರೆಗ್ಯುಲರ್ಲಿ ಸ್ಪ್ರೇ ಫಾರ್ ದಿಸ್ ಒನ್. What is that? Ascophyllum? Gibberellic acid. Gibberellic acid, very low dose. Thank you. Gibberellic acid and then this one, protein hydrolysates, amino acids. Regularly every 20 days spray ಮಾಡ್ತಾ ಹೋಗಿ. ನಿಮಗೆ it should cover most of your stresses. It should prepare your plant for most of the stresses. If you have to summarize the whole day's event is into this one is add good quality humic acid, folic acid, fulvic acid and cell-free extract beda idu, cells, microbes to the soil, and then this combination of seaweed, gibberellic acid and then this thing at the time of flowering. And if you get these cell-free extracts from IHR, you can spray any time, any day, every time, no problem. Adu one background protection ಕೊಡ್ತಾ ಇರುತ್ತೆ. I think this is my recommendation for this one. If I had any horticulture farm, I would recommend this.
Okay, thank you for that. One more question from the Zoom. As winter is upon us, can you help us, field supervisor, schedule the type of stimulants to be used with respect to the crop stage to expect healthy plant growth and high yield and quality produce? Also discuss the compatibility of fungicides with the stimulants. The question is, the winter is upon, so now we are entering into the winter. This is the stage where we get the very less rate of growth plants. For that, recommend some biostimulants stage wise if you have.
For winter and
for the winter. See, generally in winter season, winter shocks, the plants will not grow as better as in the rainy season.
Generally, condition is that the day temperature and night temperature, the difference is more than 13 degrees Celsius, where majorly we found the nutrient uptake is very slow.
Okay.
So that's why we are observing some retarded growth when compared to the normal cycle. So, yeah. So maybe I think this anti-transparents will work, or PGPR.
To be honest, I don't have answer for this one. Because
The challenge is
I understand the real challenge. So if it is, I can guess, if nutrient mobilization is the issue,
Yes.
then you have to combine organic acids again. That is the only solution. The more the organic acids in your root zone, the better it is. And how do you increase? You increase the soil temperature, automatically the passive nutrient absorption will increase. How do you do that one? But soil temperature is low. So it is much bigger problems that biostimulants cannot.
But what?
Huh?
Yeah, that's what just I about to tell. But this problem is majorly we used to see in the earlier days. But after adapting this drip and mulch, so I think this is not like a
So I
major
think you
issue.
guys are much more equipped to answer this question, not me. But from biostimulant angle, I don't think there is any research. Biostimulants can confer cold resistance or nutrient mobilization in low temperature. That is far-fetched.
Any research findings on other crops, like in the cold countries, you have any?
There is a cold stress tolerance is there, that it will contribute to cold stress and all this. But whether it is practical and it can replicate in the field, that I don't know. See, whatever is done in the lab is not necessarily working in the field. That is for cell wall fortification and hardening of this one, preventing fungal cell wall, fungal mycelia to impregnate, all those things. This is like what you call fortifying your cell walls. That is more this thing.
Because in Tamil Nadu, now we are going in Tamil Nadu, so we are having more of downy mildew problem.
Okay.
Because one is paddy, another is so stress, temperature fluctuation is there. So there we are having a major problem of downy mildew. So in that case, can we use orthosilicic acid or And also, vermi wash, is there any research finding from your side? Can we use vermi wash for Gherkin as well as alopino?
No, sir, I don't know much about it. I have to be very honest with you. There are cold stress tolerant metabolites. And for example, what you said is salicylate is, orthosalicylate is one. That means you have to find if there is a seaweed extract which is making this product, then you can spray that one. That is how that extract is there. Or if there is any cell-free extract, some particular bacterial Bacillus subtilis strain V9613 is producing more of this metabolite, then you can make a cell-free extract and start spraying it. It's a natural way. Bioabsorption is much better than naturally spraying this one. So we have to go metabolite by metabolite to understand and where it is produced, then only we can. But cold stress, sorry, I haven't put too much mind into that one. We are staying in warm country, no? That's why.
But generally for
we use auxins for development of plant and root in length. Auxins is used for development of plant and root in length.
Yeah.
Zebralic and cytocanin for the laterals.
Lateral shoot and branches.
Yeah, so if we have a combination of this, then can we solve this problem of undergrowth due to cold condition to a certain extent?
Yeah, anybody answer? Please.
This is spray, no? You are spraying. Yeah, that is land application, but this is auxins and this one you give it by foliar, no?
No, but in the cold season, now what is your major issue, sir?
The growth will be little bit less.
Growth is less.
no, if the reason
One reason is because the soil temperature is low.
Okay. Theoretically, it should work.
And second reason is you have less sunlight
Yes.
during this season.
Yes.
So both the factors are there.
Sir, if you ask me, in that cold season, now I understood the problem. One is it is cold, daylight is low, and then the growth is limited. So there are three factors. If you can increase the, what is called, light by artificial lighting or something, that could be one solution. That is not problem. It's fine. Second, you said that because of the cold, they are not growing. By combining NAA, which is the analog of indole acetic acid, it is auxin. It is, if it is produced in the plant, it is called IAA. If you are applying it, it is in the form of naphthalene acetic acid. It is. Absolutely, it should work if you combine indole acetic acid, gibberellic acid, very, very moderate, very low concentration, because you are improving both the shoot length, number of branches, and even maybe 5 to 10% is the growth reduction in cold compared to others, or
More than that.
more than that.
It will be around 25% low.
Yes, but then I don't see any other solution other than this one. Maybe plant growth promoting bacteria, drenching it into the root can also help you, because they help in nutrient mobilization.
Which
What this
bacteria
gentleman
do you
just said
suggest?
now, in cold what happens, because the nutrient mobilization is slower. That is where you need phosphate solubilizing bacteria, mycorrhiza, lot of bacillus bacteria, which can produce organic acid and which can sequester iron, which can take a lot of nutrients and give it to the plant. So in the cold season, you ensure that your root has very good quality plant growth promoting bacteria, and on the foliar you spray this combination of low dose of gibberellic acid, indole acetic acid, and then probably any other CV acid. That should compensate for the slow growth of foliage. Again, it is root active. You made it by adding the PGRs. Yes, PGR means you add a lot of bacillus, one even mycorrhiza, and also this Bacillus megaterium. And it is like you are adding how much? 10 to the power 8. That is around 10 million spores per gram. Heavy dose. And they are multiplying inside.
These are available in the market?
Yes, sir. Yes, sir. IHR is a great place. They have to give you a lot of, they can give you a lot of plant growth promoting bacteria. I think you make additional application in winter through drip irrigation. More of bacteria, you drench it into the soil. They will do that. Commercial, they give. They have. I think there is a lot of research on Gherkin with the PGRs they have done. I think what is that? There is a madam called Sudha Lakshmi or something. She's responsible for technology transfer for companies and for products. And there are some incubating companies. Yes, and they will give you guidance to these incubating companies. So there could be one company making mycorrhizal. Yeah, exactly. Yes, I think so. Cold straws, sir. I think you fortify the root very well with plant growth promoting bacteria and mycorrhiza, all those things, so that they are producing good organ. And foliar spray, start spraying all this very low dose of combination of plant growth promoters and some seaweed extracts. It should have some effect.
Taken care, because the root is the main issues, especially in the cold. If you taken care with all these biostimulants with the roots, then the above part will be automatically, the natural biostimulants will.
More is good, sir. Diversity in the root. I was sitting with one, he's an Indian professor. His name is Venkateshan. He is in Pennsylvania University. And I went to him saying that, sir, here is our product, which is Bacillus subtilis. When we add it to the root, it will give some good tolerance. He said, okay, he's a very eminent professor. And he told me, so when, what is this? He said, okay, this is, now what do you want from me? I want to know what is this doing in the plant. Can you please do some research and give? We went to him. He said, so you are applying this one product to the roots and you are getting tolerance? Yes. He rejected the product. He said, this is wrong. I think what your company is doing is wrong. You will fail. Your product will fail. You will never succeed. He was right. Because you cannot dominate a root in the plant with one bacteria. Hundreds of bacteria are there. Hundreds of fungus are there. They all work like orchestra. One is playing drums, one is playing violin, one is playing this one. Then only the symphony will come. So what IHR is doing good thing is, IHR is promoting all these microbial consortia. So if you have to get good control, don't use single bacteria, use many bacteria, many fungus. Then you will have a broad spectrum thing. So one is going up, one is coming down, one is adapting, one is not adapting. One is doing good when salts are there, one is doing good when cold is there, one is doing hot is there. So you should have a diverse microbial community in the root, is what this is. Yeah, mycorrhiza, rhizobacterium. Exactly. So combining and spraying as many as good bacteria into the soil is much better than single. Don't use one strain and be happy. It will not work.
Any more questions from the backside? It's not only about biostimulants, even the pathologists and entomologists, soil scientists are there. You can throw any questions. It may be combined with biostimulants.
As all we know, GSB gummy template is a major problem in the coming season, mainly in Tamil Nadu. Gummy template, GSB. So it is a very, compared to down immunity we can control. Gummy template, people are saying go for drenching and all. Drenching and all is not helping really. Once plant grows after 15 days, even after 30 days, it is looking very good. After that, it is coming means within 15-20 days, it is completely gone. We have seen in several cases. So we need to have proper protocol to address the gummy template. You have solution?
I think pathologist has to answer. Probably with our available fungicides or whatever it may be, we don't have a proper control.
Yeah, only with your approved chemicals with available fungicides, and also for the gummy template, the hygiene is very, very important, because especially the pathogen is a very imperfect pathogen. The spores are spreading like anything, even can transfer through air as well. So that's why the field hygiene. So for example, if the leaf is infected, so if you see the laborers in the field, they will pluck the leaf and throw it in the other rows. They will not properly plucking and throw it or dumping in one place. The proper hygiene, and also it also spread through even the water from one plant to other plant. So overall, not only spraying the chemicals, the proper sanitization, field sanitization is very important for this gummy template.
Even we know this much severity of how the gummy template is severe. We don't know exactly where and when it appears. What I have observed in field is, actually gummy stem, there is no any infection on stem. There is a huge damage on the leaves. And even there is no infection on stem, leaves. There is an infection in fruit. Even I have not understood properly how this pathogen is infecting the plants. And we don't know when it appears and when it is more damaging. So right from the beginning, what sir told, with a lot of use of beneficial it's not a 1 or 2 days work. With a long time we have to work. So improving the soil beneficial microflora. So it can take care of the initial inoculum of the GSB. And in combination with some of the very good fungicides like not sporium, with Merivan. From BASF, we can also include in that. Along with that, initially we have to use some of the bioagents like trichoderma and Bacillomyces consortium before the initiation of the disease. Only options, these are the options we have, not too high.
Actually, the gummy stem blight earlier, when we saw the infection, it was on stem. Later, now onwards, it is more visible on leaf. Actually, earlier it was like soil-borne. Once it is from soil-borne, it infected to plant. It can spread through air also. It can spread through splashing of water also. So, maintaining the good sanitation, first thing, giving the proper spacing is one of the important thing. Since the adaptation of drip and mulch, the spacing is not much issue. And also avoid excess nitrogen application, because application of nitrogen increases the intensity of gummy stem blight. So once it starts on leaf, what the field staffs do, they go drenching to the stem. Infection is not there in the stem. So it's better to go for foliar spray when you see the V-shaped necrosis on the leaf. So it's better you can go for foliar spray, whatever the approved chemical is there. So the systemic will work good. So if you go for a foliar spray only, then you can avoid the spread through air. So it will be less infection of fruit will be, we can see. So we must reduce the application of nitrogen. It's also one of the main source for the spreading of gummy stem blight. And also we must prune the lower leaf to avoid the buildup of humidity. This will also help. So we must go with IPM practices, limiting the nutrients, and also with fungicides. And also bio-stimulants, you can go with Pseudomonas and Trichoderma at initial stage. This will help to avoid the soil-borne disease like even PTM and also gummy stem blight. And part of fusarium also can be avoided with Pseudomonas and all. So it's better we can go with IPM practices for especially for gummy stem blight. And also with border crop also, because one infection is there in one field, if the border crop is there, the spreading will be less through air. So this is the thing what we have observed and also discussed with our pathologist. So thank you.
Sucking pests. No, so verticillium also. Verticillium is fungi, but it has been recommended for the management of sucking pests. Now its name and its name also changed now. It's Lecanicillium. No more verticillium. Now it's called as Lecanicillium. And regarding GSB, initially you won't find infected with entire field will not. Here and there, 1 or 2 plants will be infected. So better when you identify the infection, try to avoid excess irrigation. When you notice initially GSB, irrigation water or irrigation management is very important. Don't do excess irrigation because spore will spread like anything. So today one plant, if the excess irrigation, within a 4 to 5 entire the plant, the row will die. So try to give as much as less irrigation. And when you notice the initial infection, try to drench properly to infected plant along with either side 5 plants to avoid the further spread. Proper drenching has to be done, infected and neighboring 5 plants to avoid the spread.
Hope you answered your question, I think. It's totally a total integrated management. It is very difficult for sure because it is infected from the soil, aerial, flowers, every part. Probably like what you said is priming the plants, or priming is most important here with what they recommended is that bacterial culture, they are going to have that priming of the plants because the good bacteria will going to be in the soil with more numbers, so that this can have a less advantage to infect, I think so.
I just want to add one thing is, I don't know, you are in Gherkin, I think you cannot use any pesticide, right? You are yes.
We can use which are approved in the EU regulation and
okay,
US regulations.
Maybe put it like you are restricted in this. So that means you should be the first people to adapt PGRs very, very extensively. And what he said, the word priming, you should always prime the plants with the root priming, foliar priming. Now he said, okay, all the abiotic stress priming, we talked a lot from morning. Root, you keep dumping these things every second day. We didn't talk about biocontrols. I think they are very good biocontrols also. So biocontrols can also be used not as prophylactic, it can also be used as preventive. So you keep spraying and it is not going to do any harm. Well, like you said, it will add a little bit to your cost, but since you are not using many pesticide, spray Trichoderma. Even Pseudomonas is there now. Good sprays, keep spraying them. So the fungus at least will be preventing, it will not come there because there is so much of trichoderma also already on the leaf. These new spores that are come, they won't like to grow there. There is competition inhibition. So you can prevent it. Prime, prevent, protect. That is should be the mantra here.
Messiana and this Verticillium lecani.
For insect.
All these three are the
ಅದಕ್ಕೇನೆ.
Yeah,
See
insects
that
guy.
we used to spray this to control this black thrips. So it was very effective for that. In fact, it will control mites ಸ್ವಲ್ಪ ಮಟ್ಟಿಗೆ ಆಗ್ಬೋದು. ಹೌದು.
Again, this one name comes to my mind is Coppert has done very good work in biocontrol. So I think they have some good solutions, proven solutions that could be also people to consider.
These biocontrol agents like Trichoderma, Pseudomonas and Bacillomyces, not only controlling the pathogens, they also, what sir told, no, they also act as biostimulant along with control of these pathogens. Then many research studies have been identified.
Trichoderma is very well proven to have positive effect in saline soil. And how it really provides salinity tolerance is still people are trying to investigate. But if you provide the right dose of trichoderma in the soil, in the root zone, in saline soil, that is the best saline control mechanism for plants. But you should get the trichoderma correctly, apply it right to the root. So they are underestimated, underrated organisms. We are, India was the first country to identify all these biocontrol agents, but we are also the last one to adapt it. So Westerners, Americans, they all, though they don't need it, they are adapting it faster. That's the irony of it. We started biocontrol much earlier than United States or Europe, anyone. So
we are also interested to use, but the availability of quality material and this tropical climatic condition, their efficiency and all those matters.
Any other questions apart from biostimulants? I think Vinod sir want to speak something.
We are having some issues with importing protein bait. Health certificate is not available with the supplier. So we took trials with the material supplied by Dr. C.B. Gowda. I think we have the results from two companies. One is from Coleman and another is from IAP. So in both the cases, the results are pretty good. And I think we can use that material. At the moment, I think he doesn't have much material. It might take some time. So what I suggest is, those who are already having protein bait, please wait for some time. Those who don't have anything can approach Dr. C.P. Gowda. I will post his card on the AgriHeads WhatsApp group. So you can contact him. We have discussed about the packaging. It's 1 liter packaging. So price is something below 100. Price is something below 100. So it will be it's a good product also, because we already tested in 2 places. So you can, those who don't have anything right now, please contact first. Those who have some stocks, please wait for some 15 to 20 days. So let us give the chance for the people who don't have the stocks. Mr. Allah, so it was a very enlightening training session what we had. We were all using a lot of biostimulants and whatnot, and we didn't know much about it. So Dr. C.B. Gowda gave us a quite big presentation to us, and now you people have to go and train your staff. So, I mean, this is a very vast subject, and I know it will be a little bit difficult to train initially. So I suggest you please take the help of Mr. Murthy, Karthik, Ayanna, then Pradeep, Nagaraj, whomever who can help you. So, because it's a very vast subject and it is very new to us. Otherwise, nutrient part, we could have trained our people, but this is a relatively new subject for us, and it is quite vast subject, and at the moment it is very technical. So maybe after 2 or 3 sessions, it will also become just like the nutrients. After 2-3 sessions, because this is the first session. So another 2-3, maybe 15 years, 20 years back, if you would have asked me about the nutrients, the same thing would have come out from me. So this is the first training. So we have to have a couple of more trainings. So initially, you please take the help of these people who are definitely willing to guide. I take it for granted from all of you. So please take the help of these people and try to educate the field staff, and let us make good use of this training program. So I think Mr. Ramesh is going to sum up. Mr. Munagada wants to speak a few words. Thank you.
Good evening to everybody. From the morning, I think with the advice and knowledge from Mr. Basavanagowda sir, we have learnt some few inputs towards the product development and crop improvement and quality towards using biostimulants and other things. And as Vinod sir was telling, it is a more of technical session, and we need lot of things to digest. And this training program will not stop here. We are going to conduct much more training sessions with some more experts. And as a IGA, we will continue to do lot of training programs year afterwards. And we expect more participation from the industry, and also we expect lot of learned friends or pathologists or some experts in the industry to contribute from now onwards and think seriously towards implementing at the ground level, and see that industry prosperous for another 30-50 years going forward. But you know that, we were telling since last 2-3 training sessions, 30 years we have passed with so many hurdles, and of course we are not having much hurdles as of now, but now a lot of challenges are coming with the production, marketing and competitiveness. So all those challenges have come. To overcome all these challenges, we need to be more cautious, more productive, more learning in the industry to overcome all these challenges. Good luck to everybody. Thank you.
So we almost coming to the end. Namaskara to everybody. Firstly, we have to thank our president for taking this whole program very strongly and persistently, told all the agri senior people, IGA senior companies, that we have to do this. First, thanks to Mr. Vinod sir, our president. Great thanks to Lakshmish, who was very, very proactive. Never he was like what he's done from the past few months. Huh? He was the one who ran the whole, from the past 4 months, I think. Lakshmish, really we thank you. It wasn't for you, we would not have done it. And again, thanks to you for getting a great, very confident Dr. Chennabaswana Gowda to us. Very, very confident. Fantastic person who you identified, who gave us quite a good of knowledge. Thanks to Stalwarts, Monegodu, very, very senior man, very senior man, who is here in a presence. And he's come for almost everything. Unfortunately, Secretary General Ravindra could not come. He had an health issue. Otherwise, I was supposed to picking up today. Otherwise, he was always used to come here every time. Thanks to Prakash Gaudra. Because of him, he's the one who funds us, and he never says no for anything. Immensely thank you, Doctor, on behalf of everybody. Okay? It was a fantastic discussion. Never expected. I never expected it to be so strong, so confident. I hope everybody agrees with me. Yes. We were very confident on the subject. We had zero knowledge on this. Thanks to NANIMS. Thanks to everybody on the dais, pathologists, entomologists, Nagraj, for being proactive. Thanks to Raiswan. Again, we'll conduct a lot of big programs. Raiswan is on the line to give us more information and major trainings also. Okay? So lastly, thanks to you all. ನೀವು ಬರಲಿಲ್ಲ ನೀಂಗೋ ವರ್ಲಂದೆ మీరు రాలేదంతే ఈ ప్రోగ్రామ్ లేదు కరెక్టా ನಾವು ಸಿ ಎಮ್ ಕಳಿಸ್ಲಿ ಪಿ ಎಮ್ ಕಳಿಸ್ಲಿ ನೀವು ಬಂದಿಲ್ಲ ಅಂದರೆ ಪ್ರೋಗ್ರಾಮ್ ಎಲ್ಲ ಆಗ್ತಿತ್ತು ಎಲ್ಲ ಪ್ರೋಗ್ರಾಮ್ಗೂ ಎಲ್ಲ ಕಂಪನೀಸು ಬಂದಿದ್ದಾರೆ ತುಂಬ ಧನ್ಯವಾದಗಳು ಆತ್ಮೀಯದ ಧನ್ಯವಾದ ಇಲ್ಲಾಂದರೆ ಈ ಪ್ರೋಗ್ರಾಮ್ ಆಗಲ್ಲ 5 ಜನ ಇದ್ಕೊಂಡು ನಾವೆಲ್ಲ ಇದ್ಕೊಂಡು ಮಾಡಿದರೆ ಏನಿದೆ ಏನೋ ಮಾತಾಡಿದ್ರು ಏನೋ ಹೋದ್ರು ಆ್ಯಂಡ್ ಇಟ್ಸ್ ಮೋರ್ ಇಂಪಾರ್ಟೆಂಟ್ ಇಸ್ how you all managed it. If you look at the same place, we started the first training program. Lakshmish organized, Vinod said we must do it. Trainer ಮಾಡಿದಾಗ ಇಲ್ಲಿ ಆವಾಗ how we all done it. Then the IGA we done for the IGAH. Then we should never forget our fruit fly now we are taking it so seriously. Great, Lakshman Gar, please stand up. We have to, we'll felicitate to Lakshman Raghu. It's not because their boss is the president. They really done a great job in doing the fruit fly, the baiting system, organizing. Very thanks from Bharati Associates, 2 of your staff who done that. Coleman Rajanna. Where's Coleman Rajanna? Coleman Rajanna. ತುಂಬ ಜನ ಗೊತ್ತಿಲ್ಲ ಅವರು ತುಂಬ ಫ್ರೂಟ್ ಬೇಟ್ ಬಗ್ಗೆ ಭಾರಿ ಓಡಾಡಿ ಇದು ಮಾಡಿದರೆ ಮತ್ತೆ ಅವರೊಬ್ಬರೇ ಅಲ್ಲ ಇನ್ನೂ ಕೆಲವರು ಬೇಕಾದಷ್ಟು ಜನ ಇದ್ದಾರೆ ತುಂಬ ಧನ್ಯವಾದಗಳು ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್ ಸಾಕಷ್ಟು ಟೈಮ್ ತೊಗೊಳ್ಳಲ್ಲ ಸರ್ ವಿ ಥ್ಯಾಂಕ್ ಯು ಇಮ್ಮೆನ್ಸ್ಲಿ ಆನ್ ಆಲ್ ಆಫ್ ಅಸ್ ನರಸಮೂರ್ತಿ ಅವರು ಆಲ್ ಆ ಪ್ರೋಗ್ರಾಮ್ಸ್ ಹಿ ಹ್ಯಾಡ್ ಕಮ್ ಟ್ರೈನರ್ಸ್ ಆ್ಯಂಡ್ ಹಿಸ್ ಕೊಲೀಗ್ಸ್ ಆಲ್ಸೋ ವೇರ್ ರಿಕ್ಷತ್ ಗೌಡ ರಿಕ್ಷತ್ ಗೌಡ ಥ್ಯಾಂಕ್ ಯು okay so now we will take time we have to a long time Lakshman Gowda was very very active in spite of his age so we request Lakshman from Bharati to come here please Lakshmish why don't Monegodu sir please come doctor ಬನ್ನಿ ಬನ್ನಿ ಗ್ರೇಟ್ ವರ್ಕ್ ಲಕ್ಷ್ಮಣ್ ಅವರೇ. ರಘು ಅವರೇ ದಯವಿಟ್ಟು ಬನ್ನಿ, ನೀವೇ ಬನ್ನಿ. ರಘು ಅವರೇ ಭಾರಿ ಕೆಲಸ ಮಾಡಿದಿರಲ್ಲ, ಇಂಥ ಬೇರೆಯವರು ಮಾಡಿದ್ದಾರೆ ಇಲ್ಲ ಅಂತಲ್ಲ, ನೀವು ಸಾಕಷ್ಟು ಫ್ರೂಟ್ ಫ್ಲೈ ಬಗ್ಗೆ ಎಲ್ಲ ಮಾಡಿದ್ದಾರೆ. ಪ್ರಕಾಶ್ ಗೌಡ್ರೆ ದಯವಿಟ್ಟು. ಥ್ಯಾಂಕ್ ಯು. ಕೂತ್ಕೊಳ್ಳಿ. ಲಕ್ಷ್ಮೀಶ್ ಬನ್ನಿ. ಥ್ಯಾಂಕ್ ಯು ರಘು ಅವರೇ. ರಾಜನ್ ಅವರೇ, ಕೋಲ್ಮ್ಯಾನ್ ಅವರು, ದಯವಿಟ್ಟು ವಿನೋದ್ ಸರ್ ಅವರೇ ದಯವಿಟ್ಟು ಕೋಲ್ಮ್ಯಾನ್ ಕಂಪನಿ ರಾಜನ್ ಅವ್ರನ್ನ, ಅವ್ರ ಕ್ಷಮೆ ಫ್ರೂಟ್ ಬೇಟ್ ಬಗ್ಗೆ ಭಾರಿ ಕೆಲಸ ಮಾಡಿದ್ದಾರೆ. ಧನ್ಯವಾದಗಳು ರಾಜನ್ ಅವರೇ. ರಕ್ಷಿತ್ ಗೌಡರೇ ದಯವಿಟ್ಟು ಬನ್ನಿ. ನಿಮ್ಮನ್ನು ಮರ್ತುಬಿಟ್ಟಿದ್ವಿ ಒಂದ್ಸರಿ, ಮರ್ತಿಲ್ಲ, ಮಿಸ್ ಮಾಡಿದ್ವಿ ತಪ್ಪಾಗಿ, ನಾನೇ ತಪ್ಪು ಮಾಡಿದೆ. ದಯವಿಟ್ಟು ಬನ್ನಿ. ಪ್ರೆಸಿಡೆಂಟ್ ಅವರೇ ಮುನೇಗೌಡ್ರೆ, ಕೊಡಿ ಬನ್ನಿ. ವಿನೋದ್ ಸರ್ ಕೂತ್ಕೊಳ್ಳಿ ಸರ್, ದಯವಿಟ್ಟು ಪ್ಲೀಸ್. ಥ್ಯಾಂಕ್ ಯು ರಕ್ಷಿತ್ ಅವರೇ. ನೀವು ಕ್ಷಮೆಗೆ ಆಲ್ ದ 3 ಪ್ಲೇಸಸ್ ಬಂದಿದ್ರೆ ನೆನಪಿದ್ದ, ದಾವಣಗೆರೆ, ಕೊಟ್ಟೂರು, ಎಲ್ಲ ಕಡೆ ಬಂದು ಎಲ್ಲ ಕೆಲಸ ಬಿಟ್ಟು ನಮಗೆ ಎತ್ತೇನು ಮಾಡಿದ್ರಿ. ಪ್ರಕಾಶ್ ಗೌಡ್ರೆ ದಯವಿಟ್ಟು CB ಗೌಡ್ರನ್ನ ಕರ್ಕೊಂಬನ್ನಿ. ದಯವಿಟ್ಟು ಕುಡಿ. Please accept this small momentum from in behalf of IGA and all of us. ಇದು ನಮ್ಮೆಲ್ಲರೂ ಪರವಾಗಿ, ನಮ್ಮ ಗರ್ಕನ್ ಸಿಬ್ಬಂದಿ ಪರವಾಗಿ ಕೊಡ್ತಾ ಇರೋದು. ಎಸ್, ಖಂಡಿತ.
ಓಕೆ, ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ನೀವೆಷ್ಟು ಥ್ಯಾಂಕ್ಸ್ ಕೊಡೋದಕ್ಕಿಂತ ನಾನು ನಿಮಗೂ ಅಷ್ಟೇ ಥ್ಯಾಂಕ್ಸ್ ಹೇಳಬೇಕು. ನಮಗೂ ತುಂಬ ಓದಿರ್ತೀವಿ, ರಿಸರ್ಚ್ ಮಾಡಿದ್ದೀವಿ, ನಮಗೂ ವೇದಿಕೆ ಬೇಕು ಇದೆಲ್ಲ ಹೇಳೋಕ್ಕೆ. ಆಮೇಲೆ ಇದರಿಂದ ಏನೋ ಒಂದು ಫಾರ್ಮಿಂಗ್ ಕಮ್ಯುನಿಟಿ ಅಥವಾ ಗ್ರೋವರ್ಸ್ ಕಮ್ಯುನಿಟಿಗೆ ಇಂಪ್ರೂವ್ ಆಗುತ್ತೆ ಅಂದರೆ ದಟ್ ಈಸ್ ದ ಬೆಸ್ಟ್ ಸ್ಯಾಟಿಸ್ಫ್ಯಾಕ್ಷನ್ ಎನಿ ಸೈಂಟಿಸ್ಟ್ ಕ್ಯಾನ್ ಗೆಟ್ ಇದು. ಸೊ ಆ್ಯಂಡ್ ಲಕ್ಷ್ಮೀಶ್ ಆ್ಯಂಡ್ ವಿ ಆರ್ ಲೈಕ್ ಸೋ ಕ್ಲೋಸ್ ಫ್ರೆಂಡ್ಸ್. ಅವರು ನಮ್ಮ ಸೀನಿಯರು, ನಮಗೆ ಮೆಂಟರು ಇದರಲ್ಲಿದ್ದಾಗ ಕಾಲೇಜಲ್ಲಿಂದ ಇವರು. ವಿ ಆಲ್ಸೋ ಲುಕ್ ಅಪ್ ಟು ಹಿಮ್ ಆ್ಯಸ್ ಅವರ್ ಸೀನಿಯರ್ ಆ್ಯಂಡ್ ಗೈಡಿಂಗ್ ಫೋರ್ಸ್. ಆ್ಯಂಡ್ ಇದು ಆರ್ಗನೈಸ್ ಮಾಡಿದ್ದಕ್ಕೆ ಥ್ಯಾಂಕ್ ಯು ಲಕ್ಷ್ಮೀಶ್, ಪ್ರೆಸಿಡೆಂಟ್ ಸರ್, ಮುನೇಗೌಡ ಸರ್, ಎವ್ರಿಬಡಿ, ಆಲ್ ಯುವರ್ ಕಮಿಟಿ. ಸೊ ಇದು ಮಾಡಿ. ಆ್ಯಂಡ್ ಐ ವಿಲ್ ಬಿ ವೆರಿ ಹ್ಯಾಪಿ ಟು ಇನ್ನೂ ಎಕ್ಸ್ಟೆಂಡ್ ಮಾಡೋದಕ್ಕೆ ನಿಮಗೆ. ಇನ್ನೂ ಬೇಕು ಅಂದರೆ ನಾನು ವಿ ಕ್ಯಾನ್ ಡೂ ಸಮ್ ಫೀಲ್ಡ್. ಇದು ವಾಟ್ ಐ ವಾಸ್ ಟ್ರೈಂಗ್ ಟು ಪ್ರಪೋಸ್ ಈಸ್, ಸೊ ನನಗೆ ಇನ್ನೂ ಕೆಲವರು ಎಕ್ಸ್ಪರ್ಟ್ಸ್ ಗೊತ್ತು ಹೂ ಆರ್ ಫ್ರಮ್ ಕಂಪನೀಸ್. ದೇ ಕ್ಯಾನ್ ಟ್ರೈನ್ ಯು ಹೌ ಥಿಂಗ್ಸ್ ಆರ್ ಅಪ್ಲೈಡ್. ಸೊ ನೀವೆಲ್ಲಾದರೂ ಗರ್ಕಿನ್ ಫೀಲ್ಡಲ್ಲಿ ನಮ್ಮನ್ನು ಇದು ಮಾಡಿದ್ರೆ ಐ ವಿಲ್ ಬಿ ವೆರಿ ಹ್ಯಾಪಿ ಟು ಬ್ರಿಂಗ್ ದೆಮ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ PGPR ಹೆಂಗೆ ಮಿಕ್ಸ್ ಮಾಡ್ತಾರೆ, ಸೊ ಹೆಂಗೆ ಹಾಕಬೇಕು, ಆಮೇಲೆ ಫೋಲಿಯರ್ ಸ್ಪ್ರೇ ಹೆಂಗೆ ಮಾಡ್ತಾರೆ. ಸೊ ವಿ ಕ್ಯಾನ್ ಟೇಕ್ ಇಟ್ ಒನ್ ಸ್ಟೆಪ್ ಹೈಯರ್. ಆ್ಯಂಡ್ ಐ ವಿಲ್ ಬಿ ವೆರಿ ಹ್ಯಾಪಿ ಟು ಡೂ ದ್ಯಾಟ್ ಫಾರ್ ಯು, ಟು ಕಂಡಕ್ಟ್ ಅನದರ್ ಟ್ರೈನಿಂಗ್ ಸೆಷನ್ ಇನ್ ದ ಫೀಲ್ಡ್. ನೀವೆಲ್ಲ ಗರ್ಕಿನ್ ಫೀಲ್ಡ್ಸ್ ಹೇಳಿದ್ರಲ್ಲ ಸರ್, ಅಲ್ಲಿ ಮಾಡಿಬಿಡು. ಸೊ ಐ ವಿಲ್ ಬಿ ವೆರಿ ಹ್ಯಾಪಿ, ಸೊ ದಟ್ ಯು ಗೆಟ್ ಟ್ರೈನಿಂಗ್, ನಾಲೆಜ್ ಆ್ಯಂಡ್ ಟ್ರೈನಿಂಗ್, ಆ್ಯಂಡ್ ದೆನ್ ಯು ಆರ್ ಆಫ್ಟರ್ವರ್ಡ್ಸ್ ಯು ಆರ್. ಸೊ ಥ್ಯಾಂಕ್ ಯು ಗರ್ಕಿನ್ ಅಸೋಸಿಯೇಷನ್, ರಿಯಲಿ ವೆರಿ ಗುಡ್ ಇನಿಷಿಯೇಟಿವ್ ನೀವು ಇಷ್ಟು ತಗೊಳ್ಳೋದು. ದಿಸ್ ಇಸ್ ವೆರಿ ಮಚ್ ರಿಕ್ವೈರ್ಡ್ ಫಾರ್ ದಿಸ್. I appreciate all of you and thank you for this opportunity.
ಥ್ಯಾಂಕ್ ಯು ಬಸವನಗೌಡ್ರೆ. ಆಮೇಲೆ ಕೊನೆಗಾಗಿ ಈ ಜಾಗ ಕೊಟ್ಟವರು ಅವ್ರಿಗೂ ಥ್ಯಾಂಕ್ಸ್ ಹೇಳಬೇಕು ಯಾಲು. ಆಮೇಲೆ ನಮ್ಮ ಸ್ಟ್ರೀಮಿಂಗ್ ಮಿಸ್ಟರ್ ಜಗದೀಶ್ ಅವರೇ ತುಂಬ ಧನ್ಯವಾದಗಳು ಎಲ್ಲರ ಪರವಾಗಿ. ನೀವು ಸ್ಟ್ರೀಮಿಂಗ್ ಮಾಡೋದು, ನಿಮ್ಮ ಸೌಂಡ್ ಸಿಸ್ಟಮು, ನಿಮ್ಮ ಕ್ಯಾಮ್ರಾ. ಈಗ ನಮ್ಮ ಟ್ರೆಷರರ್ ಆದ ಇದು ಲಕ್ಷ್ಮೀಶ್ ದಯವಿಟ್ಟು ಕಂಟಿನ್ಯೂ. ಥ್ಯಾಂಕ್ ಯು, ಧನ್ಯವಾದ.
ಥ್ಯಾಂಕ್ ಯು ರಮೇಶ್ ಸರ್. ಈಗ ಪಾರ್ಟಿಸಿಪೇಷನ್ ಸರ್ಟಿಫಿಕೇಟ್ಸ್ನ ವಿತರಣೆ ಮಾಡಲಾಗುತ್ತೆ. ಆ್ಯಂಡ್ ಹಂಗೆ ಅದೇ ಥರ ಒಂದು ಗ್ರೂಪ್ ಫೋಟೋ ನಿಮ್ಮ ಕಂಪ್ನಿ ಪರವಾಗಿ ಬಂದುಬಿಟ್ರೆ ಅಧ್ಯಕ್ಷರು ಮತ್ತು ಉಪಾಧ್ಯಕ್ಷರು ಟ್ರೆಷರರು ರಮೇಶ್ ಸರ್ ನೀವು ಬಂದುಬಿಡಿ. ಸೊ ಅಲ್ಲ ವಿ ಆಟ್ ಅ ಗ್ರೂಪ್ ಫೋಟೋ ಫಾರ್ ಈಚ್ ಕಂಪನಿ. ಹಾಂ, ಗುಡ್ ಗ್ರೀನ್ಸ್ ಪ್ಲೀಸ್ ಕಮ್. ನೆಕ್ಸ್ಟ್ ಪ್ರಚಾರ ರಾಜ ದಿ ನೆಕ್ಸ್ಟ್. Somebody help me to take photos if possible. Yes. Raja, Prashara. Morris Roy. Morris. Sorry, sir. Team VRD, both people can come. If you are 3 people, you 3 people come. No problem. Suresh Dalavai and team. Next. All people can come. Suresh Dalavai and team. Suresh Dalavai and team. Tulsi and team. Tulsi and team. Tulsi and team. Tulsi and team ಆದಮೇಲೆ ನಕ್ಷತ್ರಿ and team.
Active person. Becoming more and more active.
I wanted to see more dance. Nakshatri and team. Permission team, please come. Permission team, exotic. Dayanand Sagar and team. Team Bunny. We'll have a group photo afterwards, all the group members. Dayanand Sagar and team, please come forward. Sir. Next is Bharati team, Lakshman Gura and team, please come forward. The team of Bharati should come, all members. Vegola team, please come forward. Vegola, Malaya and Sunil, come forward. Unipic team, Shiroran team. Vishnuvardhan Reddy team, please come forward. Vishnuvardhan Reddy. Kiran team, your team members, which floor? Idagli. Loganathan and team next. Loganathan and team. Yeah, be there. Okay. Loganath. Pradeep Sajjan and team. Team illa obbre bandidare. Pradeep Sajjan. Ken Agritech team, Nagraj and Nanjan Mur. ಬನ್ನಿ ಬನ್ನಿ ನಂಜನಮೂರ್ತಿ, ವಿಡಿಯೋನೇ ಮಾಡಿಸ್ತಿದ್ದೀವಿ ಬನ್ನಿ, ಡೋಂಟ್ ವರಿ. ನಿಮ್ಮ ಫೋಟೋ ಎಲ್ಲ ಹೋಗ. ರಮೇಶ್ ಸಿ ಬ್ಲಾಸಮ್ ಶವರ್ಸ್ ಅಂಡ್ ಟೀಮ್. ರಮೇಶ್ ಎಕ್ಸ್, ವೇರ್ ಇಸ್ ರಮೇಶ್? ಓಕೆ, ಹಾಂ, ಅವ್ರ ಮಾರ್ನಿಂಗ್ ಅಟೆಂಡೆನ್ಸ್ ಅಷ್ಟೇ. ನೆಕ್ಸ್ಟ್ ದಿ ಯಂಗ್ ಅಂಡ್ ಎನರ್ಜೆಟಿಕ್ ಸಂಜೀವ್ ಕಟ್ಟಿ.
ಚತುರ ವೆಗೋಲ ಚತುರ ಲಕ್ಷ್ಮೀಶ್ ಅವರಿಗೆ ಮುನೇಗೌಡರ ಕಡೆಯಿಂದ ಸರ್ಟಿಫಿಕೇಟ್. ಪುಟ್ಟಣ್ಣ ಕೃಷಿ. ಮಧು
ತುಂಬ ಧನ್ಯವಾದಗಳು.
ಹರೀಶ್ ಗೌಡ್ರು. ನರಸಿಂಹಮೂರ್ತಿ ಅವರು ನನೆಮ್ಸ್.
ನರಸಿಂಹಮೂರ್ತಿ ಅವರೇ ನನೆಮ್ಸು. ರಕ್ಷಿತ್. ಬನ್ನಿ.
ಕಾರ್ತಿಕ್, ರಿಜ್ವಾನ್.
Vote of thanks by Prakash Gowda sir.
ನೋಡಿ, ಮೈಕ್ ಯಾವಾಗಲೂ ನನಗೆ ಲಾಸ್ಟಲ್ಲೇ ಕೊಡ್ತಾರೆ ಲಕ್ಷ್ಮೀಶ್ ಅವರು. ಯಾಕೆ ಹೇಳಿ? ಕಡೆಯಲ್ಲಿ ಕ್ಯಾಶ್ ಕೊಡ್ಬೇಕಲ್ಲ, ಲಾಸ್ಟಲ್ಲೇ ಕೊಡ್ತಾರೆ ಮೈಕ್. ಓಕೆ, ಜಸ್ಟ್ ಫಾರ್ ಜೋಕ್ಸ್. ಸೊ ಈ ದಿನದ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಬೆಳಗ್ಗೆಯಿಂದ ಸಂಜೆವರೆಗೂ ಸಮಚಿತವಾಗಿ, ಬಟ್ ಟುಡೇಸ್ ಪ್ರೋಗ್ರಾಮ್ ಈಸ್ ವೆರಿ ಫ್ಯಾಂಟಾಸ್ಟಿಕ್. ಫಸ್ಟ್ ಟೈಮ್ ಐ ಥಿಂಕ್ ನನ್ನ ಒಂದು 34 ವರ್ಷ ಸರ್ವೀಸಲ್ಲಿ ಈ ಥರ ಟೆಕ್ನಾಲಜಿ, ಮತ್ತು ನನಗೂ ಕೆಲವು ಐಟಮ್ಸ್ ಗೊತ್ತಿರಲಿಲ್ಲ, ಹ್ಯೂಮಿಕ್ ಆ್ಯಸಿಡ್ ಬಗ್ಗೆ ಆಗ್ಬೋದು, ಇದೆಲ್ಲ ಆಗ್ಬೋದು. ಅದನ್ನು ತಿಳಿಸ್ಕೊಟ್ಟಂಥ ನಮ್ಮ ಸಿ ಬಿ ಗೌಡಜಿಯವ್ರಿಗೂ, ನಮ್ಮ IGA ಮತ್ತು ನಮ್ಮ ಇಂಡಸ್ಟ್ರಿ ಪರವಾಗಿ ಹೃತ್ಪೂರ್ವಕ ವಂದನೆಗಳನ್ನ ಅರ್ಪಿಸ್ತಾ ಇದ್ದೀನಿ. ಹಾಗೂ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಮುನ್ನಡಿ ಬರುತ್ತಾ ನಮ್ಮ IGA ಅಧ್ಯಕ್ಷರಾದ ವಿನೋದ್ ಸರ್ ಅವ್ರಿಗೂ, ಮತ್ತು ಮುನೇಗೌಡ ಸರ್ ಅವ್ರಿಗೂ, ಮತ್ತು ಆಫೀಸ್ ಬೇರರ್ಸ್ ಅವ್ರಿಗೂ ನಮ್ಮ ಅಸೋಸಿಯೇಷನ್ ಪರವಾಗಿ ವಂದನೆಗಳನ್ನ ಅರ್ಪಿಸ್ತಾ ಇದ್ದೀನಿ. ಹಾಗೂ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಎಂಟಮಾಲಜಿಸ್ಟ್, ಎಲ್ಲ ಕಂಪ್ನಿ ಎಂಟಮಾಲಜಿಸ್ಟು, ಮತ್ತು ನನೆಮ್ಸ್ ಕಡೆಯಿಂದ ಆಗ್ಬೋದು ಅಥವಾ ರಿಜ್ವಾನ್ ಕಡೆಯಿಂದ ಆಗ್ಬೋದು, ಅವರು ನಮ್ಮ ಕರೆಗೆ ಒಗ್ಗಟ್ಟು ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಬಂದು ತಮಗೆ ತಿಳಿದಿರ್ತಕ್ಕಂಥ ಟೆಕ್ನಿಕಲ್ ಪಾಯಿಂಟ್ಸ್ನ ತಿಳಿಸ್ಕೊಟ್ಟಿರ್ತಾರೆ. ಅವ್ರಿಗೂ ನಮ್ಮ IGA ಪರವಾಗಿ ವಂದನೆಗಳನ್ನ ಅರ್ಪಿಸ್ತಾ ಇದ್ದೀನಿ. ಹಾಗೂ ಮುಖ್ಯವಾಗಿ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಪಿಲ್ಲರ್ಸ್ ಅಂತ ಹೇಳ್ಬೋದಾದಂಥ ನೀವೆಲ್ಲಗಳು, ಅಂದರೆ ನಮ್ಮ ಫೀಲ್ಡ್ ಅಗ್ರಿ ಗೈಸ್ ಆಗ್ಬೋದು, ಕೆಲವೆಲ್ಲ ಯೂಟ್ಯೂಬಲ್ಲೂ ವಾಚ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ, ಅವ್ರಿಗೂ ಅಷ್ಟೇ ನಮ್ಮ IGA ಪರವಾಗಿ ವಂದನೆಗಳನ್ನ ಅರ್ಪಿಸ್ತಾ ಮುಗಿಸ್ತಾ ಇದ್ದೀನಿ. ಥ್ಯಾಂಕ್ ಯು.
ಥ್ಯಾಂಕ್ ಯು ಪ್ರಕಾಶ್ ಗೌಡ್ರೆ. ಅದೇ ರೀತಿ ನಮಗೆ ಸರ್ವಿಸ್ ಕೊಟ್ಟಂಥ ಎಲ್ರಿಗೂ, ಸರ್ವಿಸ್ ಪ್ರೊವೈಡರ್ಗೂ ನಾವು ಧನ್ಯವಾದಗಳನ್ನ ಅರ್ಪಿಸ್ತೀವಿ. ಎಸ್ಪೆಷಲಿ ಫಾರ್ ಜಗದೀಶ್ ಅವರಿಗೆ ನಮಗೆ ಯೂಟ್ಯೂಬ್ ಸ್ಟ್ರೀಮಿಂಗು ಮತ್ತು ಝೂಮ್ ಸ್ಟ್ರೀಮಿಂಗ್ನ ಲೈವ್ ಮಾಡಿಕೊಡ್ಬೇಕು ಅಂತ ಕೇಳಿದಾಗ ಎಸ್ ಟೇಕನ್ ಪರ್ಸ್ನಲ್ ಇಂಟ್ರೆಸ್ಟ್ ಆ್ಯಂಡ್ come forward to do all this. That's why we thank you so much. ಈಗ ಒಂದು ಸಣ್ಣದಾಗಿ ಒಂದು ಗ್ರೂಪ್ ಫೋಟೋ ಎಲ್ಲರೂ ತೊಗೊಳ್ಳೋಣ. ಎಲ್ಲ ಇಲ್ಲಿ ವೇದಿಕೆ ಹತ್ರ ಬಂದುಬಿಟ್ರೆ ವಿ ಕ್ಯಾನ್ ಟೇಕ್ ಎ ಗ್ರೂಪ್ ಫೋಟೋ. ಸ್ಮಾಲ್ ಅರೇಂಜ್ಮೆಂಟ್ಸು ಸ್ವಲ್ಪ ಹೆಲ್ಪ್ ಮಾಡಿ. ಯು ಕ್ಯಾನ್ ಟೇಕ್ ದಿಸ್ ಟೇಬಲ್ ಔಟ್. ಚೇರ್ ಇರಲಿ, ಚೇರ್ ಇಲ್ಲಿ ಇಲ್ಲಿ ಬಂದು ಇಲ್ಲಾಕಿ ಇಲ್ಲಾಕಿ ಅಲ್ಲ ಕೆಳಗೆ ಕೆಳಗೆ ಇಲ್ಲಾಕಿ. ಹಾಂ ಅಲ್ಲೇ ಇಲ್ಲಾಕಿ ಸಾಕು, ಮೇಲ್ಗಡೆ ನಿಂತ್ಕೋಬೋದು. ಹಾಂ ಅರುಣ್ ಅವ್ರೆ, ಅರುಣ್ ಅಲ್ಲ, ಅರುಣ್ ತಾನೇ ನಿಮ್ಮ ಹೆಸರು? ಚೂರು ಫೋಟೋ ತೆಗಿಯೋಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತೀರಾ?